Morning, sir. Dr. Ravi here.
Morning, good morning.
Very good morning. Very good morning, everyone. We'll start our session in a few minutes. Thank you.
Sorry, ma'am, you're not audible to us.
Am I audible now, ma'am?
Yeah, now it's better.
Thank you. A mesmerizing morning, everyone. I welcome you all to the AlliedCon International Multidisciplinary Conference on Health Science on 29th and 30th March 2024. Organized by BioLeaks and our academic partners, Benevolentia Foundation, Chettinad Academy of Research and Education, and Parul University. BioLeaks. BioLeaks is a non-profitable professional association which prominently promotes research and development. BioLeak conferences bring together the professional wizards and leaders who have explored all the avenues to reinforce the field of life sciences and medicine technology. BioLeaks conducts events worldwide, which helps in enhancing the skill set of people and diverse industries, and also form a common platform for eminent personalities, physicians, researchers, doctors, and academicians, professional business figures, and much more. BioLeak conferences encourages better comprehension about improvements and progression over the world through worldwide conferences with the speed of science and technology. We work with our motto of creating a better tomorrow by organizing conferences and creating a network which help grow a better tomorrow with the help of advanced technology and achieve. AlliedCon 2024 is a multidisciplinary program with broad participation of members from around the globe focused on learning about various facets of allied health sciences. AlliedCon 2024 will create an excellent international forum for sharing knowledge and result in alternative health, family medicine health, allied health science community, and public health. The event focuses on providing the participants from diverse fields with unique and regional opportunities to make a name for themselves. This event's focus is all about retaining a passion for science and technology through the provision of one-of-a-kind solution, cardiac technology, perfusion technology, medical lab technology, neuroscience technology, renal dialysis technology, radiography, radiotherapy technology, nuclear medicine technology, trauma care management, physician assistant, optometry, forensic science, echocardiography, nursing, surgical and emergency aid, forensic science, nutrition, dietetics, along with many other interesting topics will be covered at the AlliedCon conference. I would like to welcome all our honorable dignitaries, Dr. Priyadarshini Shanmugam, Professor and Head, Chettinad Academy of Research and Education, India. Dr. Priyankesh Mishra, Benevolentia Foundation, Faculty, Delhi Pharma Medical and Management Institute, Behela, India. Dr. Sri Mahapatna, Assistant Professor, Parul University, India. And I would like to introduce our keynote speakers of today, Professor Dr. Jugsharan Singh, Professor, Department of Microbiology, University of Delhi, India. Professor T.S. Dr. Mohammad Nasir Mohammad Desha, Head of Department, Department of Biomedical Sciences, Faculty of Medicine and Health Sciences, University, Putra, Malaysia, Malaysia. Dr. Jitin C. Saji, Senior Specialist, ENT and Head and Neck Surgery, Aster DM Healthcare, India. Dr. A.V. Srinivasan, Emeritus Professor, the Tamil Nadu Dr. MGR Medical University, former Head and Professor of Neurology, Institute of Neurology, Madras Medical College, India. Dr. Bhavana Gadbhar, Dean Principal, Professor, Pearl Institute of Physiotherapy, Pearl University. And I would like to introduce our session chairs of today. Main Hall Day 1, Dr. Sinchan Das, Director, Homeopathy and Psychiatry, Benevolentia Foundation, India. Dr. Priyadarshini, Professor and Head, Microbiology, Chettinad College, India. Main Hall Day 1, Dr. Nancy Vaibhav Gandhi, Assistant Professor, Physiotherapy, Sumandi Vidyapeeth, deemed to be University, India. Dr. Riyaz Sharif, Professor, Chettinad College, India. And Hall AD1, Dr. Ravi Jyotikant Shah, Associate Professor, Adult Neurophysiotherapy Department, the Sarvanjanik College of Physiotherapy, India. Dr. Dharman Vyas, Assistant Professor, Pearl University, India. Hall B, D1 professors are Dr. Ravi Jyotikant Shah, Dr. Ambujavalli BT, Associate Professor, Microbiology, Faculty Medicine, Chettinad Hospital and Research Institute, Chettinad, India. Dr. Uma Maheshwari, PK, Associate Professor, Department of Microbiology, Medicine, Chettinad Hospital and Research Institute, Chettinad, India. Now, I would like to welcome Dr. Priyadarshini Shanmugam, Professor and Head, Chettinad Academy of Research and Education, India, to give the inaugural message. Ma'am, over to you, please.
Thank you, ma'am. Can you hear me?
Yes, ma'am, you're audible.
Okay. Good morning to the esteemed speakers, delegates, and academic partners. On behalf of Chettinad Hospital and Research Institute, which is a unit of Chettinad Academy of Research and Education, Tamil Nadu, India, it is my honor and privilege to extend a heartfelt welcome to each and every one of you to the Allied Con, which is an international multidisciplinary conference on health sciences. Today is a significant milestone as we gather here with a total of 250 participants. And we are also going to have 91 oral presentations and poster presentations and 2 interesting video presentations. We are particularly grateful to our esteemed academic partners whose unwavering support and collaboration has helped us to organize this conference at an international level. Our partners are BioLeagues, Benevolentia Foundation, and Parul University. And we thank you again. Your commitment to academic excellence and advances in health sciences and allied health sciences is truly commendable, and we are honored to have you as an integral part of this event. This conference serves as a platform for the exchange of groundbreaking ideas, cutting-edge research, and transformative insights across various disciplines. And it's a testament to the collective dedication and passion of every one of the participants who's gathered here. And please make use of this conference to delve into a diverse array of topics, okay? And then engage in thought-provoking discussions, and then try to foster meaningful connections across these various geographical boundaries. And together we can explore new frontiers, we can address the pressing challenges, and pave way for innovative solutions, which can help to shape our future more positively for all of us, as well as for our next generation. Once again, I extend my warmest welcome to all of you, and may this conference be a source of inspiration, enlightenment, and collective progress. Thank you.
Thank you, ma'am. Now I request Dr. Priyankesh Mishra, Benevolentia Foundation, India, to address the gathering. Over to you, sir.
Good morning to estimated speakers, delegates, and academic partners. On behalf of Benevolencia Foundation, it is my honor and privilege to extend a heartfelt welcome each and everyone to the AlliedCon International Multidisciplinary Conference on Health Sciences today. That marks a significant milestone with a total of 250 participants, including 91 oral and poster presentations, as well as 2 compelling video presentations. We appreciate the initiative of BioLeaks and other academic partners, Chettinad Academy of Research and Education, as well as Parul University, in providing such extraordinary platform to the students of allied health sciences, in propagating such extraordinary platform to the students of allied health sciences, in propagating scientific exploration in the act of mankind. Similarly, Benevolencia Foundation, founded in 2013 and registered in 2021, has been working in different fields of health and social parameters through research, scientific programs, peripheral medical camps, cultural events, and many more in multidisciplinary approach. Additionally, in these 2 days, various speakers will even bound together with multidisciplinary aspect from the studies of microbiology, pharmacology, drug formation to the application of drugs and their studies on modus operandi for varied differential diagnosis of diseases in current scenario. Furthermore, this initiative of multidisciplinary conference will ignite a flame in the hearts and mind of nascent researcher who should showcase and focus on differential progression of science in social structure. Hence, being one of the research scholar of esteemed organization Benevolencia Foundation, we feel proud and honored to receive the opportunity to delve into a diverse array of topics, engage in thought-provoking discussion, and foster meaningful connection that could transcend new boundaries for the development of healthcare and healthy living. Thank you.
Thank you, sir. Now I would like to welcome Dr. Simra Patan, Assistant Professor, Parul University, India, to address the gathering. I guess we are not having ma'am online. Now I would like to welcome our eminent keynote speakers of the day, Professor Dr. Jugsharan Singh, Professor, Department of Microbiology, University of Delhi, India. Sir, over to you, please.
Thank you very much. Well, it's my pleasure to be here delivering my talk in the BioLeaks Worldwide Conferences. Very good morning to everybody. Although it is dead of night in the Canada, from where I'm speaking right now. See, the topic for today's discussion here, I would like to present my slides first. And can you see my slides?
Yes, sir.
Yes,
All
Doctor,
right.
you can see your slides.
All right. So, see, the topic for today's conference is the challenge of the antimicrobial resistance. just to orient the participants. Hello. Hello.
We request all the other participants to kindly mute yourselves while the speaker is giving his presentation. Thank you.
So, you see, just to orient the diverse audience towards antibiotics, I'll just give you a small introduction. See, antibiotics, they were introduced in human medicine in early 30s to treat bacterial infections. And within, you can say, within no time, within days, rather from the first day when penicillin was used for certain wounded soldiers, these antibiotics proved to be wonder drugs. As these antibiotics, they were introduced further into the medicine, we found that these were responsible for an unprecedented advancement in the human medicine. Means not only that we were able to cure a variety of infections and infectious diseases by the use of the antibiotics, but these antibiotics also made human beings capable, or human medicine capable, of introducing very invasive kind of sophisticated surgeries, like surgeries of the bone, surgeries of the heart, lungs, brain. See, something which could not be attempted earlier, or these were being attempted in the medicine, but the success rate was not very high, primarily because the patients would suffer from an infection and die. So, with this, you can see, as the antibiotics were introduced, there was a lot of advancement in the human medicine. All these sophisticated interventions in the human medicines could now be possible. More and more, for example, more and more complicated and sophisticated surgeries could now be performed. As the time progressed, see, the antibiotics, they were used for veterinary medicine also. Of course, they were introduced in the veterinary medicine just to treat infections in the animals. But besides that, they were also introduced into animal husbandry, into agriculture, into aquaculture, although they were not meant for that. But because they were being applied there, that means they were applicable there. As they were microbes, microbes were to be treated, they were to be tackled, so they were introduced. But in certain practices like animal husbandry, they were not used for killing microbes, but rather for weight gain in animal food animals. What was the net effect of that? The net effect was the widespread use of the antibiotics. This widespread use of the antibiotics meant that the microbes, they were being exposed excessively to these antibiotics. And as these were antibiotics were being used for microbes, the microbes started retaliating. See, it is in nature. They started using their dormant pathways in which they could tackle the antibiotics in a variety of ways. They could throw the antibiotics out. They could microbes could kill the could hydrolyze the antibiotics, could modify their target sites. So that led to the emergence of the antibiotic resistance. Those microbes which were being killed by the antibiotics, they were now resistant. So this resistance was seen as early as 1950s or 60s, but then this resistance continue to increase all over the globe. See, and as medical practitioners, they did not pay much attention to this because always new antibiotics they were coming. Yeah, just before I go into that, so this word AMR, antimicrobial resistance, or antibiotic resistance, or multiple drug resistance, or multiple drug resistant organisms, basically it means same thing. It is just a question of semantics here. So, but in the current use, the AMR, antimicrobial resistance, is the terminology which is currently the most acceptable and conveys the proper meaning, and this is being used all over the globe right now. Let's come back to the AMR. This successive use, or rather I would say it was a misuse of the antibiotics. They were being used for purposes and in animal husbandry or aquaculture where they were not required. Okay, they like food supplements, agriculture. See, this excessive use or misuse was responsible. Not only that, they were being excessively used in human beings also. They were rather being misused in human beings also. Okay, all over the globe, people would take antibiotics for even small infections or minor infections like throat infection or a running nose, whether the antibiotic was required or not. So this was again a misuse even by the human beings. Even doctors, the medical practitioners, when they were trying to treat their patients, they were using the most advanced kind of antibiotics. So more and more these antibiotics, they were exposed to the microbes, either in nature or in the human beings, or because everything at the end is ending up in the waters, in the waste waters, whether it is coming from the hospitals or human or the patients. It's get into water, okay, through agriculture, through veterinary, everything, and there the microbes, all kind of microbes are getting exposed to the antibiotics, are also ending up there. Even antibiotic waste by the pharmaceutical industries is also ending up there. So there is a lot of, you can say, exposure of the microbes to these antibiotics, and microbes started becoming resistant to them. They could not be killed by these antibiotics. You can see here residual antibiotics in the hospital effluents are discharged in the waste from pharma industry, manufacturing antibiotics into the into wastewater. Runoff from animal farm agriculture end up in wastewater. So there lot of exposure of these antibiotics to the microbes to the antibiotics take place. It takes place in the human beings also, wherever they're being used. So this is what, as I said, either it is a direct use of the antibiotics, when we are using antibiotics to treat the human subjects or the animals, or the direct use, or it could be the used material residual ending up in polluted water, river environment where microbes are there, that these are getting exposed. Now, this is a very, you can say, summed up kind of a slide from Jim O'Neill, who presented one of the most challenging or most, you can say, informative report on antibiotic resistance in 2014 in a study commissioned by the British government, and highlighted the importance of AMR. Now you can see here on this axis is the penicillin resistance. It's the resistance, and on this is the use of the penicillin, the amount of the antibiotics used. Penicillin, yeah. You can see the more the antibiotic is used, the more, as you keep on increasing the use of the antibiotics, you will see the resistance increases. There is a resistance all over the globe. This kind of a thing has been seen. That means you use the antibiotics, more you are giving rise to the antibiotic resistance. So that means somehow we have to bring down. This is another slide from the Jim O'Neill, who highlights that by this year, by the year 2050, by the year 2050, by the year, here 2050, by the year 2050, see, the number of the deaths by the resistant microbe will outnumber by all other diseases, even disease like cancer. See, this is what has been projected. This is 2050. Remember, we are already into 2024. That means we still have another 26 year or so to address this problem before AMR ends up as a pandemic, before AMR ends up as a problem which will take us to the dark ages. Means we will not be able to perform surgeries, we will not be able to treat any kind of infections. This is what has been projected, you see. This is what is being projected from all the data. Okay, so this is what we have to, that means we have to start addressing this problem right now. In fact, many, you can say, in presentation, you see AMR, the next pandemic. I mean, that kind of thing, being projected. But there is a time, we feel that there is still time to contain that catastrophe. Let's see, no antibiotics today, no cures tomorrow. No antibiotics means antibiotics are there, but they are not able to kill the microbes. So practically it is like having no antibiotics. That's the slogan of the WHO Health Organization. And then how do we address the AM? Develop new antibiotics? Well, we have been doing that for a long time. That means right from 1930s or so, when antibiotics were discovered. We also started seeing the antibiotic resistance, but we never bothered about the resistance because new antibiotics, they were coming. New antibiotics are being synthesized. We were abandoning the use of the old antibiotics and we were using the new antibiotics. As more and more new antibiotics are coming, microbes, they were developing resistance to them. We were discarding those antibiotics, and then we were again adopting new antibiotics. That means slowly and slowly there is a big log of antibiotics to which microbes are already resistant. And the new antibiotics, on the side of the developing new antibiotics, we have reached a plateau where we are not getting new antibiotics. See, the resource, the world from which the antibiotics we were getting, okay, the microbes, the plants, and other sources, that has been exhausted. We have already utilized those things. New sources are to be found out. And developing antibiotics is not a simple job. It takes a long time. The companies, it's a complex problem. Why new antibiotics? They are not. Why we cannot depend upon new antibiotics? Companies, the top pharmaceutical companies, they are shying away from developing antibiotics, but they don't, because antibiotics do not give any return. Okay, in terms of profit. On the other hand, an autoimmune disease will give them a much bigger return, because a person who has an autoimmune disease or has a diabetes mellitus will continue to take their drugs for the whole of their life. So this is again a complex problem, although in the last couple of years, around 25 world-class pharmaceutical companies, they have come and they have joined hands together and given an undertaking to the WHO or to other world bodies to say that they will be developing new antibiotics, keeping in view the AMR problem. Restrict or decrease the excessive use of the antibiotics? Yes, that's another one. But again, here, see, antibiotics have proved to be so effective that people they're not able to pull their hands out from antibiotics, use of antibiotics. See, whenever there is a need to kill microbes, they will use antibiotics. Even doctors, they will continue to use top class antibiotics when they can use a simple antibiotic. Okay, although we now know with the hospital, there is a lot of infection control, and there is a what we call as AMR stewardship. That means that all the hospitals, they have to have a infection control committee where they will monitor what is the AMR status of their institute, and they will try to educate everybody related to use of the antibiotics, that they cannot use excessive antibiotics. They cannot use the most advanced kind of antibiotics for small things or in an unwarranted way. They must use those antibiotics only when those advanced kind of antibiotics are really needed, because otherwise using them for small infections or for even minor you can say incidents, cuts, or other kind of microbial infection would mean that you are putting them at risk of developing resistance. Do not use antibiotics in the animal husbandry. Yes, see, the governments all over the globe, they're putting a lot of you can say incentives to the industry and agriculture, that if you do not use antibiotics, we will give you tax rebates and other things. But then it is not that easy. Only very few companies all over the globe, they have come forward to say that they will not they will not use antibiotics. You will be surprised to know that the use of the animal antibiotics in animal husbandry for weight gain and for especially mainly for weight gain is much more than the amount of the antibiotic which is being used for the human beings. Then, so develop rapid methods for detection of AMR. That means, how do we develop? You know the classical methods, the Kirby-Bor method, that E-test. All these methods, they take a lot of time to develop, a lot of method to know how the microbes are resistant, to which antibiotics they are resistant or sensitive. Now, that means in that intervening period, that 24-hour, 48 hours before you get the antibiotic resistance or antibiotic sensitivity report, the patient must be treated. And always it is an empirical treatment. That means without any kind of an indication. Some kind of indication is there that this could be this kind of a microbes depending upon the site of the infection involved. But it always tends up that there is a use of antibiotics of the more recent origin. Rather, that means again here it's a misuse because of the absence of the rapid method. We are using the more recent and sophisticated and advanced antibiotics more often, and then putting them to the peril of developing resistance of microbes. So if we are able to develop rapid methods to know what is the AMR status of a microbe in a hospital setting, in a veterinary setting, or in a clinic, immediately we will prescribe a medicine which is really not of a higher generation, or which is not of a more advanced, but the one which will be effective against it. In that process, we are saving our more advanced antibiotics, which can be used for our medical purposes, for which can be used for advanced surgeries, heart transplants, etc. If we use those antibiotics, Otherwise, for every small thing, they will be useless, because microbes will develop resistance to those AMR, to those antibiotics. So that means if we are able to develop some rapid methods for developing AMR, okay, for detection of AMR, that can also even ameliorate our problem. It can contribute towards that. Developing new vaccines. Yes, because see, if you have vaccines, the infections will come down, whether it is post-infectious vaccine or it is mostly they are pre-exposure vaccines. So the infections will come down. As infections come down, the use of the antibiotics will come down. The use of the antibiotics will come down, the microbes will not be exposed and they will not develop resistance. Develop rapid diagnostics for infectious disease, just like a rapid AMR detection method, they will also. So it is where these methods will also help in ameliorating the problem of AMR. So AMR is a complex problem, needs one health approach. By one health approach means that it is not to be tackled by the medical people or to the veterinary people or to the environmentalists. Rather, all the three components of this problem, wherever this problem is there, that means the medical, all the fields of medical related to that, and then the environment and the veterinary, all they have to put together and they have to give their inputs together, interact, and finally, that is what is called as one health approach. Okay, this is a recent concept of, you can say, hardly 10 years back or so, where people have realized, scientists, they have realized that it is not possible to solve the problem of the AMR in an isolated approach. But we must, all these three components, where the antibiotics, they are used in large amounts, they should come together and deliberates, and that is how the people or the scientists or organizations, they have or nations have started tackling the problem of the AMR. That means in a one health approach. This is a big, you can say, advancement towards towards a positive step towards our efforts to control the antibiotic resistance or AMR. See, so this is, I have already explained to you, what are we doing? See, what are, as a part of a scientist, what we are doing? See, we are developing a rapid test for detection of AMR. Remember, I told you that this developing this kind of a rapid test will be very helpful in you can say contributing. It's not that something which is only, but you can see it's a huge problem. The whole AMR is a huge problem, and a one health approach has to be followed. And antibiotic development of rapid diagnostic for AMR, detection of AMR is just one aspect, and that is what we are working on that. See, just I think most of the people who are familiar with this kind of a thing, that if there is a patient, see this, his samples, if there's an infection, the samples, the urine, blood, that will go to the lab, and it is only after culture and sensitivity testing that it will come back after minimum of 18 hours, 24 hours, or even 48 hours, depending upon where this test is being done. And during that intervening period of 24 hours or so, the patient must be prescribed some antibiotics, an empirical. And as I said, most of the time it is a broad spectrum antibiotic, more advanced kind of an antibiotic which will be prescribed, although he may need a more older kind of an antibiotic. Okay, so an advanced and that exposure to that advanced or more, you can say, broad spectrum antibiotic means it could be a misuse. It could be a misuse, it may not be required. So this test. Similarly, the same thing will happen in a outpatient. Although we do not have such things most of the time in outpatient departments, a person is prescribed a as soon as he goes into the clinic, the physician will diagnose it and infection, and he will prescribe some kind of an antibiotic because there is no testing normally. This is again will mean that the physician would try to prescribe a more broad spectrum antibiotic. So this is what we want to stop. See, this practice which I'm telling you, this is all over the globe. It's not I'm talking about the India. This is all over the globe. This is how antibiotics are prescribed when the patients are either admitted or they go to the OPD. This is how antibiotics are. And there is a you can say chance of element of use of broad spectrum antibiotics unnecessarily, more recent antibiotics unnecessarily, okay, putting them to the peril of developing antimicrobial resistance or getting into antimicrobial resistance. So this is what we are doing here. See, this can be done all over the globe. Many people are approaching a variety of methods people are using, but they are trying to develop new methods. But one method which takes the you can say highest place, and it's a quite an innovative procedure, is this is what we know. All these kind of 3 tests are there, anti-dilution test, and this is all over the globe. But then this is here. Always the doctor may, under this circumstances, the doctor may prescribe a broad spectrum antibiotic. And that is what we want to prevent. We don't want that it should be prescribed unnecessarily. Okay, and because if we have a rapid method to detect AMR of the microbe or in the medical sample, we can prevent that. Now, as I said, all over the globe, so this is what we call as a point of care test. Such tests are called as a point of care. These tests are they are simple, and they are to be less than 30 minutes. Okay, less than 30 minutes is just for AMR. See, some standards put by the British, you can say certain funding agencies. 1 hour is by the American funding agencies. Okay, so within 1 hour you should be able to. Whereas right now we are taking more than 24 hours. Okay, and the patient cannot be left without antibiotic within that 24 hours, where there is a danger of the prescription of prescribing you can say more broad spectrum antibiotic. So requires minimum manipulations, can be interpreted unequivocally, doesn't require much expertise. It can be done near the bedside. A sample in and results out. That kind of a test, that is called as a point of care test. These are extremely rapid tests. We do not have any point of care test available for the detection of antimicrobial sensitivity or resistance in the microbes, in the resistant microbes. But one method, see, as I said, all over the globe, people are working to develop these kind of methods. But one method which one comes very close, which may be available in the for maybe couple of years down the line, and it will be really a disruptive kind of a method in AMR detection, is the imaging. That means this is a well in which you put the sample of the antibiotic, and then all around the sample there is another jacket kind of a thing where you put the microbe. Okay, they come in contact with each other, and then we see it under the microscope. This contact of the antibiotic and the microbe, we see under a microscope, and just this contact is needed only for few minutes. And then we need to know, we find out or we record the changes in the microbe, morphological changes. And then hundreds and thousands of such microbes that can be quickly monitored and see whether it is there or not, any change is there or not with the particular antibiotic, whether the microbe is resistant or whether the microbe is sensitive to that antibiotic. This kind of a imaging to detect resistance or detect sensitive microbes is a almost, as a microbiologist I would like to tell you, is a breakthrough. It is a breakthrough if we are able to, the scientists have already shown the proof of this principle, but it may take several years before it is developed. And this can be a one method which is the forerunner. And I will just say globally, this is a forerunner. And it is, although there are a variety of other methods which I will not discuss, which people are being, which scientists are developing to rapid tests to develop, which scientists are developing for the detection of the air. Because there is a lot of, you can say, funds or a lot of, you can say, competitive funds are available for this purpose. What we are doing, we are following another approach. We are, see here, we are making a resistance gene array. This is what our technology, resistance gene array based rapid detection of AMR and algorithm-driven therapy. Okay, this is a group. Okay, we have a group at South Campus. These are the people. There are, besides that, there are several other participants to this who are developing this method for rapid detection of antimicrobial resistance. See, this is the, in a nutshell, protocol. That means the sample comes, which could be a urine sample or could be a, of course, blood would be a very difficult thing, but this is how, let's say, the sample, urine sample is maybe a simple sample, and then it contains the material, the infectious material. We take the DNA, we isolate the DNA from this, we melt the DNA, and then the DNA is tagged. Okay, once the DNA is tagged, then this goes to an array. This is an array. And these yellow color things you see are the resistance gene probes. These are probes of the resistance genes which we have taken from literature. Okay, from the literature. And once this DNA, the DNA of the microbe from the sample hybridizes with this label gets lighted, and we know what kind of resistance genes are there. So from here, we are able to know that these are the resistance genes present in this sample or in this microbes of the sample. So exclude these antibiotics. You use the other antibiotics. So there is a lot of further discussion, what kind of antibiotics, all those kind of things as per the new profile of the suggestions of the WHO. All these things, they have to be integrated into this. So, but this is in a nutshell, what is the approach? This is a resistance gene array. This is the resistance gene array, okay, which will detect the AMR profile. This is what the whole, where from we are getting all these gene data. See, this is a variety of, you can say, publications. That this is our publication where we have beta lactamases. We have done the sequencing of the beta lactamases of variety of microbes. And then, of course, this is to be taken from all over the beta lactams, other antimicrolides, other antibiotics. But currently, it is the McMaster's University, which has got a very high database of resistance gene, and we are using that. And this resistance gene data is being at the McMaster University is being it is being you can say updated weekly, more new sequences. So this is what our beta lactamase database. So finally, we end up. This is the design. This is, you can say, periodic or this sequential reiteration of the design what we have. And finally, this is what we end up. You see, for example, it will be something this kind of a equipment or an instrument where you have one module. This one module is mainly for DNA isolation, labeling, and then there are chambers for the lyses and other chemicals, and then the waste material, everything will be done here. This is the sample inlet where the sample will be added up. And then here, this is the array. And you see this is the array, the genes, different resistance gene array. Okay, so the sample will come, it is under this, it will hybridize, and then will get lighted up. The hybridization will get lighted up and will be noted up. And then finally, there is a, you can say, a lot of algorithms behind this, which will give the module 3 will give the treatment options, this and this. And this is has to be done. Everything has to be done within 30 minutes, within the putting of the sample, and before the result is out from here. The result will be out from on this side. Okay, and this is a modular. This is a modular kind of a thing. See, once you have done the sample, you will flush out with the previous sample with this, the remnants of the previous sample. This has to be taken out. This is a modular thing. This, and then the new cassette will be introduced here, and it is ready for the next thing. So we are in the process of developing. You can see this is a very, very complex process, and we are in the process of various. We have groups who are taking care of each and every aspect separately. This is what what is One Health approach, which I was talking about, and you can see here environmental, human health, and this is the One Health approach. And this is our students, my students at the University of Delhi at South Campus. This is we have almost taken every agency for funds to fund our research, and I would finally like to thank the Department of Microbiology at University of Delhi South Campus, where all this work has been done and is further continuing. Thank you very much. If you have any questions, I would be very happy to answer these.
Thank you, sir. Thank you so much for your informative session. If any of the participant is having queries, kindly drop your queries in the chat box. I guess everyone understood your presentation very well, sir. We are not having any questions. Thank you, sir.
All right.
Now I would like to welcome Dr. Simran Patan, Assistant Professor, Parel University, India, to address the gathering. Ma'am, over to you, please.
Thank you so much, ma'am. I would like to welcome all of you.
So sorry, ma'am, you're on mute.
Am I audible?
Yes, ma'am.
Okay. Esteemed speakers, distinguished delegates, and esteemed academic partners, a very good morning. On behalf of Parul University, it is with great honor and privilege that I extend a sincere and warm welcome to each one of you to the AlliedCon International Multidisciplinary Conference on Health Sciences. Today marks a significant milestone as we gather here with a total of 250 participants, including 91 oral and poster presentations, as well as 2 compelling video presentations. We are particularly grateful for the presence of our academic partners whose unwavering support and collaboration in shaping this conference, Benevolencia Foundation, Chettinad Academy of Research and Education, and Parul University. Your commitment to academic excellence and advancement is truly commendable, and we are honored to have you as integral parts of this event. This conference serves as a platform for the exchange of groundbreaking ideas, cutting-edge research, and transformative insights across various multidisciplinary approaches. It is a testament to the collective dedication and passion of each participant gathered here today. Over the course of the conference, we have the opportunity to delve into a diverse array of topics, engage in thought-provoking discussions, and foster meaningful connections that transcend geographical boundaries. Together, we can explore new frontiers, addressing pressing challenges, and pave the way for innovative solutions that have the power to shape the future. Once again, I present my Thank you.
Thank you, ma'am. Now I would like to welcome Professor T.S. Dr. Mohammad Nasir Mohammad Desha, Head of Department of Biomedical Sciences, Faculty of Medicine and Health Sciences, University Putra, Malaysia. Sir, over to you, please.
Thank you. Thank you, the organizer. Let me share the slides. Okay, so this is my first slides. Hope you can hear me. Okay, once again, thank you, organizer, for giving me the opportunity to talk on this platform, to share my work for this particular session. I'm going to talk on the Group B Streptococcus. Okay, the zoonotic potential and prevention. My area is actually molecular microbiology, and I work on some of the medically important bacteria, mainly Staphylococcus and Streptococcus species. Previously, I work on, I'm also actively working on Streptococcus pneumonia, Klebsiella, also gram-negative Klebsiella pneumonia, Staphylococcus aureus on MRSA, and Coagulase negative Staphylococcus, talking about antimicrobial resistance. So I'm inspired by Professor Duke Sharon talking about these problem associated with the AMR.
That's not the right place for you.
Okay, so I'm sure we have been talking about the zoonotic threats. I think we know about zoonotic disease, zoonotic threats. In Malaysia, normally, if we're talking about zoonotic, we will be talking about leptospirosis, food poisoning, salmonella infection. Those are quite common in Malaysia. But probably GBS, Group B Streptococcus, or GBS, probably not much in the attention until the last few years. As one of the foodborne pathogens, Group B Streptococcus or Streptococcus agalactiae is a gram-positive, catalase negative, and a member of the human commensal gastrointestinal and genitourinary flora. It infects a range of hosts. Of course, we are one of the hosts, but it also infects cattle and fish. So in humans, GBS has been a significant pathogen that has been causing meningitis and septicemia in neonates or young babies. Also, it caused infections in pregnant women as well, which is in fact is quite common among the pregnant women. And we have seen in the last few years the GBS infection in the incidence of invasive GBS infections such as the soft skin tissue infection has been among non-pregnant adults has been increasing. Okay, so if it happened among the pregnant, that was common because that has been the patterns all this while, but now we have been seeing the patterns where it involved non-pregnant adults. And at the same time, this is an interesting, this is the interesting part of this study because at the same time, GBS is also an important fish pathogen, especially in Malaysia. We have a big aqua industry involving the tilapia, fish tilapia. That's what we call it here in tilapia. I think in English, we call it Peter's fish. So it's for human consumptions. And one of the threats for the fish health is actually infection by group B streptococcus. And it has been affecting our aquaculture industry quite a great deal, quite economic loss and so on. So this is the basic characteristics of the properties of the GBS. It has virulent factors, a range of virulent factors that facilitate the disease development, such as a capsula, polysaccharides, surface protein, toxins, pili. So those are common properties, not only GBS, but in other streptococcus species as well, that play roles in adhering, invading, and causing pathological disorder of the host cells and leading to disease. And it also evades the immune system as well. So the molecular serotypes, the way to study the epidemiology is actually to look at in terms of the serotype distributions, because some serotypes are associated with with certain level of invasiveness. So for serotypes of GBS, we determine by capsular polysaccharide gene typing. Okay. As of now, GBS can be subtyped into 10 serotypes. And serotype 1A, 3, and 5 are the most common associated with invasive infection in humans, while serotypes 1A and 3 are associated with fish diseases. So we can see that 1A and 3, they affect both human and fish. But the question is, are they, although they belong to the same serotypes, 1A and 3, where they infect both hosts, human and fish, but are they really belonging to the same strains or the same subpopulation? That is the question mark. Because serotyping, as we know, is not discriminatory enough to discriminate at strain levels. So we need to do a genotyping in order to discriminate at this level. So what are the techniques that we use? We frequently use MLST, multi-local sequence typing, which is a genotypic method that will provide the genetic lineage or genetic backgrounds of the isolate by giving the sequence types, the ID nomenclature of that particular lineage. We call it the sequence type or ST. And in fact, actually, MLST is quite an established where there has been a database where if we look at the variety of ST among GBS, there has been more than 2,000 sequence types all around the world. So huge, so diverse. So in Malaysia, strains belonging to ST1, ST17, ST19 are the most common in human isolates, while ST7 and ST283 are the most common in fish isolates. Okay, so here come the interesting part of the study. Okay, so the interesting part that triggered the study is actually the occurrence of an outbreak that was in 2015 in Singapore. So in 2015, GBS ST283, okay, was identified as the agents of an outbreak that involved humans. Okay, as we know, ST283 has been causing disease in fish, but this is something like the first time where we hear that ST283 has been causing an outbreak. Although there has been outbreak among fish, among the aqua industry, there's a lot of outbreak involving GBS infection, but now we're talking about human infections. So this outbreak was associated with the contaminated raw fish salad consumption. So they eat raw fish meats. That's something like here we do eat raw uh, as part of the menu. And it was unique, because it affected non-pregnant and younger adults without having any significant comorbidities. So they end up getting a severe infection. And this is something that has sparked interest to further illustrate the zoonotic potentials. And that was one of the reasons why we are starting up this study in Malaysia as well, to look through to do the survey on the GPS. Because we have been working on GPS for quite some time, but we are focusing on humans without realizing that actually is a big issue in the aquaculture industry involving fish. And now suddenly with the outbreak, we can see that particular ST283 jump from fish to humans. So we have to, answer a lot of questions from here before it getting worse. So this is our previous study. I'm sharing our previous study. This was in 2017. This is our previous study on human isolate from based on samples isolated from a number of tertiary hospitals, major hospitals in Malaysia, collected within 2012, 2014, prior to 2015, in the Klang Valley. Klang Valley is the most densely populated area where you can go to Kuala Lumpur, or all the nearby cities are within the Klang Valley. So it's highly populated area. And you can see the serotype 1A was the most frequent, followed by serotype 3 and 5. And this is another interesting observation. Largely, they were susceptible to penicillins. So, well, this is consistent in many studies where GBS, they keep susceptible. They are quite susceptible to penicillin and also to a number of other antibiotics. So that's why penicillin remain one of the antibiotic of choice for GBS infections. So this is the extension from our earlier study where we did MLST. We can see the prominence ST, ST1 is the prominent ST from both invasive and non-invasive sites. And this is also consistent with many other studies in relation to serotypes and sequence type as far as the humans actually are concerned. Okay, so all this while, we have been focusing on humans, our group in the medical faculty, we have been focusing on humans, while actually there is another group from our university who has been extensively working on GPS as well, but in the fish settings, in the aquaculture industry, working on studying the fish pathogens until the Singaporean outbreak took place that brought us together. So this is our study. Our earlier study, this study involved about 250 fish GBS from outbreak cases of streptococcosis among the fish, the red hybrid tilapia in Peninsular Malaysia. So we can see a certain pattern here where the same serotype in human found in fish, which are serotype 1A and 3, are found. We're talking about serotype, but when we're talking about the sequence types, so we found ST7 and ST2A3, which are more common, which is consistent. So fish, normally we're talking about ST7 to A3, while in humans, there are different STs, and in fact, they are a lot more diverse. And in these fish settings, it was observed that ST2A3 appeared to be more virulent in red, but they cause a more virulent disease among the fish. Okay, so whether they can cause some level of virulency in human, that is question mark. But we have seen the outbreak where people end up getting a severe infection due to the infection by the GBS ST2A3. So this is the outbreak. This is the outbreak that involved humans. Okay, to be more specific, it involved individuals who consume. This is the outbreak. It involved individuals who consume raw fish meat and linked to the contamination with GBS ST2A3. Somehow the route of transmission is through the raw fish meats and then end up in the humans. So it was unique because it affected non-pregnant and younger adults. And now we should start questioning whether GBS now is really a human threat, because now GBS is not just between human and human transmission. Now we are dealing with the fish to human transmission. So that will just add on to the risk factors will just simply add up. Okay, this is the press statement from the Singapore Minister of Health, where they reported that between January and June 2015, they received a report on more than 200 cases from public hospitals. There has yet to be a proven link between eating raw fish meat and serious GBS disease in human, but investigation involving sampling of raw, they started investigation by doing samplings. They do found some preliminary association between the GBS from fish and end up in humans. So at the meantime, so the incident has got attention in the social media. So this some of the news came up, okay, related to these incidents. So as it is, the question is it a newly case or maybe something that has been there all this while and we have yet to realize? So this is still a question marks, because previously we underestimate, we underreported because we just concerned, we never thought that it could cross, from fish to humans. Although GBS is well known as fish pathogen, but those that are causing human are of different lineage. Okay, so it's a different STs, while we know in fish is of different fish, but now the lineage that are actually having problem that are causing problem in the fish now end up in humans. So there is the question mark that we have to answer. So, and then this is the detailed study on the outbreak published by the Singaporean groups in 2017 that managed to associate the improper sourcing and handling of fish for raw consumption with the incidents. So in the study, they conducted a SNPs, single nucleotide polymorphism, SNPs based analysis, phylogenetic analysis, involving GBS strain from various sources. So they detected GBS ST283, which the one that caused a severe foodborne outbreak in Singapore in 2015, with other freshwater fish origins, not only in food store and fish market, but also in ports from which fish were imported. So they do comparison. So comparison of human and fish isolate showed a very little differences in terms of the SNPs. So meaning that imply that they are actually they're showing that the GBS ST283 are highly related. So meaning that somehow the origin is coming from the fish. So this is another study related to the outbreak with a wider scope of analysis involving GBS genomes from other countries as well, published in 2019. So it involved a whole genome of more than 100 GBS isolates collected from 6 Southeast Asian countries and the phylogenetic analysis of more than 7,000 GBS sequences, including more than 200 variants of ST283 from human and animals. So they found GBS ST283, which actually has been widespread in Southeast Asia. Okay, so meaning that actually this issue has been there, it has been widespread, but nobody is bothering about it. So now we should be bothered about it. So this particular GBS ST283 has been, there has been accounting for a large proportion of bacterial GBS and causing disease and economic loss in the aquaculture. Okay, so but still, whether the transmission is from aquaculture to humans or the other way around, or probably it involves some of unidentified reservoir still remains unknown, that we have to, carry on more studies to establish that transmission. But as it is now regional issues, Malaysia, as we know, Malaysia is the closest neighboring country with Singapore. So it's quite concerned due to the rapid traveling activities among the Southeast Asian countries. Because Malaysians, they like to travel, they go to Thailand, Vietnam, Singapore, Indonesia quite frequently. Particularly between Malaysia and Singapore, where Malaysians, they travel every day, they commute every day to work over there, and then they come back and reside in Malaysia, and then that's when they go back to Singapore to work. So it's very rapid humans movements. So so that just simply put on the risk of, if there are GBS, somebody is carrying GBS ST283, that will just actually enhance the transmission. So we have to we took the efforts, to do survey, because nobody's doing it before in Malaysia. So now we took the survey, the effort to do the survey to screen for the potential presence of ST283 among hospital settings. Okay, this is our study that was recently published in 2000, very recently published, where we compare human and fish isolates. So we found, this is very interesting, we found two ST283 strains at the hospital settings, two. It doesn't look too much, but still it is a concern, because two, it may multiply and disseminate if we don't take precautions after this. So I'm going to share the rest of the talk. I'm going to share a bit some of the findings. So this actually are the earlier objective of studies. Eventually, we are going to study the proteomics as well to see what the protein being expressed between the two group, fish and human isolate. But this is still ongoing. So what we have completed is actually just a very basics characterization comparing between the antimicrobial susceptibility patterns between the two groups, and then determine the serotype distribution as well as the virulent genes, some of the important virulent genes, and also the MLST, the sequence type, and the clonal complex. So these are the methods. So for human GBS, we collected from sterile site of patients, so meaning that come from blood, CSF, the invasive sites, from 6 major hospitals in Malaysia. While for the fish isolates, it's already there because somebody actually had been working for quite many years. So there has been huge collection of GBS, fish GBS. So we just simply randomly pick to compare with the human isolates. And these fish isolates are actually getting, they were previously isolated from various locations throughout Malaysia. So, and then we just simply doing a very basic phenotypic method to re-identify the species for viability, as well as determining the antimicrobial susceptibility by distribution and e-test. And then for molecular work, we utilize PCR to detect the virulent genes, and multiplex PCR for the MLST. Multiplex PCR for the serotypes and also for the MLST. Okay, but MLST we just did on the selected isolates only. So, and of course with the some statistical analysis whenever appropriate. So this is the result for human isolates. So we managed to get 118 invasive. It's not just a case. This is we are targeting the invasive case, 118. But unfortunately, 5 did not grow. So we just deal with the 113, 113. And we can see, although the sampling, there are some elements of convenience sampling because of the logistic problem, and as well as cooperation from the hospitals to surrender the isolates to us. So of course we got a proper ethical clearance and so on, because it involves the Ministry of Health and the government hospitals. But we can see a majority of the samples collected from the hospital near Singapore, hospital in the state of Johor, which is very near to Singapore. And they are frequently coming from females and Malay, because Malay are the majorities ethnics in Malaysia. That would probably explain why we got so many samples from Malay as compared to the other races like Chinese or India. And interestingly, they are frequently coming from females. And then most of the select were from blood samples. We do have a mortality rate, but quite low, 8%, which is good, meaning that majority survive. But still, the 8% are very unlucky. No, we don't want anybody to die. We want to save everyone. So interestingly, this is also interesting. As I said earlier, we have been seeing the increasing patterns among infection among non-pregnant. Although GBS is very common among non-pregnant women now we have seen the pattern of infection among non-pregnant. So in this collection also, majority are actually coming from non-pregnant adults, but majority are humans. Okay, this is from fish. This is for fish. Fish, actually, we already got the collections. They are still viable. So they are collected from different states, mostly from the north part in the state of Kedah, in the east in the state of Terengganu. Okay, this is just a basics identification process where they do match the characteristic of GBS. So both human and fish, phenotypically they look the same. And then this is the result for the antimicrobial susceptibility patterns. We can see, this is quite interesting to see a very obvious patterns among fish and humans. So you can see none of the fish isolate are resistant. Okay, we use 13 antibiotics, those that are commonly used in hospitals. So none of the fish isolate were resistant except for tetracyclines. While you can see for human isolate, they are also quite high actually resistant to tetracycline, but there are also a low incidence of resistance to the other antibiotics as well, erythromycin, for example, the erythromycin, azithromycin, clindamycin, chloramphenicol, but the percentage is quite low. And none are resistant to penicillin, which is quite good, meaning that antibiotics they're still workable, they're still effective, but not for the case of tetracycline. This is quite interesting, probably because as far as fish isolate are concerned, actually tetracycline is quite extensively used in the agriculture, including aquaculture. So that will explain, as Prof. Yuksaran talked earlier, when we use antibiotic, that would just trigger the development of resistance. So we have seen a high number of resistance when in the response to the extensive use of the particular antibiotics. But it also tetracycline is so high among humans, which is quite interesting. Probably I'm not the clinician, so I don't know whether tetracycline is used by the doctors in the hospital. Okay, so this is the serotype distribution. So among the human isolate, there are 8 serotypes were determined, and the most common were serotype 5, followed by 1A and 2, while all the fish isolate only have a single serotype 3. This is a single serotype. And this pattern is actually consistent with many other studies, not only in Malaysia, but in the other countries as well, where serotype 1A and 5 were very common among human isolate, while serotype 1A and 3 are very common in fish isolates. But here we only have serotype 3 for our fish isolates. Okay, so this is the difference between distribution of virulent gene, which is quite interesting to see. There are some differences. In fact, they reach the level of significance. You can see a number of genes such as the LAM, SETB genes were positive in all humans, while a low percentage were positive among the fish isolates. For the RIP genes, most human isolate were positive, while most fish isolate were negative. While for the BAC gene, most of the fish isolate were positive, and a low percentage of human isolate were positive. So for pili genes, pili 1 were found in around 87% of human and all fish isolate, but pili 2A is absent in fish and found in 70% of human isolates, while pili 2B is found in 30% of human isolate and 9% of fish isolate. So for a number of genes, there are big differences. And the absence of the SETB and RIP genes in 95% of the fish isolate in the study is consistent. Some of the patterns are consistent as reported by the others as well. Okay. And this is the sequence types. MLST was performed, of course, because it's the costs and because of cost, we couldn't afford to do MLST on all the isolates. So we just do some representative. So MLST was performed on 46 human isolates, while for fish only 15. Why only 15? Because it all turned out a single sequence types, ST283. So we just stopped. Because from the literature we know it's all ST283 or the other one. So we did up to the 15, then we just stopped for the fish isolate because they all turned up giving one single ST. So, but we do found here GBS serotype 3, ST283 in 2 cases among humans. Okay. And these two patients are actually Chinese, and they are not pregnant ladies. They are not pregnant patients. But of course, the problem is we don't know their traveling history, so we couldn't rule out whether that particular ST283 from this patient are coming from fish or not. Okay, and we conduct a phylogenetic analysis, and the isolate were grouped into 4 major clades with 2 major groups, clade 1 and 2. So, okay. So this is the phylogenetic analysis. So there are 2 major clades, 1 and 2, and all the clone complex for 283, ST283, are clustered in clade 1. So somehow implying that these isolates are genetically closely related. Okay, as moving toward the conclusion. So In Malaysia, invasive GBS among non-pregnant adults indicates a growing burdens, especially in the elderly and those with chronic diseases. This study shows similar phenotypic characteristics between human and GBS. They look similar phenotypically because they are GBS, but molecularly, at the molecular level, genetic level, there are some differences. So, especially different features in the virulent gene profiles, antimicrobial resistance, serotype distribution, sequence types, okay, between the two groups of origins, where we can see fish GBS isolates have less genetic diversity. Somehow they are more similar. Just normally they are all a single ST283, while humans isolate, the ST for human isolate are more diverse. But the important observation, the important finding from our study is that we found two strains of ST283 in humans for the first times. Okay, that was the first time where definitely we publish it. So this was very recent publication, Fish Associated Streptococcus agalactiae ST283, first human case reported in Malaysia. But there's still limitation because we couldn't trace the traveling history of the patient to rule out potential fish origins. But tentatively, preliminary, there are we can do some inference. But of course, we need to do a more analysis. Probably we need to do. We are going towards a WGS to really establish the correlation of to establish the origins. And of course, we have to conduct a more proper studies in futures, more systematic with the proper questionnaire and so on. And of course, we want to see the solutions, how to solve this problem. But unfortunately, in humans, there has yet to be a vaccine. So there's no vaccine yet for humans. So, but luckily, okay, we can see largely GBS are still susceptible to antibiotics, except tetracycline. But there are so many other alternatives other than tetracyclines. So that's good news. But that will not make us safer because we don't know, they may emerge and become resistant in the future if you don't put a proper management on the antibiotic use. But of course, what can we do? What else can we do? We can do some prevention measures at the aquaculture setting because these fish, tilapia, are actually the major source for food source for human consumption. So they are being farmed, okay, for economic purposes, business purposes. So at least if we can control, do some measures here, perhaps the risk of zoonotic transmission can be reduced. Okay, so I'm not a fish expert. I'm not a fish expert, but there has been some recommendations to tackle the issue by the other groups. So such as the use of probiotics instead of using antibiotics, to ensure the health of the fish. Why not we just remove the antibiotic, as Professor Juksaran already talked earlier. You use antibiotics, that will simply lead to emergent resistance. Why not we just stop it and replace with the probiotics? That would be an alternative. But of course, we need to do a more long-term study to see the efficacy, effectiveness, and so on. Plus, of course, we can always apply the good practice. Malaysia, we have a good agricultural practice, GAP, to follow. And specifically for GBS, there has been some recommendation for good husbandry for GBS. And in fact, there has been vaccines. The other group has been working on the vaccines against GBS. Okay, so still under development. So, of course, this area of research, the vaccine development, the genotyping to understand the transmission, as well as the matters related to human treatments. So will have to be attended by different expertise, different experts, different researchers. So I'm a molecular biologist. So I'm more into characterization of isolate, particularly at the genetic level, to provide genetics background to understand the epidemiology and transmission pattern, while Professor Shafinas, she is a medical doctor. Okay, so human medical doctor, and Dr. Amal, he is a scientist in fish health. So we are working together towards single outputs, which is for the sake for preventing, for minimizing the disease burdens of the GBS, especially with the threats, with the potential threat, the potential zoonotic threats, where if you don't do anything from now, perhaps that particular matters may get worse in the future. So we have to act from now. And I think the Minister of Health also has, listened to us. So there will be something going on to see how to tackle this issue. But we have to keep doing the surveillance to see how serious is the potentials of the disease burdens. Okay, so that's all for me. Thank you very much.
Thank you, sir. If any of the participants is having any queries, kindly drop your queries in the chat box.
May I ask one question to Professor Nasir?
Yeah.
First of all, I must congratulate you for a very meticulous study of the GBS.
Mm.
Now, my query is that the two patients in which the sequence type 283 was detected, one was arthritis and other was probably meningoencephalitis. Were there any comorbidities in them?
Actually, we don't know. We don't have the information because this is a retrospective. And then the patients actually from government hospitals. So we cannot just simply go and trace the medical record of the patient without the permissions. So as we are also, at the time, we are also in hurry. We are trying to catch up so many things. So we couldn't manage to get the sufficient information to establish. That's why I said just now, it would be interesting if we can know where did they go, where they came from, what will they eat. There has been some. So of course that will be the limitations. So that's why for future we are trying to come up with a better plan with a better systematic study, where to come up with the proper questionnaires, where make sure we got all information needed, plus with all the laboratory work and the genotyping approach, and at the end when we put all together, we can see a clear pictures. But we don't know whether we're going to get more ST283 in humans in the future. We don't know. We haven't started. This is the first surveillance study, so we're going to start more surveillance with the collaboration with the Ministry of Health as well. So hopefully, of course, funding is very important. So hopefully, we can get more fundings to carry on with our surveillance. Hopefully, we will get a better result in the future. Yeah.
Good. See, another query. Among the GBS, I think serotyping is the most commonly used method to identify them, isn't it? Or in microbiologist day-to-day work, it is probably the serotypes which are being used.
Yeah.
So it is only the use of the MLST to identify the sequence type 283 has led to the identification of an organism causing an invasive disease. Am I right?
Yeah, but we MLST will give the genetic lineage, so we know the transmission, the lineage of the particular strains. But if we just add on the serotype as well, that will just make it more informative, because
no. But supposing had you not done the sequence typing, MLST,
we did.
So supposing it was not done, would it have been possible to identify this particular invasive organism only based on serotyping?
Well, we can identify this invasive level by simply looking at the severity of the case. We can see is the potential, the severity, the potential, the virulent level of the particular strains, and the serotypes that are associated with that particular virulent, with that particular severity. But if we just add on the MLST, that will just give more information on the origins, and then taking an example with another organism that I'm also working, Streptococcus pneumonia, the same lineage, the same ST, but there are different serotypes. And the hypothesis is that there is actually the same clonal groups, the same lineage, where in responding to these environmental pressures or vaccination pressures, they switch the serotypes. So originally, the same group or lineage, they are carrying a serotype, But along the way, they switched to different serotype because to evade the effect of vaccination. Because for pneumococci, there has been vaccines available.
Yes.
So in GBS, we don't have vaccine yet, but of course, we always do serotyping. That's a very basic information. And then we, with the complement, with the sequence types, that will just give more information, to do the traceability study. But of course,
thank you very much.
But now we are doing conventional MLST by PCR. But now with the technology, with the NGS and so on, so if you can sequence the whole genome, you can go all the genes. So we just need to create a pipeline for you to mine the housekeeping genes for the MLST. Now they are talking about core genes MLST, core genomes, meaning that for conventional MLST, we just work within 7 housekeeping genes, which believed to be discriminatory enough. But now they are talking about the whole genomes, which will increase the resolutions. But of course, it's just put you more costs. But I think we can see the trend, the cost is getting down, but not up to the level where we can afford yet.
Yes, true,
it's still
very
expensive.
true. Let's hope for microbiologists to see the whole genome sequencing comes to a very cheap level, so that everything we can get sequenced.
Yes. And I am interested in the machines, your devices, which can be taken over by WGS, whole genomes. You don't need a device, because if you can sequence the whole genome, you just need a software to find the genes. So your device I think will have problem if you want to put in the market. But it's good work. Yeah, not all can afford to WGS. We still need some alternative especially for the low from our country. We are not actually a rich country. So although not a poor country, but not very rich. So we're still struggling for fundings. So we should have alternative. Okay.
Okay. Thank you very much. Wish you all the best.
All the best. Thank you, Prof. Thank you very much.
Thank you, sir. Now I would like to invite Dr. Jitin C. Saji, Senior Specialist, ENT and Head and Neck Surgery, Aster DM Healthcare, India. Sir, over to you, please.
Am I audible?
Yes, sir.
Yeah, thank you. 1 second, let me just share my screen. Is the PPT audible now?
Yes,
I
sir,
mean,
it's
visible, it's visible, right?
Yes, sir, it's visible.
Okay. So, good morning, everyone. Myself, Dr. Jitin, consultant ENT and head and neck surgeon. I'm working with Aster, India. So, my topic for today is a little bit specific. It's a radioactive iodine refractory thyroid cancer and how to work it up and how to manage those cancers. So radioactive iodine has been the cornerstone in routine adjuvant management of differentiated thyroid cancer. But around 5 to 15% of differentiated thyroid cancers and around 50% of metastatic DTCs are refractory to radioactive iodine. So how to go about these type of cancers? That is the behind this presentation. So these cancers which are refractory to radioactive iodine, those are poor outcomes with a 5-year disease-specific survival rate of 60 to 70%. And when there is metastasis, the 10-year survival rate is in the range of 10%. So these are the different treatment options we have for thyroid cancer. And first and foremost, surgery is the mainstay for any differentiated thyroid cancer. And when there is gross, like when it is an advanced stage like T4, or when there is a distant metastasis, or histological type of follicular thyroid cancer with extensive vascular invasion, when we know the tumor resection was not complete, or the lymph node, the metastatic lymph node was more than 3 centimeter in size, or post-operative there is inappropriately very high level of thyroglobulin. These are the situations in which we would use radioactive iodine as an adjuvant treatment. Post-operatively, we keep the TSH in the suppressive range, which is less than 0.01 by giving thyroxine. And radiotherapy is considered only in select patients in which we know there is aerodigestive tract infection, or we know the resection was R2, like gross residual disease was left. In such cases, we consider external beam radiotherapy. and how to manage recurrent loco-regional or distant metastatic disease. So if these recurrent disease or metastatic disease are amenable to locally ablative interventions like cryotherapy or surgery, that we go ahead with that. And if the symptoms are not progressing very rapidly, patient is not very symptomatic, we can keep them under observation. This systemic treatment is somewhat a newer development in the field of thyroid cancer treatment, and patients who have rare refractory DTC with a significant clinical progression, or those who have symptomatic disease that is not amenable to local treatment, or those who have disease in such an area where surgery is not possible because it's near to some very critical structure, operating there would be difficult. So these are the patients for whom we consider systemic treatment. So these are the different type of radioactive iodine treatment, namely remnant ablation, adjuvant treatment, and treatment of metastatic disease. So remnant ablation is used when we know there is post-surgery residual disease, and this is also increase the specificity of using thyroglobulin in the post-op period as a marker for recurrent or metastatic disease. Then there is adjuvant treatment, and this is used for residual and microscopic disease. And when there is distant metastasis which is not amenable for surgery, we can use radioactive iodine as a modality. So coming to the topic per se, radioactive iodine refractive phenomenon. This was detected as early as 1952, and in 2014 in the 14th International Thyroid Congress, they defined it as combination of at least one lesion which does not show RAI uptake on RAI whole body scan, or clinical evidence that RAI no longer provides a benefit to the patient. In 2012, in PISA, they defined it as one or more metastatic lesions failing to take up RAI and continuing to grow. The latest definition is given by Tuttle et al., and he defined it in 2014 as negative post-treatment WBS after a properly administered RAI therapy, or structural disease progression or rising serum thyroglobulin 6 to 12 months after the previous RAI therapy. So to summarize, RAI refractiveness is basically when there is absence of radioactive uptake at initial diagnosis of locoregional recurrence, or when there is absence of progressive loss of radioactive iodine uptake in the post-therapy scan, the presence of more than one metastatic lesion with at least one lesion with radioactive iodine uptake in the post-therapy scan, or structural progression of tumors post 1 year after RAI therapy despite the presence of radio uptake in post-therapy scan. Patients who have already received 600 millicurie of radioactive iodine without any signs of remission. So all these, and also when there is a significant uptake on FDG PET, usually thyroid cancers do not show uptake on FDG PET. So these are the characteristics of a RAI refractory thyroid cancer. So American Thyroid Association has defined it in 2015 as when there is malignant or metastatic tissue which does not concentrate radioactive iodine, or the tumor tissue loses its ability to concentrate RAI after previous evidence of RAI. Like the tumor was uptaking radioactive iodine in our first postoperative scan, but it loses its ability to uptake it in the consecutive scans. Or when there is radioactive iodine is concentrated in some lesions, but there are few lesions which are not taking up radioactive iodine, or a metastatic disease which is progressing despite a significant concentration of radioactive iodine. So when we give radioactive iodine, these are the different types of response we see in the tumor, and these are classified as excellent response, biochemically incomplete response, structurally incomplete, and intermediate. Excellent is when there is no clinical, biochemical, or structural evidence of any disease. That is the best we can achieve after a thyroid cancer surgery. And then there is biochemical incomplete response, in which there is an abnormal thyroglobulin or a rising, or serially when we check anti-thyroglobulin antibody, it is showing a serial rise in the levels of anti-TG antibodies. These are the all these comes under biochemical incomplete response. In this, there is no localizable disease. Only biochemical parameters are deranged. Then there is a structural incomplete response, persistent or newly identified locoregional distant metastasis comes under this category. Apart from these three categories, there can be a fourth one, namely indeterminate response, when there is non-specific biochemical, or when When the characteristics not fitting into any of these three, that's when we call it indeterminate response. So this structurally incomplete response, usually we need to see whether these are actually responding to radioactive agent or not, because these are the ones which are usually not known for taking up radioactive agent. So can we predict whether one patient is going to be having this rare refractory cancer while seeing them in OPD? So these are few characteristics which can give us some idea about his rare refractory nature. So one is patient who are older than 40 years of age, and then there are some tumor characteristics like in pathology the histology is very aggressive, or there is some local invasion of the thyroid cancer, usually to the trachea or aerodigestive tract. Then the presence of metastasis. These things should make you suspect that this patient can be refractory to radioactive agent therapy. And in imaging, if the patient is taking up FDG PET and there is no iodine uptake, that is almost always sure that the patient can be radioactive iodine refractory cancer. And the thyroglobulin doubling time, which is less than 1 year, also points in that direction. So I told about worse histological variants. Tall cell variant of papillary thyroid cancer. This is the most common aggressive type of thyroid cancer, and in this variant there is more incidence of BRAF and TERT mutations, and this correlate with the rare refractory, and it has a very bad prognosis. Other variants which are more correlated with the rare refractory are columnar cell diffuse sclerosing, solid hobnail variant, and widely invasive follicular variant. So about thyroglobulin doubling time, this is useful in identifying those who are more likely to develop new metastasis. Like we can follow up. Thyroglobulin is something with which we follow up the patient after the treatment is completed. So we look for thyroglobulin doubling time, and if it is less than 1 year, those patients we can have an idea that this patient can go into developing a local recurrence or new metastatic lesions. So about FDG PET, usually FDG PET is of no use in thyroid cancers, but in patients with high serum thyroglobulin and an increasing trend of thyroglobulin, this FDG PET can be useful, and lesions which are positive on FDG PET are less likely to respond to even higher doses of radioactive iodine. So a thyroid cancer which is getting lightened up on FDG PET is not very good for the patient. So how to identify? These are the three ways with which we can identify. One is iodine 131 whole body scan, FDG PET, and genetic characteristics. So iodine 131 whole body scan, it is useful in detecting remnant and rye with residual or recurrent lesions, and then we can plan on giving definitive iodine 131 treatment for the patient if the lesion is habit. And it is a very good investigation modality we have. It has very high specificity, but sensitivity is low, but its accuracy is in the range of 90%. The negative imaging on iodine 131 scan with an abnormally elevated thyroglobulin level should make you suspect that the patient is having a rare refractory disease. So coming to FDG PET, as I told, there is an inverse relationship between RAI uptake and FDG PET. So this phenomenon is known as flip-flop phenomena, and this loss of ability to concentrate RAI is because of the dedifferentiation in the tumor cells and the higher demand of glucose by tumor cells. So they tend to take up more FDG PET rather than taking up radioactive iodine. And this is recommended for patients with high risk thyroid cancer with an elevated thyroglobulin level. Like stimulated thyroglobulin means we are keeping the patient. We have withdrawn the thyroxine and we stimulate the thyroid to create thyroglobulin. Even after that, we look for the thyroglobulin level, and if it is more than 10 nanogram per ml, It is that is known as stimulator thyroglobulin, and in patients with a very high thyroglobulin level, we can use this FDG PET. So coming to genetics, BRAF 600E mutation is the most common mutation. This correlates very well with the recurrence and poor outcome. And this BRAF 600E mutation negatively regulates iodine metabolism and through abnormal activation of MAPK pathway. And this is why such patients are refractory to RI, because their sodium metabolism, sodium iodine importer is negatively regulated. So these are few newer imaging modalities. These are only upcoming. Among these three, only prostate-specific membrane antigen has shown some kind of promise. PSMA is used mainly in prostatic cancer for its treatment and also in diagnosis. There was a paper published by Verma et al. Who showed that PSMA could detect 30 out of 32 lesions when FDG PET could only detect 23 out of 32. So this might be suitable for diagnosing and treating RI negative differentiated thyroid cancer, but this is still under trial. So thyroid cancer cells express the somatostatin receptors, mainly type 2, 3, and 5. Diagnostic value and therapeutic value of these receptors are still under debate. This is just for the completion sake. I have included this choline imaging. There is no convincing evidence, but papers have shown incidental thyroid uptake in FDG PET and choline. When a patient had undergone choline PET CT for prostate cancer, it had shown incidental uptake in the thyroid also. So that is how they started thinking about whether this can be used in the diagnosis and management of RI refractory thyroid cancer. This is still under trial. So how to manage these patients? There are 4 options basically. One is we can just observe the patient. So which all patients are suitable for observation? If the patient is having an asymptomatic disease with a very low tumor burden, and the tumor size is less than 1 centimeter, and the tumor is progressing very slowly, these are the patients whom we can keep under observation. And wherever surgery is possible, we should go ahead with surgery. That is the treatment of choice for primary thyroid cancer, recurrent thyroid cancer. For every type of thyroid cancer, the treatment of choice is that, provided surgery is possible. And in case of bone and CNS metastasis, along with surgery or as a single modality treatment, you can use radiotherapy. So coming to some newer development, some specific tyrosine kinase inhibitors have been studied, and among these two, mainly lenvatinib and sorafenib are approved also. So these are used in this RI refractory thyroid cancer as a systemic therapy. Vantinib and cabozantinib are still in phase 3 trials. Its results are not yet out. So this is select trial which had assessed the ability of lenvatinib in treating RI refractory thyroid cancer, and it showed that the progression-free survival benefit was very good with lenvatinib. So these lenvatinib and sorafenib are basically the only 2 approved TK for the use in RI refractory thyroid cancer. This slide is just about basic differences between lenvatinib and sorafenib. Basically, the target is different, dose is different. The problem with all these tyrosine kinase inhibitors are its side effects. So, like we already talked, BRAF 600E is the most common mutation, and this is the reason behind RI refractoriness, because it inversely correlate with sodium iodine import, and this is mainly done through 2 pathways. It causes activation of TGF beta pathway and epigenetic histone deacetylation of H3 and H4 lysine residues of sodium iodine import. And this in turn prevents this transcription, and there comes a downregulation of sodium iodine. When this symbiote is not working properly, cells obviously cannot take up iodine. So this BRAF positivity correlates with the high level of PD-L1 and PD-1 receptors, and these tumors show high level of suppressive T cells and macrophage components in them. Vemurafenib is a newer drug which is specifically against mutant BRAF 600E, and this can be used in patients with papillary or refractory to radioactive iodine who have never been treated with tyrosine kinase inhibitor. So now comes the question: who can be taken up for tyrosine kinase treatment? So any patient with these listed comorbidities, they should not be taken up for oral tyrosine kinase inhibitors. And patients who are having life-threatening disease progression or symptomatic disease or diffuse disease progression, these are the candidates whom you should consider for systemic tyrosine kinase inhibitor therapy. These, as I told, these are the major side effects seen with the tyrosine kinase inhibitors, and these are the reason why it's not suitable for all patients. Most common side effect is obviously hypertension, and patients develop cutaneous and mucocutaneous toxicity, hepatotoxicity, cardiotoxicity. So the point being, selecting a patient for tyrosine kinase inhibitor should be very strict, because of if any comorbidity is already there, on top of that, these side effects can make it worse for the patient. So ATA stand about tyrosine kinase is that it is good, but not without problems. And problem being its side effects. And as I mentioned earlier, sorafenib and lenvatinib has already been approved in US and European Union for patients with advanced rare refractory thyroid cancers. So is appropriate for TKI. So patients with measurable disease on imaging, patients whose tumors no longer take up RI or who have exceeded their lifetime dose. Patients with progressive disease or patients with more than one lesion, which is more than 1 centimeter in size, and they should be symptomatic also. These are the patients whom we can consider for tyrosine kinase. So role of immunotherapy, these are all very newer development in the area of thyroid cancer treatment. We mentioned in one of our previous slides that BRAF mutation is correlating well with the overexpression of PD-L1 and PD-1. So pembrolizumab is a newer drug which is specifically anti-PD-1, which can be used in rare refractory thyroid cancers. Sulfatinib is another newer drug. All these are basically under trial only. They have not been approved. Like pembrolizumab was a anti-PD-1 inhibitor. Acetolizumab acts against PD-L1, programmed death ligand 1 receptor. So coming to the current recommendation for managing a patient with rare refractory thyroid cancer, in patients who are having high risk metastatic progressive rare refractory cancer and they are not amenable to conventional therapy, then we can consider tyrosine kinase inhibitor. Provided, like we told earlier, they do not have any comorbidity which is going to affect their life. Then immunotherapy and resensitization therapy can be used after use of approved TKI such as lenvatinib or sorafenib. And radiotherapy or radiofrequency ablation or surgery for symptomatic distant metastatic lesion can be used, provided surgery is not possible. Like doing surgery at that area would do more harm. Like some critical structures are involved. Resecting complete resection is not possible, or complete attempt at a complete resection is going to land up the patient in more trouble than helping him. So these are the situations where we can think of radiotherapy or radiofrequency ablation. So, like we mentioned, the basis of rye refractiness is the downregulation of sodium iodine import. So people thought, why not re-differentiate these cells, make it capable of taking up sodium and iodine again. So there comes these two new drugs, which are shown some amount of redifferentiated, and patient have started taking up radioactive iodine, who were previously not taking up. And selumetinib and dabrafenib are two main drugs in this area. So to summarize, this is how we go ahead when we have an iodine refractory thyroid cancer. If the patient, we see whether the thyroglobulin values are high or is it in an increasing trend. After that, we categorize them into like this. If it is not, then we categorize it as a biochemical evidence of disease only, meaning there is no structural disease. Such patients are kept under observation. Then if these patients with a high thyroglobulin level are simultaneously having a cervical disease, see whether the lymph node is small or large. In case of a large lymph node, we go ahead with surgery. If it is small, such patients can still kept under observation. And in case of distant metastasis, look whether the distant metastasis is a very slowly growing small tumor. In such cases, we can observe. But if it is a single lesion and it is growing, then go ahead with local therapy. Like if it is in neck, or if it is and in local therapy, choosing surgery or embolization or EBRT depends on the previously mentioned factors, whether surgery is amenable or not. Like I told before, if surgery is amenable, that is the treatment of choice, irrespective of primary, recurrent, or metastatic disease. And in case of a rapidly progressive tumor, that is where we consider systemic therapy with the tyrosine kinase inhibitor. In case of progressive radioactive and refractory thyroid cancer, if a progression is only in a single lesion or only in a single organ, look whether local treatment is feasible. If feasible, then go for that. And in case of multiple lesions or in multiple organs, if it is a rapidly growing lesion, consider systemic therapy with tyrosine kinase inhibitor. If it is a very slowly growing lesion, he can be kept under observation, and we can take up from there. So this is basically what I just told. This is the NCCN guidelines for rare refractory thyroid cancer, which essentially these were the things which we have already discussed. So explaining this slide would be a repetition. So I'm just skipping this slide. So this is again NCCN guideline for bone and CNS meds. And in case of bone meds, we can consider intravenous denosumab or bisphosphonate, and cryoablation can be considered in bone meds. And if it is rapidly progressing, then consider tyrosine kinase inhibitors. So that brings the end of my presentation. Thank you. Hello.
Thank you, sir. Thank you so much for your informative session. If any of the participant is having any queries regarding the presentation, kindly drop your queries in the chat box. I guess there are no questions, sir. Thank you.
Thank you.
Next, I would like to welcome our last keynote speaker of our day, Dr. Bhavna Gadvi, Dean Principal Professor for Institute of Physiotherapy, Pearl University, India. Ma'am, over to you, please.
Good morning.
Very good morning, ma'am.
Good morning, everyone present in this online conference. First of all, I would like to thank the conference organizing committee for giving me this opportunity to share my knowledge. I represent Parul University, who is a very young university having NEC A++ grading. And it is just 8 years old university. But in last cycle of NEC assessment, we have got NEC A++, and I'm a Dean of Faculty of Physiotherapy and Principal of Parul Institute of Physiotherapy. I would like to talk on nociplastic pain. So now I'm going to share my screen. Is my screen visible to everyone?
Yes, ma'am, it's visible.
So they will talk about nociplastic pain, a new rehabilitation perspective. I'm sure every one of you have heard about pain and pain management and different types of pain. But a newer concept, I would not say it is a newer concept, but more familiar concept in a field of physiotherapy rehabilitation. So let us start with this picture. So in this picture, there are 4 patients who are having different pains and aches in the body part. 2 are very young. One is having neck pain or upper back pain, and the other one is having lower back pain. Both looks young, so it may be due to we can guess by seeing this picture that it may be due to faulty posture at work, it may be due to faulty posture at home, or abnormal ergonomics, because it seems they are not having any degenerative conditions which causes them to have pain. The lower 2 figures, which is obvious to see that these people may be having pain due to aging process. Now, if we see all these conditions, there are different origin or different types of pain where these people are getting pain. But if in common, if we see commonly, then the rehabilitation usually, what I have seen in my 30 years of clinical experience, and when any of these patients goes to clinic, the common treatment to be given is electrotherapeutic modality, which heat on the cryotherapy and along with this flexibility or active motions, and then later on strengthening. But the cause and the physiology of pain is not at all being focused or given weightage while assessment. So it is very important for us to know what are the different types of pain. So first and foremost is this is very important for a patient to know, because as a physiotherapist, we all know that what is pain? Does pain serve any purpose? We know that pain is a symptom and it is an alarming sign which says that there is something damaged in the body. But it's not just the damage which is represented in the pain. Pain has so many other aspects. So if I elaborate pain as a mnemonic, then pain stands for psychology, that is perception of pain, plus physiology, that is a tissue damage. A stands for alteration in health behavior. So it's not just about the magnitude of damage or the perception, but it's also about the overall health of a patient, which in turn leads to behavioral changes of the patient and perception. I stands for inflicted stimulus, that may be mechanical stimulus which causes pain, chemical, thermal, or emotional stimulus, which is very important for us as a physiotherapist to understand. And N stands for nervous system activity or sympathetic. Now let us go in detail. So according to International Association of Study of Pain and American Society of Pain, pain is described as an unpleasant sensory and emotional experience. So they also have said that pain is not just a sensory symptom, but it also represents the emotion associated with actual or potential tissue damage. Pain is a complex experience which not only includes somatic mechanism where the actual tissue damage is there, but it is also associated with psychological influences. It is always a subjective method, and that is why when we in rehabilitation, when we actually assess the patient as a whole. In a subjective part, we assess pain, but we don't just rely on the subjective tool which we are using for pain assessment or checking the recovery. Pain can be classified into three terms, but the basic classification which we use with the terminology is acute pain, which is the pain originated since last 3 months, and the chronic pain, which is the pain lasts for more than 3 months. But in terms of mechanism, there are two basic types of pain that are nociceptive pain, which is originated from nociceptors, and a neuropathic pain, which is originated from nervous tissue or neurological tissue. Now let's understand what is nociceptive pain. So again, a nociceptive pain has two types or two origin or two structures. When these structures get damaged, the nociceptive pain is symptomatic or evident. The one is somatic pain, where the pain originates from actual tissue damage of the somata like bone, joints, connective tissues, and muscles, which we consider in a physiotherapy as musculoskeletal pain. The other one is the visceral pain, where also nociceptive are situated, but they are the deep situated pain originated from organs, and they are mostly derived from systemic conditions or originated from systemic conditions like heart, liver, pancreas, gut. So visceral pain is always reflected on a body part, but it is not in a dermatomal presentation, but it comes in a sclerotomal presentation or a referred image we see. Because everyone knows that the commonmost symptom everyone knows about the visceral pain and its referred presentation is the pain due to cardiac condition on the left side of shoulder or arm. The kidney stone pain comes on the back, lower back, and these are the very common symptoms which everyone knows. And that is why when we differentiate the pain, the differential diagnosis, and consider the red flags, these are very important parts which we need to understand. Neuropathic pain has three different aspects, where the pain actually originates from neurological structures, basically the peripheral neurological structure, but it is not just always a neurological structure, it has different central nervous system involvement also. So what do we mean by these two things? Now let us understand first the nociceptive pain. So what happens whenever there is tissue damage? Whenever there is damage, be it a microtrauma or a macrotrauma. What is microtrauma? It is a repetitive injury where a small amount of tissues or cells are getting damaged. Like we see it in tendinitis, we see it in other soft tissues or musculoskeletal conditions. Like trapezitis, simple trapezitis, where the abnormal posture causes the damage or abnormal stretches of the tissues, and that is when the fascia gets inflamed and then there is a spasm which causes pain. So tissue damage causes the giant cell sensitization like this one. So whenever there is any pain, aches, or any discomfort in the body, the immune response occurs or sensitized. Whenever this immune system gets sensitized, not only the tissue which is actually damaged, but the giant cells which are present within the tissue or within the area of the damage, they also get sensitized, or the immune response gets sensitized due to giant cell sensitization within that tissue. So whenever the perception of this nociceptive pain is there in the brain or the somatic area, then what happens, even though the tissue damage is smaller or slower or very minimal, Due to this sensitization, the brain forces a lot of pain. But with this lot of pain, at an instance, gradually there is an adaptation of this pain, and then psychological or social stimulus may causes the inflammation. So it is like two ways. One is adaptation of the pain, so there is no stimulus to the psychological or social part. Second one is if they so we usually feels that whenever patient comes to you, patient says that today I have less pain or today I'm feeling good with the pain. Some days it happens that patient comes that I have a noxious pain or I have too much of pain. So the graph goes like this, right? Why? Because it's not just the tissue damage, but it's a psychological and social stimulus also gets influence with the intensity of pain, and this leads to pain and inflammation. Now, it's not every time the psychological pain means no physical symptoms or physical changes or if we say physiological changes, I think. Why? Because whenever there is any psychological stimulus, it would lead to a psychosomatic stimulation, and which leads to not just the psychological but the somatic influences which are there, which again leads to inflammation. Okay, what would be the symptoms if it is somatic pain or nociceptive pain? So pain will produce along with reduced circulation, which means ischemia. With the reduced circulation with ischemia, again the ischemia leads to ischemic pain, waste product built up within the area, which leads to formation of trigger points and reduced movement. So this is a vicious cycle which turns acute pain into chronic pain. And again, some stimulus which leads to acute inflammation and acute and chronic pain in turn. Now, neuropathic pain, the another word, the neuropathic pain, it has three clinical picture, I would say. The first is deafferentation, which is usually occurs in the tissue damage is there. Due to this tissue damage, there is a stimulation or inflammation to nervous, neurological tissue also. Like for example, in an orthopedic patient, you experience pain due to induced neurological inflammation or usually induced neurological system, nervous system also. But the another one which is very common is the sympathetic maintained pain. So what is the sympathetic maintained pain is, as I said, the psychosomatic pain, where there is stimulation of the sympathetic channels, and this sympathetic channel leads to another characteristic of pain. It's not just the pain, but it is there is increase that is allodynia. So along with pain, there is some temperature difference. So you feel sometimes you patient feels heat within the area. Then there is hyperalgesia to increase the sensitivity to pain. Autonomic pain with the hot flushes, with the autonomic changes with the body, tropic changes like changes in the skin, redness and all, motor dysfunction in the sleep underlies this. These are the common symptoms along with pain, which is sympathetic pain and a peripheral pain, which we call it as radicular pain, radicular fatigue, or radiculating pain, like sharp jabbing, throbbing, burning, tingling, kind of. So it's not just actually the pain, but along with that, there are other symptoms also. Now, recently, so where comes the neuroplastic pain? We have seen the somatic, that is the nociceptive and the neuropathic pain. But in 2021, it's like they created another, or they have invented another terminology, which is called the nociplastic pain in the mechanoceptic descriptor of a chronic pain. Now, what is this? The nociplastic pain is defined by International Association of Search of Pain or Study of Pain. Right? They say that pain that arises from altered nociception despite no clear evidence. So there is no actual tissue damage present, even though the patient feels the pain or a threatened tissue damage is there, still patient feels it. So many a times when patient comes to you and we assess the patient would say that my vast scale of pain or the intensity of pain is 6, 7, 8 out of 10, which is so severe pain. But when you examine the part of pain, there is no actual tissue damage like swelling, redness, brain. There's not much of the tissue damage which is evident in through symptoms and signs, but patient says that the perception of pain is so high. So it is due to the activation of peripheral nociceptor or evidence of vasodilation of the somatosensory system that causes the pain. Now, what is actually nociceptive pain? So there is a model which is called psychosocial model, which includes all these things. So if we see the pain, and if we see the deeper core of the pain, the types of pain, the physical and physiological aspects of pain are nociceptive, nociplastic, and neuropathic pain. But there are other consideration for understanding pain, which is a motor. Why motor? Because inhibition of any motor activity is due to pain. So actually, the patient does not. So if we say it that kinesiophobia, right? So what is kinesiophobia? There is nothing related to muscle, but due to pain, the patient stops moving that part. So the motor symptoms are also present, and along with that motor symptoms, there are psychosocial symptoms which are also contributing to the intensity or presentation of pain. The symptoms observed in nociceptive pain, it includes the multifocal pain. So it's like not a single type of pain, but it's a multiple. So here, the physiotherapist or pain management specialist makes the mistake that they just treat the physical aspect of pain. Sometimes they just treat the physical aspect of pain. Or we have seen the patients are really happy with some physios who does not give actually much more amount of treatment in the form of physical intervention, but they treat patient as a whole and the patient is more satisfied than the knowledgeable patient, the proper assessed patient, but they do not touch the aspect of psychosocial. So I just want to show you one video which explains the basic nociceptive pain. So, basic concept here I want to explain is the protein, which is being released into the cell, the glial cell. So many a times what happens when there is actual tissue damage or a condition where there is tissue damage, there is a stimulation of this glial activation. But even though the tissue is being recovered from the injury, this glial activation maintains due to sympathetic stimulation, and that is why the nociplastic pain continues even though the nociceptive pain gets relieved, right? So that is what everyone has to understand, that this stimulus has to be cut off or this stimulus has to be addressed well Even though the chronic pain is there without any enlarged reason, right? Or a magnified reason. So what happens with the neuroplasticity? Why this chronic pain syndrome? We call it as complex regional pain syndrome, chronic pain syndrome, psychosomatic pain. There are multiple many terminologies which we use. It's this that initially when there is tissue damage, the pain is produced. With the tissue damage, there is amplification of immune system, which I already explained. With this immune system activation, there is acute pain, enlargement or magnification of pain. But gradually, these two aspects, one aspect is the physical aspect, and second one is the psychosomatic aspect. The physical aspect is being recovered by itself because body is being consistent. The psychosomatic aspect, if it is not addressed, then it tunes down. The pain tunes down, but it does not goes to zero, and it remains constant. A smaller amount of pain remains constant for a longer time. Again, that constant smaller amount of pain magnifies with the psychosocial stimulation or with the other stimulation, which in turn leads to neuroplasticity. So if anyone can relate this with the patients who come, there are two types of pain. One patient here, they have a specific pain. Like this patient has a specific pain on the back or the radiating pain. So it's a very specific pain where you can relate that either it is nociceptive pain to the musculoskeletal system or a neuropathic pain to the neural system. But these pain in turn leads to central sensitization or disinhibition or facilitation, which is the response of the bodies to pain, which in turn leads to or converts to the neuroplastic pain. So here, even though these pain is there, the patient will feel pain here on the upper back, on the shoulder, or the hand, knee, neck, everywhere. So when patient comes with a complex regional pain or complex pain, so this pain usually has three aspects rather than only one aspect. So these three aspects. So in 1977, the model has already been established, but not much focused into the physiotherapy rehabilitation, which in turn evolved later on into 2000, like 2002 and then 2021, again, as I say. So there are three parts or three aspects which we need to address. It's the neurobiological process, which actually needs to be addressed. So maybe psychosomatic pain or visceral pain or a neurological pain. It has to be addressed. Along with that, the social contextual factor also needs to be addressed, and the psychological status, right? So a simple algorithm that the pain, if it is have organic pathology, then it has to be addressed with a neuropathic or a neuropathic pain. If it, there is any unidentified pathology, then psychological origin. But do not say this to patient that it is a psychological, because no pain only is evident with psychological. The cause is psychological, which in turn leads to somatic system, and the pain actually is there in the body. So it's not just the psychological or pain in the thoughts. Pain is there in the body, but the cause is psychological or psychosomatic. This is again a vicious cycle or a flow chart where you can differentiate. So if pain is experienced, for example, as I said, some takes pain in other way, some takes pain in other way. So what is that? So if the patient has no other sympathetic liberty to accept the pain as the pain to continue with, then patient would accept that pain and go ahead with the things. So that is how injury pain experience, no fear, confrontation, and recovery happens. So many times, similar patients would have a different outcome or a presentation. So this is what this chart shows, that no fewer than confrontation and recovery, but if the patient includes the psychological aspect, that is the pain catastrophizing, magnification of pain, which leads to pain-related fear and avoidance and hypervigilance. That is what I say, this kinesiophobia. If my trapezius is just paining, then what I would do, I would not move my neck for a longer period of time, and then in turn, that pain spreads to the different surrounding tissue. And that leads to disuse. Again, if I'm not able to use my neck, I'm not able to move my neck freely, my mental status would also get affected, which leads to disuse, depression, and disability. And again, this vicious cycle goes on and on until you address this pain experience, or you address the all three aspects, biopsychosocial. So negativity affecting, threatening, illness, information. Again, this is very important now in a day, in an era of internet informations. Everyone would Google what is happening. So many times when patient has back pain, they might have read that the back pain can be caused by any internal metastasis. Then the patient would get feared and panic, and this would lead to again intensification or magnification. So there is one study which has shown that the factor of physical or biological or actual tissue damage is the smallest part which contributes to the pain intensity or pain presentation. There are other aspects also like health factors. That is why everyone says that immunity, the build the strength, everything. So health factors, social factors. The patient is not much comfortable in a social environment, and the patient feels more amount of pain. Sleep factors, mood factors, and environment. All this together makes the pain intensity or pain presentation. The psychological factors like patient may experience the pain due to attention demands, cognition, that is perception of pain, expectation from the surrounding people or from the maybe physio, and the emotional factors. So this is cognitive behavior therapy, which is very much helpful by treating the patients with a chronic pain. And this is the flow chart which I had already explained in a previous slide. And that is what is, if less than 3 months, then not chronic pain, more than 3 months, then chronic pain, again divided into either nociceptive or non-nociceptive pain. If it is not nociceptive, then nociplastic pain. Nociplastic is entirely responsible for pain or not, evoke hypersensitivity phenomenon in hospital. So this is every physio should go through this algorithm to assess the pain. Do I have 5 minutes more to finish? Otherwise, it's all done for the pain presentation of nociplastic. If you allow me for 5 minutes, then I would just highlight few things to assess the pain so that you can even address the nociplastic.
Oh,
Do
yes,
I
ma'am.
have-
Yes,
do I
ma'am, you
have
can
5
have.
minutes more? Okay. So pain assessment as a physiotherapist, it is not just a physiotherapist, but pain management physician also. It is very important for us to assess the pain properly. The aspects which I highlighted in my presentation, we need to address every aspect of this. But usually, I would say about physiotherapists, I don't know about the other pain management professionals, but I would say about the physiotherapist, or the conventional method of assessing the pain is usually the intensity. So how much intensity do you have? What is VAS? What is the intensity of pain you feel? APRs, right? But describing the pain only in terms of its intensity is like describing the music only in terms of its loudness. So it is very important to see all the aspects of pain. So that is it. But before that, I just want to take the the assessment parameters, as a physiotherapist, what are the assessment parameters you also know. These are the pain that is provocative and palliative. Provocative is how does the pain aggravates, the aggravating factors, and palliative is relieving factors. Very important, because if there is a palliation, if there is any relief of pain, that means the pain is more of the damage to the tissue, if it is evident, or more of the, if you have addressed. But if it is a constant, it's nothing which works, then this is systemic illness of pain, right? We will consider that later. Quality, again, what types of pain, which also defines the somatic, neuropathic, and these quantity, as I said, the intensity of pain, the region and radiation. There is what is the area of pain. So if the area is defined, the dermatomal presentation, then it's more of the somatic pain. But if it is radiating in a proper dermatomal region, then it's again a neuropathic pain with either the peripheral or the deafferentation. Severity of pain, again, mild, moderate, severe, which we call it as, but it's the conventional way. We usually rely on the intensity. Timing and treatment already, we know discussing palliative. What is timing? Very important, 24 hours pain pattern, which we consider whether the patient experiences pain more in the morning or in the evening, or as it progresses in the night, 24 hours. Constant, it makes the pain identifiable for its region. If you see, I have explained a bit about inflammatory and mechanical pain. Mechanical pain is due to degeneration, or mechanical pain is due to the actual tissue anatomical damage, but inflammatory pain is the physiological damage, right? So inflammatory pain usually comes with the morning stiffness. Why? Because during the night time, there is inactivity of the muscle and sluggish circulatory system. Due to that, there is accumulation of waste products within the area of pain, which leads to stiffness, which leads to morning pain, aching pains in the morning. But as the day, as patient involves into activities, as the muscles starts pumping or increasing the circulatory waste product, like increasing the oxygenation, increasing the nutrients, increasing the waste products output, then the pain reduces, right? So inflammatory pain is more in evening as they progresses, or as patient involves into activity, the pain relieves. Whereas mechanical pain, there is actual tissue damage. When patient rests, the pain relieves. When patient has weight bearing activity, the pain increases. So increases with activity, and that is the degenerative pain progresses with day progresses, and evening there is so much of pain, right? Now, assessment of pain does not include intensity, but there are many other tools also, like VARS. And verbal rating scale is simple, mild, moderate, severe. NPRS, that is 1 to 100 or 1 to 10 also is nowadays very popular. Visual analog scale is an analog scale of 10 centimeter, where 0 is no pain and 10 is severe pain. We need to ask the patient to put a mark on this scale without having any numbers on the scale. And when patient says that, that is a pain perception, how much pain patient has or perceives. Pain drawing, that is front and back outline, and you can ask the patient to draw the area where patient has pain. Many times patient is not able to express the area, but when you give them a chart, they would be able to give it properly, and that is how we can even come to know that it is a non-separating of the plastic pain, where there is a discrete pain in more areas. And there are magnetic pain questionnaires where the explanation of pain is more. And for the pediatric patient, we are in color scale, where there is face, and Wong and Baker faces, where there is color scale, or where there is from green to red, or where there is faces from dull to bright, kind of, right? But whatever we have seen in the assessment, I just want to explain one thing which is very important for everyone to understand, that pain is a symptom which in turn patient perceives. But pain as a pathway, if you see, there are four aspects which is very important for everyone to understand. So what is transduction? Where the actual tissue damage happens and the nociceptive gets stimulated and it transmits. So transduction is the area where actually tissue damage is clear. Then transmission from the periphery to central nervous system to the ascending pathway, the pain reaches to the pain perception area. Then there is conduction to the spinal cord. So both the spinal tracts, which are very spinothalamic and spinoradicular tract, which conducts the pain to the higher centers, and then there is a pain centers which perceives the pain. Now it is very important for everyone that whatever patient says or whatever patient NPRs, fast, or whatever, that is the perception of pain. But we do not actually assess the pain at transduction, transmission, or conduction level. Right. So many a times there are, if you see the tissue, for example, patient comes with a swelling, redness, a presentation or a spasm. If you see the tissue, which is very actual damage, and if you ask the patient to have perception like fast, what is that, right? To give intervention, you see that into the tissue area, there is improvement in the signs. But when you ask the patient says the pain is same. Why? Because it is a perception which incorporates not just the biological aspect, but the physiological and psychological aspects also. So many a times, if you want to show that patient that there is a recovery, and that also helps them to reduce the psychosommatic stimulation, then the transduction and transmission aspect also can be addressed. So these are the different tests. So what is VAS or whatever I have seen is the perception level which we see. But there is another very simple method to analyze or to assess pain is at the transduction or actual tissue damage level pain is the current perception threshold. So CPT is the method where you can just simply use a small stimulator and you can just gauge or assess. You can just increase that intensity, increase the intensity gradually. The intensity where patient feels the pain is your current perception threshold. Feels the pain, the highest level of pain, where they cannot tolerate it, right? So that is. And then the actual tissue damage is lowered down. The chemical reaction within the tissue is lowered down, then this current perception threshold is also higher, like at a higher level patient. So I would give you an example. The PCOS patient in this conference may relate it very well. Many a times you start with IFT or a TENS with a very higher intensity. Why? Because their perception threshold is so low due to the increased amount of tissue or a chemical changes within the tissue, right? So, but after 5 days, 6 days, or some duration, the patient would feel that no, you can increase the intensity. So, you can increase the intensity, that means the threshold has improved so that the physiological or the pathological aspect of pain is addressed. And there are other things like now conduction study for conduction level and pain imaging MRI or blood cortisol level for transmission. But that is not a part of physio. You can do one transduction and perception. So along with that, you need to understand what are the red flags before starting with the management. So red flags are persistent pain, constant pain, shortness of breath along with pain, discoloration of pain. So shortness of breath and weakness identifies the cardiac or neurological or neural damage, discolored circular damage, swelling and redness in the joint with no history of pain, systemic condition, pulsating pain, again the circulatory compromises, in constant and severe pain. So these are the pains which is usually due to metastasis, any systemic illness where the physiotherapy management is on primary basis. Right. So that is all from my side for pain and obstructive pain and the pain assessment, which is very much needed for our to understand. The pain management aspect I'm not particularly into this. Thank you so much. One question my friend had. My friend had an accident with paralyzed leg during continuous feeling that person is going to feel So is this pain due to accident or an actual tissue damage, or it is the patient is feeling pain after there is some paralysis or there is some nuclear damage? Again, I think I have already explained the steps to follow. If it is actual tissue damage, then we can go with proper pain management treatment, there are multiple pain management treatment. If it is due to accident, the tissue damage is there, but there is, I would say, additional added effective effect of paralysis or again the psychological impact that which may have felt that there was paralysis and now it is another thing which is I'm coping, I need to cope up. So that may be that in this you may have to deal with the psychosomatic aspect. So psychosomatic aspect, the cognition behavior therapy was so good to me.
Thank you, ma'am. If any of our other participants is having any questions, kindly drop in the chat box, please. Thank you, ma'am. There are no more questions.
Thank you so much for this opportunity.
Thank you. So now I request all the participants who belongs to main hall kindly join the breakout rooms, and the main hall participants can stay in the same hall. For joining the main hall, in your screen you could see an icon called breakout rooms. Once you tap on the hall A, the join button will be seen. You can tap by joining it. All the participants, I request to join their respective halls as per the agenda. All the main hall participants can stay here, and rest of the participant who belongs to hall A kindly join the respective hall by tapping on the breakout rooms. If any of you having any queries regarding that, you can text us on chat box. If any of the participant is having any queries regarding that, they can ask out for help in the chat box. Thank you. Now I request, yes.
How do I know if I have to join for the breakout room? Is there any specific session mentioned for specific things?
Okay. The part is we have shared you the agenda, right?
Yes, yes, ma'am.
In that your name will be mentioned either in a main hall or else in the hall E. You can check out your name and you can join the respective hall.
Okay, okay, ma'am. Thank you.
Thank you. Now I request Ms. Salma to take over the session.
Good morning to one and all present here. I'm Salma and I'll be the moderator for the oral presentation and posters in the main hall. So I would like to welcome you all to the Allied Con International Multidisciplinary Conference on Health Sciences organized by BioLeaks and in collaboration with our esteemed academic partner, Benevolencia Foundation, Chettinad Academy of Research and Education, and Parul University. I'd like to welcome all our honorable session chairs and dignitaries. Our first dignitary is Dr. Sinjan Das, Director of Homeopathy and Psychiatry, Benevolencia Foundation, India. Our second dignitary for today is Dr. Nancy Vaibhav Gandhi, Assistant Professor, Neurological Science, Department of Physiotherapy, Sumandip Vidyapith Deemed to be University. And our third dignitary for today is Dr. Uma Maheshwari, PK MBBS, MD Associate Professor in Chettinad Academy of Research and Education, India. Welcome, sir and ma'am. I would like to confirm if our first 5 participants are present in the room right now. Yati Hemant Kumar, Dr. Habibu, Harika Madumbai, Loris Joseph, and Suja. Please respond in the chat box if you are present right now. Okay, Yati is present. Dr. Habibu, if you are present here, please respond to the chat box. Okay, so I'll explain the rules of the paper and poster presentations for today. Each participant will be allotted 10 minutes for explaining their study. And after 8 minutes, there'll be a reminder that only 2 minutes are left. Our judges and session chair will have 1 minute to ask the queries and questions to the participants. So we'll begin with the first participant, Yati Hemant Kumar, for the oral presentation. And the title of the paper is Formulation of Lactose-Free Breast Milk Using Partially Purified Lactase Enzyme from Bacillus clausii enzyme from Bacillus clausii. Yati Hemant Kumar, you can please take over.
Dr. Shinshan Das and Priyadarshini ma'am, can we start with our presentations, please?
Yes, you can.
Yes, sir. Thank you, sir.
Hello. Am I audible?
Yes, you are audible.
Yeah, this is Yati Vaidya from Sri N. Patel. Here I'm representing my work, which is on formulation of lactose-free breast milk, partially purified lactase enzyme, which was used in Bacillus clausii, which was isolated from the breast milk sample from the healthy mother volunteer. So let me explain about what is the lactase enzyme and how it works. Basically, lactose are the substrate for the lactase enzyme, and this lactose, that is the uh, disaccharide which converted into a galactose and glucose, they are monosaccharides. And then, as we are seeing here, that is the lactose intolerance that increases the age. When we seen towards the fetus that having the high amount of lactose and lactase enzyme, but when we are aging at that time, that the lactose intolerance increases and lactase enzyme is reduces in the nature. So here is the graph which shows the data that China having the highest amount of lactase intolerance. When we see all over the globe that having the high to moderate amount of lactase intolerance that occur. So let's see how it works when we seen towards a normal human being, they convert lactose, or we can say the metabolized by the lactase enzyme and convert to the glucose and galactose. They are the sugars and transport to the liver. On the other side, then see the lactose intolerant people that cannot metabolize lactose because they didn't have a enough amount of lactase enzyme. But on the other side, we can say certain organisms are also there, and this organism can digest it, but they convert into the H2 and carbon dioxide, and it will transport to the lungs and liver. So here my study is, which is on the isolation of bacteria from the healthy breast milk sample. We collect at least a 5 healthy breast milk sample from the healthy mother volunteer, and we get certain organism. We get a 5 individual species, but here we taken only one for it. And here we confirm the bacteria, which is probiotic in nature, by the gram staining, colonial and morphological characterization, and biochemical test was done. Here is the some isolates, that is the first isolate that I get from the healthy breast milk sample. Here is the or we confirmed it by the 16S rRNA sequencing, that is the Bacillus clausii, and here is the gram staining result. And here is the biochemical characterization that was done by us. And here the certain sugars, that is sugar utilization test in it, glucose and galactose, while lactose also produce acid, and the tests are positive in our case, and maltose and mannitol that not showing the positive result. And here is a certain biochemical test, that one which is the methyl red test. In case this methyl red test, we get a positive result, and in bogus Klusker test, we gave a negative results. Citrate utilization test, which is also negative, and then indole production test, which shows the positive result, while the hydrogen sulfide, which shown the positive, and urea hydrolysis, which shows the negative result, while in case of nitrate, which also shows the positive, and ammonium production test, which shows the positive result, while the starch hydrolysis also occurred, and the gelatin test, which shows the negative result. So by that we can confirm it. Our organism shows a certain activity, and we show certain bacterial species, that is the one which is the Bacillus clausii. We go for the molecular identification and the of the 16S rDNA sequencing of it. And here is a band which shows isolated genomic DNA, which is on 1% agarose gel, and which shows a 900 base pair. See, 900 base pair. Then we are go for the screening for potential probiotics. Is it working properly or not, or when it is surviving in the like in vivo condition or not? So we are go for the acidic environment, resistance test, and bile tolerance test. In case of acidic test, we provide the pH2 buffer to our organisms, and we incubate for the 24 hours. As same, and the bile tolerance, we provide 0.3% bile salt, and we checked for the organism to survive to particular condition, like same as in our gut. So here is the taste that shows the low pH condition, that our organism survive from all 3 hours incubation, while tolerance to high bile salt concentration also. And the third test we was done by the gut adhesion capacity. In that, gastric, intestinal, and GI simulation was occurred. For the gastric simulation, we go for the 3 hours, as same is in our gut condition, and the intestinal condition we provide a 4 hours treatment, and then GI simulation for 7 hours simulation. So here is the result, which shows that our organisms survive from all 3 conditions. And then we go for the auto-aggregation and co-aggregation study. So here is the data that shows the our organisms survive, or our organism shows the positive aggregates against this control result. It will survive and go for the positive result. And on the other hand, co-aggregation study was also done against the certain gram positive and gram negative strain. Gram negative strain, which shows the positive impact or positive co-aggregation percentage as compared to the gram positive results. And then we go for the hydrophobicity of the cells, and we check for the whether it is survive in the different types of buffers condition, like different type of reagents, we can say the hexadecane, chloroform, which is acidic reagent, and ethyl acetate, which is the basic in nature. So in that, the chloroform, which shows the positive impact or positive surface hydrophobicity. Then we go for the enzyme production, which is basically a lactase enzyme. We go for the partial purification of our sample or enzyme, and here we choose the ONP as a standard for our result, because ONP, which shows the positive, we can say the indicator reagent against the beta galactosidase. We can simultaneously use the name of a lactase or beta galactosidase. And here is the data. We go for the ion exchange chromatography. And here is the purification step that we conduct. We take the cell-free supernatant. This is
Excuse me.
basically a broad.
Yes,
Hello.
you only have 2 minutes left, so make sure you summarize within
Yes,
the
yes, sure. So here we get a purification fold, which is higher in the ion exchange chromatography as compared to ammonium sulfate precipitation. Then we go for the check for the pH and temperature study of our lactase enzyme. That is basically optimum pH is 6, and the 60%, 60 degrees Celsius temperature, our organisms survive and get the optima. And here is the SDS page that shows the 34 to 47 Dalton of or kilodalton of our molecular weight. And we also go for the glucose and galactose standard observation test. Then it shows the linearity. Then again, we go for the 3 different type of condition. We immobilize our lactase enzyme and check for the 3 different condition. One, which is lactose with the immobilized enzyme, and then milk sample with the immobilized enzyme, and another which is milk sample with the immobilized bacterial culture rather than the lactase enzyme. So here is the lactose plus immobilized enzyme that gives the higher result as compared to the another one. So here I can say there are the certain procedures and things that we have to done. We have to perform. We will perform in the future. There are the certain studies that we taken out. And here is the before breast milk composition is here. And as compared to the after hydrolysis, so our result shows some positive aspect, and we need to do a lot more. Thank you.
session chairs. I request you to go ahead with your questions if you have any.
No question from my side.
Yes, sir. Thank you. So we would like to call our next participant, Dr. Habibu, for oral presentation. And the title of the paper is Effect of Plant Extracts and Their Combination on Quorum Sensing Mediated Virulence Factors of Staphylococcus aureus, MTCC 737. Dr. Habibu, if you are present, please respond.
Yeah, good morning.
You can start with your presentation. Your time starts now.
Yeah, good morning all. I'm happy to tell you I'm going to present my paper, Effect of Plant Extracts. So, effect of plant extract and their combination on foreign sensing mediated virulence factors of Staphylococcus aureus. So, in the introduction, Staphylococcus aureus is a gram-positive pathogenic microorganism causing skin infection, and mostly if there's wound, and also it can cause bacteremia if it is opportunistic, it finds its way into the body. The challenges we're having about antibiotic is that the resistance have been happening and we're having issues. So treating bacteria with the conventional antibiotic, they easily get resistant. So we are trying to see that the current trend in treatment is researchers look into the virulence factors, which actually degrade the virulence factors produced by this organism and
affect the adaptive rather than the survival mechanism. So not necessarily killing the bacteria, but it can affect their comfortability in the environment, thereby clearing them. And plant extract are promising in doing that. And the issue we have here is combination. So individual plant extract may have active component, but it is seen that maybe the active component in individual plant might not be enough. So a combination will try to improve and increase the efficacy of the drug used. So the main point of this paper is the polyherbal combination. That is to see whether we are going to have a combination of different plant extracts so that we can have synergistic and other positive interaction. So the hypothesis here that we have is whether combination can have effects more than the individual, or the individual and the combination does not have any difference. So the aim is to investigate the current sensing activity of the aqueous extract of three plants: Bacopa, Honere, Calotropis gentiana, and Acalypha indica. The objective actually is to look at the minimum inhibitory concentration, to look at to optimize the concentration for the combination, then to look at the effect of the plant as an individual and combination on the current sensing. So the materials here, I don't need to spend much time here. We collect the plant, we prepared by extracting lyophilized, then we optimize the concentration of the plant. Then three virulence factors actually were seen: catalyst activity, hemolytic activity, and lipolytic activity of this plant individual and in combination. So MIC was done. We discovered that the Bacopa have 5 mg per ml, Acalypha with 2.5 mg per ml, Calotropis gentiana 2.5 mg per ml. But this concentration, we have to optimize it again, because the virulence factors inhibition should be in a very comfortable concentration. If it is too high, they might not even produce the virulence factors. So we optimize this concentration using response surface methodology, because this plant we are seeing in wound treatment. So we try to optimize, although results not shown for this, about how they affect the fibroblast cells in vitro experiments, so that we can see what concentration will be effective for the wound healing. So we see a much more reduction in the concentration from what we see in the MIC down to like 15 microgram per ml for Bacopa and 11.6 microgram per ml for Acalypha, then 1 microgram per ml for Calotropis gentiana, which is still below the MIC, which is also good. Then the three virulence factor we check, the catalyst here, still in all the experiments, summarily is that the individual have, the combination have more effect than the individual compared to the control, untreated control. And still it has a very significant difference compared to the individual also. So meaning the combination have more positive interaction with compound to give a positive result that we have seen here. And in summary, 3 medicinal plants, as I said, were used. This is the MTCC 373. That is the Staphylococcus aureus that we have used. Below the MIC we've seen here, then the combination appeared to be superior, giving a much more inhibitory property. And the prospect of this research is try to say that the combination, the different pathogenic bacteria, not only Staphylococcus, should also be tried with individual combination. Then the combination, this can be made into a formulation which might be also better in trying to treat this kind of wound opportunistic organisms. Then bioassay guided fractionation also should be done, so that you can characterize the active component, which was not done in this research. Then gene expression studies also will give more information to which genes are affected in this, and also more virulence factors can also be checked with these plants and their combination as well. So I have to acknowledge the contribution of Professor Lilama, adjunct professor deemed to be university, and a research scholar by name is Karima Lakati, for her contribution and critical suggestion in this experiment. These are my references. Thank you very much. I'm done. Hello. 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 I can't hear anything. Hello. No.
Session moderator, are you present there?
Yeah.
We can hear you.
Dr. Habibu is done and no question from our side. So we can progress.
Stop sharing.
I would like to ask the session chairs to go ahead with the questions if they have.
Don't have any question. You can progress further.
Okay. So next, I would like to call the next participant, Harika Madhupai. And the title of the paper is In Silico Pharmacokinetics and In Vitro Antimicrobial Evaluation of Tricin against Vancomycin Resistant SODs against vancomycin resistant SODs. Harika, please go ahead. Okay, your time starts now. You can go ahead.
ऑडियो ही नहीं आ रहा।
Ms. Harika, you have to explain the poster. So please start with your presentation. You are not audible to the audience.
Am I audible now?
Yes, you are audible. You can start.
Thank you.
You have 7 minutes for now.
Okay, thank you. This is the in silico pharmacokinetics and in vitro evaluation of ricin.
Ms. Harika, you are still not audible.
Hello?
Yes.
Is it audible now?
Yes, it's audible now, but if you're having connection issues, you can resolve it. We can move ahead with another participant.
Am I audible now?
Yes, you're audible.
Okay, thank you. In this study, the chrysin being a phytochemical isolated from basically present in honeycomb.
Sarika, you are still not audible.
Try to resolve it and get back.
Yes. Till then, I would like to call the next participant, Gloris Joseph. If you are present in the room, please respond.
Yes, hello ma'am, I'm present.
Okay, so next we have Gloris Joseph presenting detection of clostridioides difficile toxin from patients with diarrhea in a tertiary care hospital, Mysore. It's an oral presentation. Gloris Joseph, you can go ahead with the presentation. Your time starts.
Good morning, Joe. I'm Gloris Joseph. My title of the study, I'm presenting the abstract of my study titled Detection of Clostridioides difficile toxin from patients with diarrhea in a tertiary care hospital, Mysore. It was guided by Dr. Ranjita Shankar Gowde. So the abstract of my study is Clostridioides difficile is the leading cause of hospital-acquired diarrhea, where a wide range of antibiotics is the primary risk factor for the development of Clostridioides difficile infection or CDI. So the main purpose of this study was to identify the glutamate dehydrogenase, which is GDH enzyme of Clostridioides difficile from stool samples, which is collected from diarrhea patients with a history of prior antibiotic exposure, and to identify the C. difficile toxin A and toxin D in GDH positive samples. So I have taken 50 stool samples and have screened, I screened for the presence of GDH enzyme and toxin A and toxin B by using alpha enzyme. Diarrheal samples and non-diarrheal stools were collected from the patients who were on antibiotics for more than 7 days, and I have excluded the patients with the antibiotics for less than 7 days. So out of 50 stool samples, 8% were found to be positive for GDH enzyme and 2% were positive for C. difficile toxin A and toxin B. And Clostridioides difficile infection can lead to Clostridioides difficile associated CD/AD manifested by diarrhea, and in fulminant cases by pseudomembranous colitis that can lead to death. So the most commonly used test for CDI diagnosis is the detection of GDH enzyme along with C. difficile toxin A or B toxin assay. GDH positive C. difficile expressing either toxin A or toxin B are pathogenic in nature, and C. difficile positive GDH and negative for toxin A and toxin B indicates the presence of non-toxigenic strains. So the keywords which are used for my study are prostatitis difficile, glutamate dehydrogenase or GTH, toxin A and toxin B, antibiotic associated diarrhea, alpha, enzyme linked fluorescence assay. Thank you.
I would like to request session chairs if they have any questions.
Hello, am I audible?
Hello, yes sir.
Yes, sir, you're
Yes,
audible.
yes, sir.
If you have mentioned their antibiotic exposure, what sort of antibiotic exposure you are considering of?
So the antibiotics which is mainly causing C. difficile infections. Sir.
What are the examples?
Examples including azithromycin, ceftriaxone, then third generation of cephalosporins, clindamycin.
Have you any study with the norfloxacin or ornidazole or fluoxacin?
I think no, sir.
Okay, okay, thank you.
Yeah, thank you.
Thank you, Flores Joseph. Now we'll move ahead with our next presentation by Suja. It's the study on occupational stress-induced immunological and inflammatory biomarkers alteration in doctors. Suja, if you're in the room, please respond.
Yes, ma'am. Am I audible, ma'am?
Okay. Yes, you're audible and your time starts now. You can go ahead with
Yes.
your presentation.
Yes, ma'am. A very good afternoon to one and all present here. So, myself, Suja, I'm a scholar in Chettinad Academy of Research and Education, and the title of my study is a study on occupational stress-induced immunological and biomass alterations in doctors, and under the guidance of Dr. Priyadarshini Shanmugam ma'am, professor and head of the department, Chettinad Academy of Research and Education. So, as we all know, everyone in the world exhibits some sort of stress in all fields. Not it is not up to a certain field. So, according to Hans Sale, he defined stress as unspecific reactions of the human body to any kind of need for change in 1936, and healthcare professionals, especially doctors, experiences more stress. All experiences doctors, but I have selected particularly doctors. They exhibit more stress because being a doctor is physically and emotionally demanding. And research conducted in different parts of the world reported that this kind of psychological stress was related to alterations in immunological and inflammatory biomarkers. And the main rationale of this study is that, to our knowledge, this was the first study that mainly analyzes the relationship between the job stress and immunological changes in the southern part of Tamil Nadu, especially in doctors. And the main aim and objectives are to evaluate the occupational stress-induced changes in immunological and inflammatory biomarkers in doctors. My objectives to study the socio-demographic or job-related characteristics through question to score the job stress or occupational stress in doctors, to study the immunological markers, mainly CD3, CD4, and CD8, and also to study inflammatory biomarkers, interleukin 2, 6, and tumor necrosis 6 factor alpha. And my last objective is to compare the stress levels and the levels of the biomarkers. The property is Coimbatore and the sample size is 100. My inclusion criteria include apparently healthy doctors, both male and females of age group 25 to 55. And exclusion criteria includes the disease conditions like diabetes, immune system disorders, HIV, and the subjects with long-term corticosteroids all were excluded. And the data collection was done after getting concerned from the doctors, and the questionnaires were administered. And from the questionnaire, we have calculated the demographic and job characteristics. And also it was scored, the job stress in the doctors were scored based on the perceived job stress scales with small modifications. And after getting concerned for blood sample collection, under the standardized protocol, blood samples with 5 ml were collected from each of the participants. The samples were transported immediately for flow cytosis, mainly for CD4, CD8, and CD3 cells, followed by the cytokine analysis, which was performed by ELISA. Moving on to the results, from this study, based on this gender distribution of the doctors, the predominant participants were female doctors, which is of 56%, and the male were only 44%. And the age distribution, when looking on into the age distribution, the predominant age group is 36 to 45 years, that is 53%, and 25 to 35 years, which is 33%, and lastly 46 to 55 years, which is only about 14%. And and the health problems, based on in the questionnaire, the health problems were asked. Most of the doctors were repeatedly says that 81% of them were had sleep disturbances, followed by a headache, that is about 62%, and 44 with depression, 36 with back pain, and 12 with hypertension. And the job stress factors also calculated in that 78% of the doctors had additional consultations other than their regular work, and also they are not 70% were not satisfied with their job, and salary issues, conflict with the co-workers, travel time to workplace, and also telemedicine inquiry plays an important role as a job stress factors. And the stress level were scored based on the questionnaire. The stress level was classified as moderate stress and high stress. Among 89 subjects undergone moderate stress, and 11 of them had high stress, and they are the stress score well calculated, and they are very and are significant, statistically significant. And coming to the biomarkers, the cells, the CD3 cells, 4 cells, and CD8 cells were not significant, but the percentage of the corresponding CD3%, CD4%, and CD8%, when they are compared with the moderate and high stress, they mainly CD4 and CD8% show significant. They are statistically significant. And moving on to the inflammatory biomarkers like IL-2, IL-6, and TNF alpha. Only IL-2 and IL-6 shows significant changes when compared with the moderate stress and high stress. So to discuss and conclude, occupational stress was found to be a major problem in the present scenario. The participants, as I said, like mostly clinical doctors, and about 36% of them were females within the age group of 35 to 48. And based on the stress level, the doctors in the study were moderate to high level of stress, and they reported job dissatisfaction and additional consultation as the frequent stress factors, and also sleep disturbances, headache, and depression as their major health problems. And moving on to the biomarkers, mainly CD4% and CD8% were found to be significant when compared with both moderate and high level of stresses, which shows an alterations in the inflammatory cytokines like interleukin 2 and interleukin 6. So from this study, we concluded that occupational stress might alter the level of immunological biomarkers, thereby releasing the inflammatory biomarkers that cause a wide range of health issues in physician. So future studies with a higher sample size in different occupational fields may provide better results, and to confirm either CD4% or CD8 as a measurable stress biomarker. I thank my guide, Dr. Priyadarshini Shanmugam ma'am, for his support and guidance in all my endeavors, and also these are my references. Thank you.
Thank you, Ms. Suja. I would like to request session chairs, if they have any questions, please go ahead.
You have mentioned here the stress factors, but how the stress score has calculated?
The score, there was a score range, like people with who are exhibiting a similar points. We are scoring based on we have a set of questionnaire. We have given a mark like 0 to 4 or 4 to 0. Like that, we are scoring the stress, and we have calculated based on the perceived stress scale. We have calculated a low, medium, and high stress group. So we have
so you choose perceived stress scale for this.
Yes, sir. Not exactly the scale, but we have some modification, which is which mainly requires for our study. We have modified slightly the perceived stress scale.
Okay, and you have mentioned here occupational stress factors, but
Yes, sir.
how will you differentiate it between the occupational stress factors from the domestic stress factors or any
So,
other?
sir, we have included like mainly the salary issues and job dissatisfaction. All we can included in job stress factors when compared to other like other issues like health problems. We can taken in domestic stress factors, sir.
But those are also playing the significant role in the enhancement of the biomarkers, no?
Yes, sir. Yes, sir.
So you have to be differentiating from the original one, the predisposing factors from the occupational factors also.
Yes, sir.
Okay, thank you.
Thank you, sir.
Thank you, sir. Ms. Harika, are you ready with your presentation?
Hello.
Yes.
Am I audible now?
Yes, you're audible. Just wait for 2 minutes.
Okay.
So before we go ahead with Ms. Harika's presentation, I would like to ask Dr. Neeti Mishra, Dr. Tanvi Bansal, Dr. Khushbu, Dr. Meenakshi, and Dr. Dharti to please respond in the chat box if you are present in the room. The next 5 presenters, please be ready. Okay, Ms. Harika, you can start with your presentation now. Your time starts now.
Thank you. Let me share my screen. Good afternoon, everyone. I'm Harika from Department of Biotechnology, Andhra University. I'm here to represent my work on in silico pharmacokinetics and in vitro antimicrobial evaluation of chrysin against vancomycin resistant SRS. We all know that Staphylococcus aureus is a devastating pathogen in humans, poultry, and dairy animals. The emerging and rising of methicillin-resistant Staph aureus and vancomycin-resistant Staph aureus have become a major public health concern. And resistance in Staph aureus occur with different patterns, like multiple factors, including penicillin-binding protein, antibiotic resistance genes presence like BLAST, MECA, MECB, VAN-A, VAN-B, and VAN-C genes, and the production of biofilm associated genes like ICA-D. Here, the chrysin is a phytochemical mostly found in honeycomb. In this present investigation, chrysin in combination with vancomycin was tested for antimicrobial and antibiofilm activity in vancomycin-resistant Staphylococcus aureus. The samples were collected from deceased birds and milk samples. The isolated samples were culturally characterized, biochemically and molecularly confirmed using certain specific techniques. Later on, the antibiotic resistance was determined using 19 antibiotics, and the first graph shows the incidence of this antibiotic resistance. And coming to the quantitative assessment using the microtiter plate assay, later molecular detection of antibiotic resistance genes was done, in which the PCR amplification showed the presence of all the genes. Later, in phenotypic detection of biofilm production on Congo red agar medium, all the isolates have turned the red color medium into the black color, indicating the presence of biofilm production. In the quantification of biofilm production, the OD reading shows that the graph is around 0.45, indicating the organism is a strong biofilm producer. And in the PCR amplification, the biofilm producing genes ICA-D was present, and the vancomycin resistant gene VAN-C was also present. And coming to the focus of the study, when the chrysin is combined with vancomycin on Muller-Hinton agar medium, it shows the zone of inhibition has been increased. And also the Congo red agar medium, also the zone of inhibition has increased compared to the vancomycin alone. This confirms the increase in the efficacy of vancomycin when associated with chrysin. Coming to the SEM analysis, it clearly confirmed that vancomycin in combination with chrysin has clearly disrupted the biofilm production compared to the control, which doesn't have any antimicrobial agent. When this chrysin is docked, it shows the binding energy of minus 8.31 energy, which indicates the high binding energy. In the in silico pharmacokinetics, the boiled egg method was used to determine the blood-brain barrier mediation and all. And here, coming to the in silico pharmacokinetics, this chrysin is a CYP3A4 inhibitor, stating that actually vancomycin is being eliminated without metabolism from the body. This chrysin being the cytochrome P450 inhibitor, this increases the binding of the vancomycin in the system. It can prolong the duration of action of vancomycin by preventing its metabolism. This chrysin prevents the metabolism of vancomycin by binding to cytochrome P450. In the boiled egg medium, gastrointestinal absorption penetration through blood-brain barrier was done. And in some non-target toxicities, it shows non-AIMS test around 0.89. Concluding this shows that chrysin when combined with vancomycin effectively increases the efficacy of vancomycin. Again, that can be used against the organisms to prevent the production of biofilm. And this chrysin alone does not have any antimicrobial activity. And according to this in silico pharmacokinetic studies, it does not exhibit animal toxicity also. Thank you.
Thank you, Ms. Harika. I would like to ask session chairs for the questions.
No questions from my side.
Thank you, sir. We'll move ahead with our next presentation. Next oral presentation is a study on prevalence of nomophobia and associated factors with smartphone users among medical students and academicians by Dr. Neeti Mishra. Dr. Neeti Mishra, please respond.
Yes, is my screen visible?
Yes, your screen is visible and you're audible and your time starts.
Okay. Okay, so good afternoon, everyone. The topic of my study is a study on prevalence of nomophobia and associated factors with smartphone users among medical students and academicians. Mobile phones have become indispensable part of our lives. It is estimated that approximately 6.628 billion smartphone users are used globally. About 94% of those in age group of 18 to 29 are users of smartphone. In recent times, there seems to have been a transformation of the cell phone from a status symbol to a necessity because of the countless perks that a mobile phone provides, like personal diary, dispatcher, calculator, camera, etc. Nomophobia has recently been used to describe the discomfort or anxiety caused by the non-availability of a mobile phone, personal computer, or any other virtual communication device in individuals who uses them habitually. The controlling factors for smartphone usage are restriction from family, security concern, and feeling of missing out, that is FOMO. Since the younger generation is the latest consumer of the mobile phones and the underage 25-year group in professional colleges like medical colleges use mobile phones quite frequently. Hence, the current study was an attempt to explore and collect precise data relevant to the causes, symptoms, and complications of nomophobia that may help to take the effective preventive measures. The need of the study is research in this area is critically important given the persuasive use of cell phones by young adults, especially college students, to determine possible association of nomophobia with socio-demographic determinants and to assess the health-related consequences. Significance of study. The present study makes several contributions to the literature in area of research. It is performed to investigate which of a wide range of cell phone activities are most closely associated with cell phone addictions. Research in this area is critically important given the persuasive use of cell phones by young adults. Coming to aims and objectives, first aims to find the prevalence of mobile phone usage addiction among medical students and to find correlation between mobile phone usage and associated factors. The study design was cross-sectional correlation study, sampling design being purposive. The study population comprised of medical students and academicians of either sex of 18 to 50 years of age, sample size 200, and the study duration was 6 months. In the selection criteria, the inclusion criteria included age group of 18 to 50 years of both sexes, medical students pursuing graduation and post-graduation in college using mobile phones, academicians in medical field using mobile phones. Student and exclusion criteria included students who were not belonging to medical field were excluded, students who were not using mobile phone were excluded, students not willing to participate in the study were excluded. Coming to the procedure part, a self-administered questionnaire, that is Google form, was used to study the mobile phone dependence among the subjects. The questionnaire was distributed among 500 students pursuing education, internship, board day scholars, and hostlers through WhatsApp and email. These mobile phone dependent students were designated as nomophobes. The data were collected to elicit information on demographic and psychographic aspects of the respondents. The individual responses thus obtained were then compiled, processed, and analyzed. Outcome measure. The demographic variables included age, gender, and education. The psychographic variables included attitude towards usage of cell phones, mobile phones dependence, and associated anxiety. The questionnaire focusing on nomophobia had various components like duration, anxiety, stress experience because of faulty connections, loss of mobile and battery discharge, frequency of use per hour, purpose of maximum usage, phone usage at night, hindrance in concentration at work, and increased phone dependency. Coming to the stats part, the statistical analysis was performed using IBM SPSS version 20. Descriptive statistics were carried out for age, sex, and outcome measures. Prevalence of nomophobia was evaluated percentage-wise. Pearson correlation test was performed to identify the relationship between associated factors related to mobile phone usage, and the level of significance was kept at p less than or equal to 0.05. So the total, there were 31 questions in the Google form. So these are the questions. Frequency of checking smartphone per hour. Do you check your smartphone without reason? What is the purpose of maximum usage? Do you check your smartphone early in the morning? What is the context of maximum usage? Do you believe using smartphone causes below mentioned problems like headache, eye strain, neck pain, disturbed sleep, fatigue, etc.? Do you have a perception of phone using hampering academic performance? Is it possible for you to stay without a smartphone? Do you feel connected when you have a smartphone? Do you feel very lonely at those places where you cannot have access to phone? Do you feel like you are missing something when you are on the phone? Do you feel that if you don't use phone, you will be outdated about everything? Has using a smartphone increased your independency? Do you sleep keeping your phone next to you? Is the phone first thing you see in the morning? Do you agree that a smartphone provides easy access of information to you? Do you think smartphone is convenient method of completing tasks online? Do you have tendency of replying immediately as soon you see the message? Does your phone much only because you have excess data and want to utilize it? One becomes very eager to check the phone to see whether he have got any calls or message as soon as the work is finished. Do your parents scold you saying that you are always on the phone? Do you think face-to-face interaction is now replaced by video calls? Do you have insecurity issues that if your number goes into wrong hand? Have you ever stopped the work just to check the messages? Do you get tensed when your friends haven't seen your message and don't reply back? If you forget your phone at home, will you compulsory go back to get it? Do you feel worried about leaving your phone at home? So these were the questions and their responses were recorded in the percentages. Now coming to the overall result. So the percentage prevalence of nomophobia was about 90% of the population has some or other symptoms of nomophobia. About 90% of the population checked their smartphone without reason. 68% have a perception that phone use was hampering academic performance. 64% felt that if they didn't use the phone, they will be outdated about everything. About 68% were tensed and annoyed when their friends have seen their message and don't reply back. About 49% felt worried about leaving your phone at home. Now coming to the correlation part. So we can see this table demonstrates weak positive relation of lonely at those places where you cannot have access to your phone along with insecurity issues. If your phone goes into wrong home, worried about leaving your phone at home, and tensed and annoyed when you, tensed and annoyed. Next is correlation between positive correlation of duration with phone use without reason, hampering academic performance, lonely without phone, and getting tensed. These are my graphs showing positive correlation and negative correlation. Coming to the discussion part, the present study was conducted on prevalence of nomophobia and associated factors with smartphone users among medical students and academicians.
Excuse me, ma'am.
Yes.
You only have 2 minutes left.
Okay. So studies by Kanmi et al. And Pavitra have found that social networking have the highest proportion of users among medical undergraduate students. The most common health-related consequence of mobile phone dependence observed in the present study was eye strain, followed by headache, and 26.5 participants considered that it does not cause any health issues. The proportion of respondents checking their smartphones soon after waking up was 55% in the study of J. Jirisha, whereas Kanmani et al. Found this proportion to be 69%. Coming to the conclusion, the present study indicates almost 90% of population has some or other symptoms of nomophobia, and 90% of population check their smartphone without reason. So while usage of mobile phones include positive outlook also, like attending online classes, referring for academic purpose, the extended addiction and dependence of mobile phones have to be avoided by the students. Limitation, variation in use of mobile phones, region state was not analyzed. The tool used in the study was not translated into different languages. Questionnaires provided to the respondents in English format only. The sample in the study was mainly restricted to education section or society of the population like academicians and clinical therapists. And the questionnaires adopted were only tools used for collection of data. Future recommendation, cluster sampling could be used to understand region state-wise perspective, and questionnaires could be translated into regional languages, and survey can be performed via interview method to include uneducated population from society also. These are my references. Thank you.
Thank you, ma'am. I would like to request session chairs to go ahead with their questions.
Hello, Dr. Niti.
Yes, sir.
Have you checked the screen time?
Have you
checked the screen time for samples?
Screen time?
Yeah.
No, sir. Total screen time have not been checked. But total duration has been checked, sir.
It is the continuous duration or it is interrupted manner?
It is interrupted duration, sir.
So from the interrupted duration and from the continuous duration, the data should be different, right?
Yeah, it can be different, sir. Definitely.
Okay. Thank you.
Thank you, sir. Now I'd like to invite our next participant, Dr. Tanvi. And the title of the paper is Effect of Static Erect Neck Pose in Patients with Cervical Spondylosis, a randomized controlled trial. Dr. Tanvi, please respond.
Yes. Yes. Good afternoon, everyone.
Good afternoon. Let's start with the presentation.
Is my screen is visible to everyone, ma'am?
Your screen is not visible.
Yes, ma'am. Ma'am, can you start from your laptop? 1 minute. Is it?
Dr. Tanvi, you are not audible.
Ma'am, am I audible?
Yes, you are audible now, but it seems like you are having a network issue. So you can resolve it, then we'll move ahead with our next participant.
Is it visible?
Yes, it's visible.
Yeah, may I start my presentation, ma'am?
Yes, you can.
Okay. So topic of my study was effect of static erect neck pose in patients with cervical spondylosis, a randomized control trial. So as we all know that cervical spondylosis is the degenerative condition involving the cervical vertebra, and gradually it affects the disc also, and if it is not being treated, it affects the limb also as a radicular pain. So significance of my study is in usual clinical practice, the intermittent cervical traction is usually being given by the machine, but the machine is very costly. So it might not be possible for the each and every therapist to buy the ICT machine at the personal clinic. But as an alternative, I wanted to find out the exercise which is effective as the ICT, as equal as ICT. So this study was designed to give a innovative exercise therapy to the literature along with the conventional therapy. So in methodology, my study design was a randomized control clinical trial. Then study population was patient with cervical spondylosis. Then sampling technique was purposive sampling. Then sample size was calculated on the basis of pilot study, and I have included the 57 samples. The study duration was 1 year. Selection criteria, these were my inclusion criteria: patients with cervical spondylosis, both male and female, participants who gave informed consent to participate in this study, then age between 30 to 60 years, acute and chronic neck pain, medially stable patients and medically stable patients, and diagnosed cases of CS. Exclusion criteria were cervical trauma with fracture and dislocation, rheumatoid arthritis, osteoporosis, history of previous cervical injuries and surgeries, any infectious disease of spine, any inflammatory disease of spine, any contingent deformity of cognitive deformity of cervical spine, then patient with prolonged steroid use, then tumor and metabolic disorders. Am I audible, ma'am?
Yes, ma'am, you're audible. You can go ahead.
Thank you, ma'am. Yeah. Procedure. Participants were preliminary screened based on the inclusion and exclusion criteria. The purpose of the study was explained, and written informed consent was obtained from all the subjects. Demographic details and a baseline assessment were taken from the participants. They were randomly allocated into two groups, Group A and Group B. In Group A, static erect neck pose plus conventional exercises were given, and in Group B, only conventional exercises were given. On the first day of the treatment, the assessment was taken, and the outcome was only NPRS, which I have included. And 5 times patient have performed the static erect neck pose in a day, and the exercise was repeated for the 5 days per week up to 4 weeks. Post-assessment data of pain was taken at the end of the second week, at the end of the 4th week. To avoid the bias, patients were blinded. So this was a single blinded study. The static erect neck pose to be in the sitting position on the higher stool or low stool. Patient was instructed to sit with erect trunk. Then patient was instructed to erect the neck by stretching the cervical spine as if stretching something from cow, assuming like erect and extended giraffe neck pose. Patient was supposed to hold the erect neck pose statistically for 5 seconds. No forward head pose was allowed, and no any accessory trick motion was allowed. And the same pause was repeated 7 times in a single session, and patient was asked to perform the same session 4 times at the home in a day. The conventional treatment which was included, that is active range of motion exercises of neck, isometric exercise of neck, and along with the strengthening, then shoulder pressing exercise, chin tuck exercise, stretching of trapezius, and stretching of scalene muscle. Statistical analysis was done by using Jamovi version, that is 2.3.28. Normality was checked by using Shapiro-Wilk test. As data of both the groups were not normally distributed, further data analysis was done by using non-parametric ANOVA test, that is Friedman test, for comparison of effectiveness between different time intervals in both the groups. So table shows the normality of the data. It was tested by the Shapiro-Wilk test. Here it shows that data of both the groups are distributed. Table 2 shows repeated measure ANOVA of the group A, in which p-value is less than 0.001. That indicates that there is a difference between different time zones. Then in the third table, there is a pairwise comparison by the Durbin contour. There is a p-value between each two groups, that is pre and first week, that is pre-NPRS and second week, pre-NPRS and the fourth week. Then first and second week, then first and week and the fourth week, and second week and the fourth week. All the values were falling less than 0.01. That indicates there is a difference on all the two groups. Now coming to the fourth table, to differentiate in which interval the difference was maximum, I have looked up for the mean. So PRS mean was 7.26 in the group A. First, at the end of the first week, it was 4.41. At the end of the second week, it was 3.15. And at the end of the fourth week, it was 1.74. In the table 5 shows ANOVA for the group B, that is Friedman test. It is less than 0.001. That indicates there is a difference among all the time interval in the group B also. But next table shows the pairwise comparison of the group B between pre-NPRS and first week, pre-NPRS and second week, pre-NPRS and fourth week, first week and second week, first week and fourth week, and second week and fourth week. So p-value is less than 0.001. It also shows the difference. But the table 7 shows in which time interval the difference was how much. So I have looked out for the mean in the group 2 also. So in pre-NPRS, the mean was 7. At the first week, it is getting reduced only 6. At the second week, it is getting reduced up to the 5. And at the fourth week, it is getting reduced up to the 4.26. Looking to the graph 1 shows that initially the pain was above 6, nearer to the 7, and at the end of the first week, it is getting fall up to the 4th or 4 NPRS. And in the graph 2, we can see at the end of the first week, still the NPRS is nearer to the 6. That indicates that in the group A, at the first week only, we get the improvement in the NPRS by giving the static erect neck pose compared to the only conventional exercise. So coming to the discussion part, in the present study, total 15 folks were included. They were randomly allocated into two groups. So 27 patients were in each group. Erect neck pose along with the conventional exercise. In group B, only conventional exercise was taken before treatment, and at the end of the recovery. These were the statistical analysis. Now, into the clinical significance, yes, the MCID of NPRS is 1.5. This shows
Dr. Tanvi, you are not audible. Can you please check your network connection?
Yes, sure, ma'am.
And also, you only have 2 minutes left, so please summarize and finish before.
MCID of the group A showed more improvement at the end of the first week compared to the group 2. So coming to the conclusion part, this study concludes that static erect neck pose along with conventional exercise provides early and better pain reduction statistically as well as clinically compared to conventional exercise alone. So therapists must add it as an adjacent treatment along with treatment. So coming to the last slide, limitations and further recommendations. As in this study, only 57 participants were taken. Further, with the larger study carried out, only NPRS we have taken. Further, study can be done with the other outcomes, and the exact mechanism behind the pain relief in first week of the treatment is not known. So, further study can be done with the EMD studies. Thank you.
Thank you, ma'am. Session chairs, please go ahead with your questions.
No questions from my side.
Thank you, sir.
Thank
Next, I would like to invite Dr. Khushbu Valodwala for their oral presentation on effectiveness of manual pertubation exercises to improve balance and mobility in subacute stroke patients. Dr.
Yes,
Khushbu,
ma'am.
please
May I,
respond.
Yes, ma'am. Am I audible, ma'am?
Yes, ma'am, you're audible, and you can start with your presentation. You have 10 minutes.
Is my screen is visible to you all?
Yes, your screen is visible.
And am I audible clearly?
Yes, you are audible.
Okay, ma'am. Just wait a second. So, good afternoon, everyone. So today's my topic for the presentation is effectiveness of manual perturbation exercise to improve balance and mobility in subacute stroke patients. Ma'am, wait a minute. I'm just putting it in a slideshow. So the World Health Organization defines the stroke as developing clinical signs of focal disturbances in the functions and symptom lasting 24 hours or longer with no apparent and cause other than the vascular origin. So worldwide, about 20 million persons are suffering from the stroke, while 5 million may die due to consequences of the stroke, and 15 million may survive, and among them, 5 million may be disabled due to the stroke disability. The inability to maintain the upright position within the limitations of the stability or base of support was defined as balance impairment. Balance impairment has, however, a favorable link with the elements of function such as mobility, activities of daily living, and falls. Sensory motor function in the patient lead to loss of static and dynamic functions, and there will be deterioration in the proprioception, somatosensory function, and vestibular dysfunction. After the stroke, the first posture to achieve is sitting, which is required for most of the activities of daily living, which is including eating, transferring, bathing. Within 6 days of beginning of the stroke, 93% of the patient can attain 1 minute of independent sitting balance. In the stroke patient, loss of balance can lead to increased postural sway, reduction in the reduction in a weight on the affected side.
PPT is
lower
not
leg
visible
and
to everyone. PPT is
ma'am,
not
wait.
visible to everyone.
Okay, ma'am, just wait a minute. Now is it visible, ma'am?
Oh, it's still not visible. Please
Not
open.
visible.
Just wait a minute, ma'am. Now is it visible, ma'am?
Yes.
Yes, now it's visible.
Okay. So, according to the principle of specificity, physical training exercise physiology in the training process is an effective intervention and should be incorporated closely to mimic a specific function in our balance recovery exercise. So, the objective of the study states that the aim of the study is to check the effectiveness of conventional physiotherapy treatment to improve balance and mobility in subacute stroke patient. Other aim of the study is to check the effectiveness of manual perturbation exercise to improve balance and mobility in subacute stroke patient. And the third aim was to compare the effectiveness of conventional physiotherapy with manual perturbation exercise to improve balance and mobility in subacute stroke patient. So significance of the study states that many studies were done to check the effectiveness of various treatment in stroke patient. These studies were done in different phases, that is acute, subacute phases of stroke, but there is a scarce literature regarding the balance and mobility in subacute stroke patient. Hence, the present study provide a supportive literature to check the effectiveness of manual perturbation exercise to improve balance and mobility in subacute stroke patient. So the hypothesis states that null hypothesis states that there is no significant difference between conventional physiotherapy treatment and manual perturbation exercise to improve balance and mobility in subacute stroke patient, while alternative hypothesis state that there is a significant difference between conventional exercise and manual perturbation exercise to improve balance and mobility in subacute stroke patients. So for materials and methods, we have taken the data from various OPDs of Surat, and it is experimental kind of study. So sample size was calculated here was 32. We have included subacute stroke patient and purposive sampling technique was used. Duration of intervention was around 4 weeks and the duration of the total study was 1 year. We have included the males and females having 45 to 75 years of age with the first episode of cerebrovascular stroke. The post-stroke duration 6 to 8 months participants were included. ability to walk independently with or without assistive devices, and subjects who were willing to participate were included. Subjects who are having neurological deficit, sensory dysfunction affecting balance, those who are having perceptual dysfunction, musculoskeletal dysfunction, including lower extremity and instability in pain, recent trauma or surgery in past last 6 months, they were excluded from the study. So we have basically taken ethical clearance from the institutional ethical committee, and the purpose of the study was explained and written informed consent was taken from the participant. Subjects were preliminary screened on basis of inclusion and exclusion criteria, and on first year, first week, their demographic data were collected. They were allocated in group A and B, and their BBS and TUG outcome measures were taken for the assessment of balance and mobility. So here they were divided into two groups. Group A, that is for conventional physiotherapy treatment, in which we have given the mat exercise, bed mobility exercise, ambulation, transfer, weight bearing activities, gait training, that was given for the 45 minutes for 5 days a week and 4 weeks. While in group B, we have given the manual perturbation exercise along with the conventional physiotherapy treatment, in which 15 minutes of extra perturbation training was given, and safety precautionary method were given by the therapist. Manual perturbations were given in different positions, that is sitting, kneeling, and standing position, with the 10 seconds of hold with the 10 repetition, and that were given for the 5 days a week, that is given for the total 4 weeks. At the end of the 4th week, again the outcome measures, that is BBS and TUG, were taken and data were collected and analyzed. So these are the two basic outcome measures which we have analyzed, that is work balance scale and time upon go test. So data analysis was done by using IBM SPSS version 20. So baseline demographic data were collected, that is including age, gender, BMI, MMSE, and Brunstrom scale, along with the outcome measures. In present study, total 32 participants were included, and for the normality of the data, we have taken the Kolmogorov test of normality. So on that basis, so these tests are mentioned here, and here we have found the majority of the parameters are having more than 0.5 value. So the data is basically normally distributed. So for the that purpose, we have used the paired t-test for the within group comparison, while unpaired t-test that used between group comparison. So these are the various parameters from that we have found the mean values, that is given in the table. So age is basically mean value having 50.90, BMI is 23.40, MMSE is having 29.07, Brunstrom having 4.15, pre-BBS values were 46.81, while post-BBS we have the 51.34, and pre-TUG values were 18.99, while post-TUG values were 15.3. So these are the different graphs which represent the gender distribution as well as the MMSE mean and SD values. So for group A and group B, we have done the paired t-test. So for group A, we have taken the two parameters, that is BBS and TUG, and here we can easily find out that these are the values which will suggest the less than 0.05. So it is showing the significant result, while group B is also having the values less than 0.05, which will again signifies the significant result. Between group comparison was also done for both the outcome measures, and here we have not found the significant improvement in work balance scale, while TUG value are improving in group within group between group comparison. So for discussion part, we have done the paired t-test for
Excuse me,
the
ma'am.
within. Yes,
Please
ma'am.
summarize your study in the
Yes,
next
ma'am.
2 minutes.
So we have used paired t-test for the within group comparison, while unpaired t-test was used for the between group comparison. So the same study was done by the Minjoni et al, and they have also found that at least 1 hour of balance training will improve the gait and balance in the patient with the stroke. The similar kind of study done by the Belia, and they have given the perturbation based balance exercise in older patient. They have found that there will be less chances of fall after training. So this will suggest our study. This will support our study. Authors here have also found that the balance and mobility will be improved by using the coordinated multi-joint movements, which are effective for the maintenance of postural control. So the same type of study will be done by the Shamve Cook also, and they have given the conclusion that giving small and large manual exercise, external perturbation will lead to improvement in the balance. So conclusion of the study states that along with the physiotherapy, conventional physiotherapy additional balance exercise that is perturbation based exercise will be helpful for the patient to improve the balance and mobility in stroke patients. So limitation of the study states that we have taken small sample size. It is purposive type of sampling method. Follow-up of the participants were not taken, and we have included the ambulatory stroke patient. So further study can be done with the large sample size, and wide range of area can be covered. So clinical implication of the study states that there is always window of opportunity in every condition by early rehabilitation program. So we can improve the certain functions, mobility, and ultimately quality of life can be improved. So these are the list of references. Thank you, ma'am.
Thank you, ma'am. Session chairs, I request you to go ahead with your question.
No questions from my side.
Thank you, sir. Okay, we'll move
Thank you,
ahead with
sir.
our next presentation by Dr. Meenakshi on the topic frailty, oral health, and nutrition in geriatric denture wearers, a cross-sectional study. Dr. Meenakshi, please respond.
Hello. Good afternoon, ma'am. Ma'am, just a minute. I'm just opening my PPT. Just give me 1 minute, ma'am.
Yes, sure.
Thank you.
Dr. Meenakshi, please start with your presentation.
Ma'am, just I'm unable to open it, ma'am. Just a minute, ma'am. I'm just opening it.
Ma'am, if you're having network issues, you can resolve it and you can present in the next one.
Ma'am, opening it, ma'am. Just a second, ma'am, please.
Okay. Ma'am, you can resolve your issue. We'll move on to the next participant. Dr. Dharti,
Ma'am, what time can I resume my PPT, ma'am?
After the next participant.
Okay.
You can resolve your issue till then.
Thank you so
Dr.
much.
Dharti, are you present in the room? Please respond.
Yes, ma'am, I am there.
Okay, so we'll start with Dr. Dharti's presentation on comparison of primal reflex release technique and positional release technique on pain and function in patients with chronic plantar fasciitis, a randomized control trial. Dr. Dharti, you can start. You have 10 minutes to present your.
Okay, ma'am. Is my screen visible, ma'am?
Yes, your screen is visible and your audio is.
Okay, thank you, ma'am. Good afternoon, everyone. Myself, Dr. Dharti Pansala, working as an associate professor in SPB Physiotherapy College, Surat. I'm going to present a research entitled Comparison of Primal Reflex Release Technique and Positional Release Technique on Pain and Function in Patients with Chronic Plantar Fasciitis. This is one of the randomized control trial. Plantar fasciitis is an inflammation of the plantar fascia and the perifacial structures. It is defined as a syndrome resulting from repeated trauma to the plantar fascia. At its origin, on the medial tubercle of the calcaneus. It is estimated that 1 in 10 people will develop plantar fasciitis during their lifetime. Primary reflex release techniques, that is PRRT, is a treatment paradigm that involves down-regulating an overstimulated autonomic nervous system in order to reduce pattern of pain. It addresses the neural system by resetting hyperaroused primal reflexes within the body. The treatment involves providing 12 seconds of light, swift sensation in the form of repetitive deep tendon reflexes that tap or stimulate the skin to inhibit painful areas. Positional release technique is a manual therapy approach designed to enhance muscle flexibility. This technique involves maintaining the muscle in a certain position to reduce muscle relaxation as opposed to placing the muscle in a lengthened or stretch position. PRT is an indirect myofascial technique targeting the neurologic component of neurovascular myofascial somatic dysfunction. Also, the number of patients of plantar fasciitis and its prevalence rate is increasing day by day. Furthermore, research is needed to make plantar fascia back to the normal function immediately. Though various treatment techniques have been documented for PF, it is still difficult to choose best guidelines for the management of plantar fasciitis. Various methods of treatment exist with their own claims of success without any attempt of comparing the maximal effective methods. Since there is a paucity of studies about the comparative effect of PRRT with other techniques till now, hence there was need to evaluate the same. The purpose of this study was to compare the effect of PRRT and PRT along with conventional physiotherapy on pain and function in patients with chronic plantar fasciitis. Aim and objectives of my study was to compare the effect of primal reflex release technique and positional release technique on pain and function in patients with chronic plantar fasciitis. My secondary objectives were to study the effect of PRRT on pain and function in patients with chronic plantar fasciitis, to study the effect of PRT on pain and function in patients with chronic plantar fasciitis, and to compare the effect of PRRT and PRT on pain and function in patients with chronic plantar fasciitis. The hypothesis were null hypothesis. There is no significant difference between the effect of PRRT and PRT on pain and function in patients with chronic plantar fasciitis. While my alternative hypothesis was there is no significant difference between the effect of PRRT and PRT on pain and function in patients with chronic plantar fasciitis. Research methodology, my research design was randomized control trial. Population was patient with chronic plantar fasciitis. Sampling technique was purposive sampling. A study duration was 1 year. Sample size 36 participants with 18 subjects in each group, which was calculated on the basis of pilot study. From the basis of pilot study, the effect size was calculated and it was put in G power. From that, this sample size was calculated. Outcome measures was numerical pain rating scale and patient-specific functional scale. Inclusion criteria, both male and female, age group between 25 to 65 years, diagnosed and referred cases of plantar fasciitis by orthopedic surgeon, duration more than 3 months, patients having one or more trigger point with elicitation of nociceptive startle reflexes over plantar aspect of foot. And my exclusion criteria were any history of lower limb fracture, presence of other musculoskeletal complaints in the lower limb, surgery for plantar fasciitis, subject who had taken corticosteroid injection in heel past 3 months, clinical disorder where therapeutic ultrasound is contraindicated, such as infective conditions of foot, tumor, calcaneal fracture, metal implants around ankle, ankylosis or congenital deformities, any neurological disorder, neurological pain, or diabetic foot. The procedure, the ethical clearance was taken from Institutional Ethical Committee. Total 49 patients were screened. Out of that, 36 were selected for the study on the basis of inclusion and exclusion criteria. Participants were explained the procedure and written informed consent obtained. Pre-treatment values of NPRS and PSFS were measured. They were randomly allocated in 2 groups by sealed envelope method, and subjects were blinded on either type of intervention and to which group they were belonged. So this study is a double-blinded study. Description of groups. Group A received primal reflex release technique plus conventional physiotherapy. Intervention was given 5 days per week for 2 weeks. There were 5 components of primal reflex release technique. That is medial knee reset, peroneal tendon reset, gastrocnemius reset, hamstring reset, and tocal. So among them, the tocal portion was performed twice up to 2 minutes, while the other 4 segments were performed once for 12 seconds each. In group B, there is a positional release technique and conventional physiotherapy. Intervention was again given for 5 days per week for 2 weeks, and these are the technique for PRT, that is palpation of tender point. The tender point was palpated, and then the PRT technique with plantar aponeurosis stretches were performed. The 3 repetitions of this position should be held with rest interval, and the duration of each treatment session lasted for 15 minutes. While conventional physiotherapy for both the groups was, intervention was given again for 5 days per week for 2 weeks for both the groups. That the conventional method treatment were include ultrasound, exercise for intrinsic muscle strengthening, active ankle exercises, stretching of plantar fascia, and stretching of tendo-Achilles. Statistical analysis for this study was done by using the SPSS version 15 software. Sapir-Wicks test was applied to check the normality. All the quantitative data of this study were not follow the normality. Hence, the non-parametric tests were used to analyze the data. Baseline characteristics were compared to check the homogeneity between intervention group. Predman test was used to analyze the pre-intervention, post-intervention, and follow-up differences with each group. That is the non-parametric test for repetitive measures. While the Mann-Whitney U test was used for between group analysis, that is baseline and pre-post comparison. Confidence interval was kept at 95%, and the level of significance for all the statistical data was set at 0.05. This is a baseline characteristic of subjects of group A and B. You can see the p-values are more than 0.05. That means there were no significant difference between the group before intervention. That means both the groups were homogeneous before the intervention for all the demographics as well as for all the outcome measures. This is
Ma'am, please summarize your study within the next 2 minutes.
Yes, ma'am, sure. This is the result of my study. This is an intergroup comparison. You can see there is a significant difference in all the p-values. That means both the group has improved post-interventionally compared to pre-intervention. This is the intergroup comparison. This is all here also the p-values are less than 0.05. That means both the group has significant difference in improvement of NPRS and PSFS. The PRRT group was significantly improved in pain intensity on NPRS and function on PSFS after 4 weeks of intervention compared to PRT group. This additional reduction of pain intensity is maybe the effect of primal reflex release technique. This is based on the premise that the overstimulation of body's primal reflex creates pain and keeps painful pattern occurring again and again. So according to LMS, the startle and the withdrawal reflex are the main primal reflexes linked to the fight or flight responses of the autonomic nervous system, are called upon to protect the body when encountering a painful starting or stressful experience following injury or even perceived tissue damage. So these reflexes lead to muscle spasm, and possibly pain can be due to maladaptation or abnormal stimulation of the nervous system. These primal reflexes influence motor behavior and can be sustained in a state of hyper-readiness, upregulation, leading to patterns of pain. So PRRT addresses the neural system by resetting the hyper-aroused primal reflexes within the body and downregulate the overstimulated ANS in order to reduce pattern of pain. So PRRT also addresses muscle and joint receptors along with their spinal modulation. This technique facilitates rapid neural reboot. This neural reboot resets the neural control of joints, muscles, and fascia to release joint restrictions, trigger points, and facial restrictions. So the result of my study is also supported by the study done by the different author in previously, but most of the study.
Your time is up.
This is the
I
conclusion.
request the ma'am, your time is up.
Okay, ma'am.
I request the session chairs to please ask the questions.
No question from my side.
Thank you, sir. Meenakshi ma'am, are you ready with your presentation?
Yes, ma'am. Just sharing the slide. Can I start sharing the slide,
Just
ma'am?
2 minutes. Just two
Yes, yes,
minutes.
yes, yes.
So the next 5 participants, Miss Neha, Miss Pinky, Dr. Anita, Ruben Thomas, Dr. Shweta, Tanvirul Sakib. If you are present in the room, please respond in the
Yes,
chat
ma'am.
box. Okay, please be ready with your presentations. Dr. Meenakshi, you can start.
Ma'am, just a second, ma'am.
Okay. Dr. Meenakshi, please start with your presentation.
Excuse me, ma'am. I'm having small glitches, technical glitches. Is it possible? Can I present after the next presenter, ma'am?
Yeah,
After
no
half an
problem.
hour or so?
Yes, no problem. You can resolve
Okay,
your
thank
issue in text in the text box.
Sure, sure, sure.
We'll
Thank you
continue
so
with
much.
the next presentation.
Yes, yes. Thank you so much, ma'am. Thank you.
Thank you. Miss Neha Verma, are you there? Please respond. Miss Neha Verma.
Am I audible, ma'am?
Yes, you're audible.
And my
So we'll
screen
start
is
with Miss Neha Verma's presentation on comparison of Bosu ball exercise with versus Otago exercise program on balance and gait in patients with diabetic neuropathy.
yes.
Ma'am, you can start. You have 10 minutes to complete your presentation.
My PPT is visible.
Yes, it's visible.
The title of my study is Comparison of Bosu ball exercise versus Otago exercise program on balance and gait in patients with diabetic neuropathy. Diabetic neuropathy is an ordinary disorder and is expounded as signs and symptoms of peripheral nerve dysfunction, patient with diabetes mellitus, and DPN is a progressive degeneration of the peripheral nerve that affect the sensory, motor, and autonomic component of the nerve, causing loss of protective sensation, intrinsic foot muscle dysfunction, and foot anastomosis. The significance of the study: Bosu ball exercise and Otago exercise program have been effectively used as a treatment in balance issues and in prevention of falls in elderly patients. But till date, very less studies have been done comparing the Bosu ball exercise with Otago exercise program in patients with diabetic neuropathy. The aims and objective of the study were to see the effect of Bosu ball exercise in patients with diabetic neuropathy, to see the effect of Otago program in patients with diabetic neuropathy, and to compare the effect of Bosu ball exercise program versus Otago exercise program in patients with diabetic neuropathy. The null hypothesis states there is no significant difference between both the treatment regimens, and alternative hypothesis suggests that there is a significant difference between Bosu ball exercise and Otago exercise program. Methodology: the study design was a pre-post interventional study. All the patients with diabetic neuropathy purposive sampling was done. Study duration was 1 year. Sample size with dropout was 40, and study setting was various OPDs in Surat. Inclusion criteria: patient diagnosed with type 2 diabetic neuropathy, BBS score of 35 to 45, and male and female in the age group of 50 to 75 years. Exclusion criteria was people with recent fracture, surgery, psychiatric illness, any other neurological problem, vestibular disorder which can affect balance, any cognitive impairment, perceptual dysfunction, and gross visual visual field deficits. These were the material and tools, including the Bosu ball and the score sheets for the dynamic gait index and work balance scale form. The outcome measures which are used were balance for work balance scale for the balance and dynamic gait index for assessing the gait. This is the consult flow diagram showing the procedure. Like 46 patients were assessed for eligibility, and after the exclusion,
40 patients were randomized into both the groups, 20 patients in each group. The intervention, both group received the conventional treatment protocol for 20 minutes of time duration with 5 minutes of rest period. Treatment was given to all the participants for 5 days per week for 4 weeks. Group A received the boostable exercise for 30 minutes. Each set of exercise was for 5 to 8 repetition with rest of 2 minutes. And Group B received the Otego program for 30 minutes. Each set was 5 to 8 repetition with rest period of 2 minutes. This is the group A, boostable exercise, including the exercises like unilateral heel raise, marching, knee bending, split stance with torso rotation, and one leg stand. The group B Otago exercise program included sit-to-stand exercise, sideways walking, backward walking, one leg stand, and toe walking. Statistical analysis and results. Statistical analysis was done using the SPSS version 20. Shapiro-Wilk test was applied for the normality testing, and my data was normally distributed. Both the groups were matched in terms of age, pre-BBS score, and pre-DGI score. The baseline characteristics of the qualitative variables were also matched. Intergroup comparison of BBS for both the groups shows improvement in both groups. And the intergroup comparison of BBS, that is the Buck Barrel scale for both the groups, shows more improvement in the BOSU ball exercise program as compared to the Otago exercise program. Same way, when the gait was measured using DGI, the intergroup comparison has shown improvement in both the groups. But while comparing the both groups together, the mean difference for BOSU ball exercise program was 5.2. That was more than the Otago exercise program. So the purpose was to examine the effect of BOSU ball and Otago program on balance and gait in patients with diabetic neuropathy, and the results suggest that BOSU ball exercise has shown more improvement than Otago exercise program. The exercises on unstable surface generate rapid changes in the length of the ankle ligament by stimulating the ankle in multiple plane of motion. So generating afferent stimuli and reflex motor responses to produce rapid joint stability. So instability devices like BOSU ball trainer undermine the principle of exercise, that is specificity, which poses the specific demands on the neuromuscular system, thus by improving the balance in motion for dynamic activities. So the conclusion is the 1 month of training imparted to both the group was effective in improving balance and gait in diabetic neuropathy patient. However, BOSU ball exercise was more effective than Otago exercise program. Performing exercise on unstable surface like BOSU, the subject found the exercise to be more motivating, valuable, and a fun-filled skill, which are safer, effective, and a challenging balance exercise program for patients with diabetic neuropathy. So finally, we concluded that the BOSU ball exercise is better than Otago in improving balance and gait in patients with diabetic neuropathy. These are my references. Thank you.
Thank you, ma'am. I request the session chairs to please go ahead with the questions. I assume there are no questions. Thank you, ma'am. I would like to call Miss Pinky Gupta for the next presentation. If you are present here, please respond.
Hello. Madam, can I start sharing my screen? Can I start presenting? Is that
okay? Yes, ma'am, you can start.
okay? Okay, just give me a second.
We'll start with Dr. Meenakshi's presentation. Miss Pinky Gupta, you will be the next one. Please be ready with your presentation. Dr. Meenakshi, ma'am, please
Is
start
the
with
screen
the
visible? Yeah, is the screen visible, ma'am?
Yes, the screen is visible, but your presentation is not visible.
Presentation not visible, huh? I've just shared the presentation right away. Is it visible now, ma'am?
Yes, it's visible. You can start.
Okay. Okay. Good afternoon, everybody. Warm greetings from Heritage City, City of Palaces, Mysore. I'm Dr. Meenakshi from JSS Dental College and Hospital. So my topic of today's presentation is correlation of oral health, diet, frailty, and cognitive function in the institutionalized elderly, an observational cross-sectional study. So to introduce the topic to the audience, the process of aging presents forward a wide array of changes, be it physiological or psychological, rendering the elderly population more prone to geriatric syndromes. Symptoms generally in the aged mainly decline in the hemostatic reserve compensatory capacity of all organ systems. Individuals are more susceptible to stressors. When the threshold of an individual's reserve capacity crossed overwhelming and unpredictable response at times, even as a symptom of an actual disease. Hence, the individuals with low reserve capacity are more frailer and more prone to diseases than the others. And hence, frail individuals have higher chance of, or geriatric individuals have higher chance of frailty or inclination towards diseases. So what are the other symptoms which we could actually see in frail individuals? There is dysfunctional metabolism, chronic inflammation, oxidative stress, malnutrition, endocrine dysfunction, and lack of immune system functioning or alterations in the immune system mainly contribute to frailty. From a general perspective, the older individuals will contribute substantially to the healthcare demands, as this population has a decline in general well-being and quality of life and is generally frailer than the younger population. So how do we define frailty? Frailty is a biophysiological disorder that affects many activities of daily living, characterized by diminished physiological reserves and resistance to stressogenic insults. Frailty actually has a multidimensional and multisystemic nature, leading to a marked susceptibility to a cluster of adverse health-related events such as falls, injuries, disability, hospitalization, institutionalization, dementia, and leading to death. Oral health is a new concept when considering the frail older individuals, and increased life expectancy is contributing to a growing scientific interest. The oral frailty phenotype is a novel construct proposed as a conceptualization of age-related gradual loss of oral function driven by a set of impairments that worsen the oral daily functions. So that includes loss of teeth, poor oral hygiene, inadequate dental prosthesis, or lack of ability of patients to manage and maintain the dental prosthesis, difficulty in chewing associated with age-related changes in swallowing. Hence, oral frailty per se has been defined as a decrease in oral function together with a decline in cognitive and physiological functions such as oral microbiota and Alzheimer's disease neurodegeneration. So the need of the study is the life expectancy is continuously increasing in this aging population. So according to WHO, by 2050, 80% of all older people will live in low and middle income countries. Countries like Brazil, China, and India will have slightly more than 20 years to adapt to a change from 10 to 20% in the proportion of the population that is older than 60 years. So how can we, dentists, contribute towards early identification and provision of care of special needs if required? And hence, the oral signs and symptoms, which are the indicators of frailty, needs to be assessed. Poor oral health are highly prevalent among older individuals and has been linked to general health and chronic diseases, including different components of frailty. So poor oral health throughout life worsens with time, and this breakout of new diseases is also common, and expectation to new dentures, especially in edentulous patients, also increases over a period of time. There is competing financial priorities, lower value, which leads to lower value on oral health. So hence, can oral frailty and physical frailty be correlated in the context of the Indian subcontinent? The geriatric medicine, often overlooked in traditionally structured health services and in the epidemiological research. These syndromes cross organ systems and discipline-based boundaries. When accompanied with their multifactorial nature, they challenge the traditional ways of viewing clinical care and research. And hence, this epidemiological scenario and lack of definitive evidence regarding the correlation of oral and physical frailty in context of the Indian subcontinent can create an impact on the sustainability of healthcare systems nationwide. Hence, the need of my study is does a gender predilection exist for oral and physical frailty due to cognitive dysfunction? So taking that into consideration and owing to the biological and social construct, the aging pathway and associate outcomes also appear quite differently for either genders, be it males and females. And hence, the purpose of my study is to define a direct correlation between cognitive dysfunction, oral frailty, and general frailty and malnutrition in the elderly population residing in Karnataka beyond 60 years of age. Additionally, this study also aims at finding a gender predilection, if any, for the targeted population. So how do we measure the frailty, oral frailty, cognitive function, and malnutrition? So these are the defined self-administered questionnaire already available, pre-validated, which includes modified REFS, oral frailty index number 8, mini mental examination or MMSE for the cognitive functional assessment, and mini nutrition assessment questionnaire to assess the malnutrition. So this is an overview of the questionnaire along with the scores which we have utilized for conducting the study and administering the questionnaire to our geriatric population. So this is the mini nutrition assessment questionnaire which we have used to assess the malnutrition among geriatric denture wearers. This is the generalized oral health assessment questionnaire which has been pre-validated in the local language which has been administered. With these questionnaire, our aim is to conduct a gender-based comparative evaluation among all the 4 parameters. So the objectives includes to correlate the gender frailty in the targeted population, to find the gender predilection, and to find if the gender predilection for oral frailty among the target population and for cognitive dysfunction as well. So this is the sample size calculation which came up to around 110 per sample size using the formula, and these were the 4 questionnaires which we have administered. So coming to statistical analysis, these are the statistical methods we have used, and the latest version of the application of G-power statistics has been used to analyze the statistical data. Coming to the results, so this is the apparently vulnerable population which we have come across, which shows that how, what happened? Yes. Is the slide visible, ma'am? Hello?
Yes, it's visible.
Okay. So in figure 1, it actually shows that around 32.7% are apparently vulnerable to more subjected to frailty, and moderate frailty mainly accounted to around 9.6% in our geriatric population, and severe frailty was around 1.9%. When scoring of oral frailty was conducted, a significant proportion of population is potentially susceptible to developing of frailty, which can have profound implications for their oral health, quality of life. When coming to scoring criteria of the cognitive assessment, around 88.5% exhibit no cognitive impairment. However, it also highlights the presence of varying degrees of impairment within the remaining proportion of population. There was mild cognitive impairment in 7.7% of individuals. When the gender predilection was assessed, the table here mainly shows that around 17 males and 12 females are classified as not being non-frail, while 17 males and 6 females are classified in the verge of developing the frailty, but a chi-square stent found no significant association. When tabulated regarding the association between oral frailty and gender frailty scale, we got to know that around 3.8% were individuals were under non-frail conditions, while in the frail group, there are 9.6% of individuals highly susceptible for frailty. An association between oral frailty and gender was also conducted. The chi-square test has been conducted to associate between the oral frailty and gender, and we got to know the negative correlation between both. Association between frailty scale and gender, a chi-square test has been conducted to examine the association between the frailty scores and gender, and the results indicate a not significant. Yes.
Please summarize your study within the next
Sure. Yeah, sure. So 49% are classified, individuals are classified at risk of developing malnutrition. So the statistical analysis demonstrated a highly significant association between the GOHI scores and MNA, and when correlation was done between age and oral frailty, and MNA and oral frailty, we got to know that there was a positive correlation between general frailty and a negligible relation with the variables such as age, oral frailty, and MNA. So there are limited existing data on the oral frailty in frail individuals. There are all previous studies have demonstrated, but there are numerous barriers to provide good oral care, including availability of dentists, resistance to care, transportation, financial difficulties, and awareness of both patients and caregivers. So to conclude, our study also supports the previous study who reported strong association between GOHI and MNA scores, and hence promoting dental health in geriatric inpatients by early screening and referring and integrating dental services with special attention to dietary management. Close monitoring of low salivary flow and xerostomia may improve both oral health and frailty. So these are my references. A humble thanks for the organizing committee members of the Allied Conference and my parent institution. Thank you so much.
Thank you, ma'am. Before we move ahead with the questions, I would like to invite Dr. Priyadarshini ma'am Professor and Head Department of Microbiology, Chettinad Academy of Research and Education. She's our session chair for this session. I would
Thank
request
you.
session chair to please go ahead with the questions.
Thank you. I think I will start from the next presentation because this presentation I was only halfway through. So please ask the
Okay,
previous people to do the questioning. I will start from the next one. Okay. Thank you.
Okay, ma'am. Thank you. So I assume there are no more questions.
Thank
We'll
you.
go
Thank you
ahead
so
with
much,
our
ma'am.
next. Thank you. We'll go ahead with our next participant, Dr. V. Anita. If you are present in the room, please respond.
Can you hear my voice, ma'am?
Yes,
Hello?
yes, you're audible.
Yeah, I
So
am
the
the
next
co-author of this
presentation is
paper.
by Dr. V. Anita on probiotics in Siddha system of medicine, a systematic review.
Okay.
Please go ahead with your presentation. You have
Yeah,
10
sure.
minutes.
Dr. Anita?
Yeah. So I'm co-author of Dr. Anita's presentation, myself R. Tyagarajan.
Dr. Anita isn't presenting?
Yeah, no, ma'am, no. I am the co-author of this paper.
Okay.
Is my screen visible?
See, actually, just before starting this session, I want to tell two things. So first of all, please stick to the time because we have so many presentations. I want you to just give the title of your topic and then maybe a few points on introduction and directly go on to the materials and methods, results and discussion. Okay. And any other questions, we will ask you in those 2 to 3 minutes. Introduction, just give me an outline of the reason for why you chose this study. Okay. No need to give too much of an introduction about the thing that you're doing. Okay.
Yeah, sure.
Thank you.
Okay. So myself Tyagarajan from Sri Sairam Siddha Medical College and Research Center, and my topic is probiotics in Siddha system of medicine, systemic review. So, as Siddha system of medicine contains numerous preparations which are probiotic in nature, which promotes our gut health and overall well-being, and many preparation mentioned in Siddha pharmaceuticals, which are fermentation products and alcoholic in nature. These preparations have therapeutic effect as well as nutritional value. So, aim and objective. The objective of this review is to examine the traditional knowledge and the scientific evidence supporting the utilization of probiotics in the system of medicine and their potential therapeutic applications. So, methodology, a comprehensive literature search was conducted using various Siddha literatures, textbooks, and databases such as Padatha Sindhamani, Noyananga Vidhi, and Gumapadam Tadu, and various databases such as PubMed, Embase, Medline, Scopus, etc. So, dietary preparations and their therapeutics in Siddha system of medicine, which promotes gut health. So, milk and its byproducts, it is compared with the 5 elements. Butter is compared with land, and buttermilk with water, ghee with fire, curd with ether, and milk with air. In Siddha system of medicine, these dairy products used in purification of raw drugs, preparation of medicines like Tayir Chungi Churnam and adjuvant for various medicines, which increases the efficacy and maintains the gut health. So first one, milk, Pal Umbum. So it is the reference taken from Padatha Sindhamani. So dietary foods such as milk can protect prebiotic bacteria from high acid levels in the stomach, allowing higher levels of probiotics to reach the intestine. Next curd, Muta Tayir Umbum, which is also taken from Padatha Sindhamani. There are various curd mentioned in our literature which cures various diseases such as urethritis and SKD in curd, and buffalo curd is used for treating pitta diseases, and camel's curd is used for worm infestations. So the wormwood curd that we are using contains a spectrum of lactic acid bacteria that have and potential to exhibit probiotic effects in individual consuming the curd. Next, buttermilk, which is also mentioned in Padatapna Sindhamani as more perky. So we should use buttermilk as mixed with water, which can use in treating various diseases such as ascites, abdominal pain, and embu murivu, which is known as TB. So in our literature, buffalo's buttermilk is expressed as the Manigarka Kigaita Amridam, which means it is one of the Amrita which can people consume to maintain their healthy life. And next one, ghee, which is mentioned as neyurki. So we should milk the ghee and we should use for our daily practices, which contain lot of free fatty acids, phospholipids, and fat-soluble vitamins, which maintains the brain health and gut functions. And last one is fermented rice water, which is also known as angakari. So karunguruvai, which is a type of rice, is taken and it is cooked and it is mixed with water and it is transferred to earthen pot and it is covered using a cotton cloth and it is kept under sunlight for 6 months and daily we should stir the earthen pot so that there is no contamination. This has to be kept for 6 months and the fermented water should be taken for various diseases such as giddiness, blocking and indigestion. So consuming this fermented rice water enriches the intestinal flora which benefits bacteria and enzymes. So now it is compared with as apple cigar vinegar which are using nowadays. And last one is cool, which is peculiarly used in Tamil Nadu, especially in rural areas in summer season. The traditional fermented millet which is widely consumed. Kool is made from a pearl or a finger millet flour, and it gives stamina over an extended period. So this food contains probiotic effect because the presence of oligosaccharides and non-starch polysaccharides. So discussion and results, the dietary and fermented preparations explained can be said to have the benefit of enriching the health with good bacteria. In the process of fermentation, the microorganism converts the chemical composition of raw materials. Fermentation not just calculates the taste of the product, but augments the therapeutic and nutritive value. A lot of research can be carried out in these areas of fermented products of Siddha pharmaceuticals. The indigenous microbes from traditional fermented food as natural biological resource for health beneficial can be worked upon. So acknowledgement, I thank the management of Sairam Institutions, Dr. S. Madhukumar and Dr. V. Anita for this presentation and I thank this opportunity for presenting in this conference. Thank you.
Thank you, sir. I would like to request session chair to please ask the questions.
Thank you, Anita and team. Actually, your presentation was very interesting. Actually, many of the facts are already known, however. Have you done any study using this or this is just a review kind of paper?
Yeah, it's just a review paper, ma'am. We are studying a new topic.
What are you
based on
working on?
ghee. Actually, in our literature, they told that we should melt the ghee every time we use, ma'am.
Okay.
So what is the difference between unmelted ghee and melted ghee? So that process we are undergoing now.
Okay.
So
Is there
what
any
changes
difference?
happens?
Is there
Yeah,
any
we are
difference?
we are not skilling process, ma'am.
Okay. What is the difference between prebiotic and post and probiotic?
Probiotics which controls the gut microorganisms and prevent the acid level of the intestine.
What is prebiotic then?
Prebiotic is sorry, ma'am, I can't take it.
Pardon?
I'll refer, ma'am. Sure.
Yeah. See, actually, prebiotics or what you're telling, no, all your foods, they do not contain bacteria. Okay. So they are actually the foods which help your bacteria to grow. So these are only called as prebiotics. Probiotic is actually a gut healthy bacteria. Okay. I want all the others to kindly mute yourselves. I don't want any unnecessary interference when the person is presenting. Kindly mute yourselves, the other people. Okay. So this is the difference between pre and probiotic. Okay.
Okay.
Yeah. Thank you for your presentation. We can go on to the next person.
Thank you. Thank you.
So the next presentation is Miss Pinky Gupta.
Yeah, am I audible?
Yes, you are audible.
Okay.
I'll share my screen. So the next presentation is by Ms. Pinky Gupta on antimicrobial potential of Blumia lacera against disease-causing clinical bacterial pathogens.
Okay.
And we have 10 minutes. Please start your presentation.
Yeah, one more thing. The person who is next on the list, kindly be ready. We won't be calling out your names continuously. The next person, be ready. Thank you.
My screen is visible.
Not yet.
Okay.
The screen is not visible. Yeah, now it's visible. You can go to slideshow. Pinky Gupta, I'm not able to hear you. Please check your net connection.
Ms. Pinky Gupta, please unmute yourself and start with your presentation. If you are
Now
having connectivity issues, you can resolve it and come next.
Okay.
So the next participant is Ruben Thomas. If you are present, please respond. Ruben Thomas, if you are present in the room, please respond. Dr. Shweta Muni. Dr. Shweta Muni, if you are present in the room, please respond. Mr. Tanveerul Saqib.
Actually,
Yeah,
Dr.
hello,
Shweta is still
hello,
logged
ma'am.
in. Dr. Shweta is logged in. I don't know why she's not presenting.
Hello, ma'am. Can I present it?
Yeah,
No.
is it Dr.
Yes.
Shweta?
Ms. Pinky Gupta, if you are ready with your presentation and your network is stable, you can start.
Yeah, okay. Now it's
Mr.
audible.
Tanveerul Saqib, you are the next presenter.
It's audible, ma'am. Is it audible, ma'am?
Yes, you are audible. You can start your slideshow. You have 10 minutes to present.
Okay, ma'am. Okay. So, the overview of Blumia lacera, it's a natural plant or it's a medicinal plant which is increasing worldwide, particularly in different countries like India, China, and other Middle East countries. Like in India, approximately 80% of the people rely on traditional plant medicines for their primary healthcare needs. So, medicinal plants have been utilized since ancient times for ailments caused by microorganisms with a wide range of possessing various pharmacological activities. So, in recent interest, the plant-derived drugs reflects recognition of traditional claims regarding natural product values in healthcare. So, phytochemical investigation are crucial for the quality control of the traditional medicines. So, my research work on to find out the plant-derived drugs in Blumia lacera and Tinospora cordifolia. Actually, I'm working in two plants, which is Blumia lacera and Tinospora cordifolia, and I am finding out the noble cause of to kill the resistant microbes, which is available all near the healthcare institutions or hospital. So overview of Blumia lacera, it's like over 7,500 plant species are reported to be used in Indian traditional medicinal systems, including ethnomedicine. So many medicinal plants globally in India have been screened for antimicrobial activities, leading to drug formulations. So medicinal plants are extensively used in medicine against drug-resistant bacteria, addressing one of the significant challenges in infection diseases treatment. So what my work was on, like I have taken the plant powder form of that Blumea and I have finding out the antimicrobial agents. So basically, what it is also called as Kukrondha, growing wildly in wasteland, roadside areas, drying ponds along drains, and river margins, and it is also best anti-inflammatory, septic, ophthalmic, digestive, anti-healthalmic, liver tonic, expectorant, fibrous, antipyretic, diuretic, desorbent, stimulant, and antioxidant. And it has a very significance against screening of the clinical pathogens. What I have done, my research work, I have tested the antimicrobial activity, which is also reported in different research papers like bacteria and fungus. And I have taken the 8 strains of specific microbial species, which cause the hospital acquired infections, which is the nosocomial infections, and that will be the Staphylococcus aureus, Pseudomonas aeruginosa, Escherichia coli, Klebsiella pneumonia, Salmonella typhi, Shigella soli, Acinetobacter baumannii, Proteus mirabilis. These are the 8 strains I have taken. So whole plant I isolated using different extraction method. So tested all the 8 bacterial clinical pathogens isolated from the hospital patient sample. So the brief overview of the antibiotic resistant crisis is, we already know in the year of 2050, like everybody will be in the pandemic state. So the antibiotics, which is miracle drugs, saving countless lives since their discovery, overuse and misuse, leading to the emergence of antibiotic resistance. So what is antibiotic resistance? Natural process where bacteria evolve to withstand antibiotic effects, renders antibiotics ineffective, making infections difficult or impossible to treat. So causes of antibiotic resistance, which is over prescription and inappropriate use of antibiotics, agricultural use in livestock, poor infection control practices, and global spread of the resistant bacteria. So impact of antibiotic resistance is increased morbidity and mortality rates, longer hospital stays and higher healthcare cost, compromised ability to treat common infections. Solutions to combat like antibiotic resistance is the rational use of the antibiotics, like prescribing only when necessary, and developing new antibiotics and alternative therapies, and enhanced infection prevention and control measures. And what are the conclusion we are getting? Like antibiotic resistance is a critical global health threat which is happening now in whole world, and urgent action and collaboration and fund agencies are needed. Now this is the antibiotic resistant mechanism, how it is happening. It is happening around all the globe, and it is basically happening with the resistant genes, which is already which is already present in the bacteria as well as in the plant, and as well as whatever we are taking, whatever we are eating, we are going through, we are like all around near us, it all happening to us. So my main motto is to to do the plant extraction, and what I did was to like natural antimicrobial activity, that Blumea lacera is the natural antimicrobial activity, and has the traditional medicinal uses and phytochemical analysis, and we can do the addressing antibiotic resistance, uh, the Blumea lacera, and future research and development and conclusion. So Blumea lacera has demonstrated antimicrobial activity against a range of pathogen, including methicillin resistant and Pseudomonas, Escherichia coli, and others. And Blumea lacera presents an opportunity to explore alternative therapies against drug-resistant pathogens also, and it has shown the anti-inflammatory and digestive and antipyretic effects also, and to elucidate the mechanism of Blumella-Serra antimicrobial activity facilitated the translation of its therapeutic potential and into clinical applications. So my methodology was what I have taken the sample in the Gaya district, and I have done the extract preparation. I have isolated culture identification of the bacterial pathogens, all the 8 strains, and then I did the antibacterial AC, which is happening the antimicrobial activity and sensitivity and MIC. And the sample collection, I have taken the fresh leaves collected, and extraction, I have taken the 3 different concentration, which is the 300, 450, 500. Actually, I have taken the 100 also, but I was not getting the zone of inhibition properly against this bacterial infection. So I have increased the concentration. So what I have done was I have isolated culture and identification of bacterial clinical pathogens, and I have also studied the patients, like how many patients are coming, and I have done all this study in the Department of Microbiology, Anugrah and Magar Medical College in Hospital Gaya, Bihar, and I have strained the bacterial clinical pathogens from also the IGIMS, Indira Gandhi Institute of Medical Sciences, Patna. And these are the graph, like male 188 patient and female are having more. I have got more in females. And these are my previous research papers, which I have got doing the
Excuse me, ma'am.
yes, yes.
Please summarize your study within the next 2 minutes.
Okay, ma'am. So these are the hospital samples which I have taken, and I have segregated based on the collection area. So what and all I have done the biochemical test and all. So all these 8 species I have got. So what is the determination? So I have done the antibacterial assay. I have done the agar well diffusion method, which is Muller-Hinder agar plates. Then I have done the Newton broth incubation, test organism onto agar plates, clone and sterile spreader, additional of methanol, and aqueous extraction concentration, which is 3 different concentration to well. And I have taken the control well as a solvent. So waited for 24 hours, and measurement of zone of inhibition is seen. And after
Madam,
that,
can you just directly go to the results?
Yes, ma'am. Yes, ma'am. So what I have got, I have got the maximum is in Pseudomonas aeruginosa and Salmonella typhi, which have shown the 53.5 the zone of inhibition, and the rest of in distilled water, I have seen the complete inhibition in most of the bacterial pathogens. Like this is the Salmonella typhi plate, and these are the 3 different concentration, and I have seen like in 500 microgram per ml, it has shown the most ZOI, zone of inhibition area. So Blumella-Serra, like I have extracted in Gaya district, and Blumella-Serra I have got the highest activity of 53.5 mm based on the middle highest
Ma'am, your
concentration.
time is up.
Okay. This is the graph which I have seen, and the highest is the Salmonella typhi against the Blumella-Serra extraction. So this is the conclusion. So in the present study, the result confirmed that the plant herb Blumella-Serra have significant potential to counter the growth of bacterial clinical pathogens in hospital under tested laboratory conditions. So further research on identification of the particular antimicrobial resistance and identification is needed. These are my acknowledgement I have done under the Dr. Sanjay Naksar, assistant.
Excuse me, ma'am, your time is up.
These are my
I would
references.
request the session chair to please go ahead with the questions.
Thank you.
Yeah, what are the fungi you tested against?
Sorry, ma'am.
What are the fungi you tested against, madam?
Ma'am, I haven't tested any fungi, ma'am. I have tested only 8 bacterial pathogens, ma'am.
Yeah, but in your introduction and materials, I think you have mentioned it has
no, ma'am. I have.
Just check your presentation, okay?
Yes, ma'am.
And what is the common name of this thing? Blue meal?
Ma'am, it is Kukronda, ma'am.
Pardon?
Kukronda, ma'am. Kukronda.
In English?
No, ma'am, in Hindi.
In Hindi. What is the name in English?
In English, ma'am, it's a ma'am. It's a garlic, ma'am. It's a
Okay, leave
not
it.
that garlic what we are taking, that different garlic, ma'am.
Okay. Thank you. I think we can go on to the next person.
Thank you, ma'am. Thank you. Thank you.
Thank you. Mr. Tanveerul Saqib, are you ready with your presentation?
Yes, ma'am. Yes, ma'am.
Okay, so the next presentation is on passion as the correlate of psychological well-being among prose writers by Tanveerul Saqib.
Yes, ma'am. Good afternoon, everybody. Hello.
Yes, you're audible. Kindly
Am I
start.
audible, ma'am? Am I audible?
Yes, you're audible.
Okay, ma'am. Just a second, please. So my topic is passion as the correlate of psychological well-being among prose writers. Am I audible, ma'am?
Yes, madam. Please proceed.
Yes, ma'am. So basically, this is a psychological topic dealing with the prose writers, their psychological well-being and their passion. So starting with the
Do you have any
definition.
presentation? Do you have any presentation slide deck or anything?
Yes, ma'am. Yes, ma'am.
Yeah,
I
can
do
you share
have.
it then?
Yes, ma'am.
We're not able to see anything.
Okay, ma'am. Just a second. Is it visible now, ma'am?
No. In case you're already sharing your screen, stop sharing and then start sharing again.
Okay, ma'am. Just a second. Is it visible now, ma'am?
Yes, madam, it's visible.
So starting with the definition of prose writers, these are one of the forms of artists who are involved with the literary expression of thoughts related to any field of human knowledge like religion, politics, science. In other words, we can say that these are those people who are having some peer and public recognition and who contribute their abilities and potentials towards the field of art and culture for their development. So in Kashmir, there are about 300 recognized and registered prose writers who use their literary work as the expression of their thoughts in different languages. That is English, Urdu, Kashmiri. This artwork has various benefits in an individual's life, like it brings insight, coherence, stability, develops creative powers, increases longevity, brings consciousness and mindfulness. So the reason behind this for the conducting of this research is basically we have notion that these artists, these writers, they are great from the clinical point of view. Hello. Hello.
Others, kindly mute yourselves. Participants, others, please mute yourselves. Madam, you continue.
Yes, ma'am. So the notion is that we have a common notion regarding these artists, or we can say regarding these writers, is that they are having the clinical symptoms. They are bipolar. They are having manic depressive illnesses. They have impersonality. They are impulsive. They are antisocial. It means that we weigh them from the clinical point of view. And on the big five personality theory in psychology, we view them as neurotic, and we say that they are having the clinical aspects mainly. So the reason for doing this research is to bridge the gap, which is basically that I conducted a study from the positive point of view. Keeping this clinical point in consideration, I studied the prose writers from the positive point of view by studying their passion and psychological well-being, which are the positive psychological constructs. So definition of this passion is that it is basically a positive non-cognitive psychological construct, which can be defined as a strong internalized emotional desire or feeling towards a personally important value preference that motivates intentions and behavior to express that value or preference. So the dualistic model divides this passion into two components. One is harmonious passion and the second is obsessive passion. Harmonious passion, if we have to define it, it is basically an autonomous internalization of the activity, harmonious relationship with the other activities, enjoyment in the activity, full integration of behavior, and the activity remains under the control of a person. Obsessive passion, on the other hand, is related to one's partial behavioral integration, controlled internalization of the activity, no control over the activity, disharmony with the other activities in life. The other variable in the study is the psychological well-being, which is a dependent variable in the study. So defining this psychological variable, it refers to a state which encompasses the higher aspects of life like love, purposefulness, happiness, success, actualization, fully functioning of an individual, and the realization of one's potential. This psychological well-being includes autonomy, purposefulness, personal growth, environmental mastery, positive relations with others. This psychological well-being is divided into two perspectives. One is hedonic perspective and the other is eudaimonic perspective. The former is linked with the one's subjective well-being, means one's positive affect, negative affect, and satisfaction with one's own life. And the later is related with the broader perspectives, that is with those aspects which go beyond experiences and the subjective well-being, means it is the broader aspect of life which deals with the higher qualities of one's life. Model of psychological well-being used here is eudaimonic identity model. The psychological well-being model used in the theory is eudaimonic
Excuse me,
identity
ma'am.
model, which is,
Ma'am, please
yes,
summarize
ma'am.
your study within the next 2 minutes.
Okay, ma'am. So the rationale behind this theory is that it gives us comprehensive understanding about these artists. Since the research is scarce, and I wanted to add some research to this kind of population. And the second thing is that going beyond the subjective well-being, this study will help an individual to enable them to go beyond the immediate pleasures and pursuits and experience the higher aspects of life. Social policy making, the eudaimonic well-being allows one to practice aspects of life from the social perspective or for the societal good, means the purpose of human life is more than the individual utility and provides a framework for social policy making. So the methodology used here was conducted on the the study was conducted on the registered artists from the Kashmir Valley located in Lal Mandi, Srinagar. Number of sample 100 writers, majors here, used here, psychological well-being scale by Waterman, which includes 21 items, passion scale by Valorant, it includes 14 items. So purpose of sampling technique was used here. It is basically a quantitative study. Correlational analysis was used for measuring the relationship between these two variables. Inclusion criteria was only the registered writers. Age range was 15 to 80, and these artists should have some recognition in their writing work. So the data analysis and interpretation, these are the two variables, and a harmonious this passion is having subdivision of further two variables. One is psychological well-being and the other is harmonious passion and the obsessive passion. So the study here shows that the harmonious passion is positively correlated with the psychological well-being, and the obsessive passion is negatively significant, moderate negative correlation between these two variables. And the relationship between harmonious passion and the positive and psychological well-being is 0. Or is equal to 0.647, and negative correlation is minus 0.537 between those two variables. So the interpretation, the robustness of these correlation was substantiated by
Ma'am, your time is up.
Just a minute, ma'am. Discussion. The strong positive correlation suggests that individuals who engage in activities they love and find personally meaningful, means harmonious passion, they experience higher levels of psychological well-being, and the individuals who have who have obsessive passion, they are more susceptible to the depressive illnesses, and they experience lower levels of psychological well-being. Thank you, ma'am.
Thank you, ma'am. I would like to request session chair to please go ahead with the questions.
Yeah.
Yes, ma'am.
Actually, I have no questions. So, I just pass on to the next chairperson.
Okay, ma'am. Thank you.
Thank you, ma'am. Before we start with our next presentation, I would like to call the next 5 participants to please respond in the chat box. Dr. Sheetal Sharma, Ms. Polly, Cyril Thomas, Pallavi Yelarthi, and Dr. Hekta Doshi. If you are present, please respond to the chat box.
Yes, kindly be ready with your presentation. Yeah. So who's the person who's going to be presenting next?
Okay, so next we'll have Ms. Polly with the study of effectiveness of yoga therapy on menopausal challenges faced among postmenopausal women.
Yes, ma'am.
Okay, ma'am, you can continue with your presentation. You have 10 minutes.
Please stick to the time limit, okay? Thank you.
Good afternoon, ma'am. I'm Pauli Pipi. I'm
Good
presenting
afternoon.
Ma'am, you're not audible.
I'm Pauli Pipi, presenting the topics of effectiveness of yoga upon postmenopausal symptoms among women.
Please share your screen. The presentation is not visible. Ma'am, if you're having any connectivity issues, you can resolve it. We will move on to the next participant. If that's okay,
ma'am.
Okay, thank you. So next is Cyril Thomas. Miss Polly, please mute your mic. Next, we have Mr. Cyril Thomas presenting a comparative study of selected anatomical fractionation variables between the senior national level male kabaddi and kho-kho players of Manipur state. So you can start with your presentation. You have 10 minutes.
Am I audible?
Yes, you're audible.
Okay. Good afternoon, everyone. I got this. This is my topic: the comparative study of selected anatomical fractionation variables between the senior national level male kabaddi and kho-kho players of Manipur state. So the contents would henceforth be introduction, methodology, results, and discussion, and conclusion and acknowledgement. The introduction, it would be like the study of analysis, the anatomical features of national kabaddi and kho-kho team players of Manipur, India. The study focused on several variables such as adipose mass, that bone mass, fat mass, and the muscle mass and the residual mass in order to the better understanding the athletes' distinct physiological adaptation to their individual sports. The comparison of the data from both the sports may give an insight of distinct physical demand of Kabaddi and Kho-Kho, perhaps enhancing the training technique and injury prevention strategies for each and every activity. So, moving forward towards the deep into the topic a bit about anatomical factorization, what are they? It is a process of splitting the human body into smaller sections or fractions. It can also be said as anthropometric fractionation. That would be a more relevant word according to ISAC, that is International Society of Advancement in Kinanthropometry. So we are going to focus more upon these four components under which it comes. That is bone mass, muscle mass, fat mass, and the residual mass. See, the purpose of the study is to compare anatomical fractionation variables between Kabaddi and Kho-Kho players of Manipur senior state. We are hypothesizing that there will be a significant difference between Kabaddi and Kho-Kho players in terms of fat mass, bone mass, and muscular mass and residual mass. Significance of the study: comparing the fitness of the players and also his level among the team as well as with the other group. This study will help the Kho-Kho and the Kabaddi players specific physiological adaptations with respect to International Society of Advancement in Kinanthropometry, comparing the anatomical fractionation variables for different performance in the future. So the study is delimited to achieve the purpose of the study. Kabaddi and Kho-Kho players are these two groups are selected. The total population is 20 and the sample size would be 10 and 10. The ages are from 18 to 25 years. The study is restricted to International Society of Advancement in Kinanthropometry, comparing the anatomical fraction variable limitations. The hereditary and environmental factors of Kabaddi and Kho-Kho are not taken into the consideration. The research competency of the ISAC level 1, I'm an ISAC level 1 practitioner, so that would be a factor which is going to limit. Methodology. The purpose of the present study is to compare specific anatomical fractionation variables between senior national level male Kabaddi and Kho-Kho players from Manipur state. For the purpose of the study, population is 20 and the sample size is 10 and 10. Administration. The fractionation equipments which are being used are a slim guide, anthropometric sheet, Wolverine caliper, and weight machine and stadiometer. Hence, moving forward for the preparation is that we have to say to the people, the sample should have minimal clothes with them. And I'm audible, right?
Yes, you are pretty audible. You can continue with your presentation.
Yeah. So the preparation would be like we have to make the people a bit more comfortable with when we are going to take the anatomical those four components. So because we are going to have some of the skin fold sides, 8 skin fold sides, followed with the body mass and with the girth measurements, as well as one more component that we are going to say, and with the land markings and measurements. These are the things we are going to do henceforth. Adipose mass, it is related to all adipose mass, muscle mass, and bone mass and residual mass. These things are being taken from the ISAC Metry. ISAC Metry is again a software in which we have to put on with other components. And this is just an overview of how it was being collected and it was being done. So the basic measurement sides include the 4, that is body mass, a stress stature, sitting height, and arm span. Skin fold has 8 skin fold sides, that is biceps, triceps, subscapularis, iliac, crest, supraspinal, abdominal, and front thigh and medial calf. Girth measurements: arm relaxed girth, arm flexed tensed, waist girth, hip girth, thigh middle girth, and calf girth. Breath measurements: humerus breadth, bistellar breadth, and femur breadth. So the statistical technique that I used for this study was that is independent t-test, because we are going to compare the two groups with the level of significance of 0.05 confidence. So the results somehow came with the adipose mass of Kho players were 12.5 to that of if Kabaddi, if I were to measure that, they were having a more amount of adipose tissue layering out, that is 18.51. Bone mass of the Kho players were more considered to as that of Kabaddi players, 11.26 to 9.7 of that of Kabaddi players. Muscle mass was 3.3. 33.34 for the Kabaddi players and Kho players for 28.64. Residual mass is 9.51 for the Kho players, and Kabaddi players were having a more residual mass, that is 10.83. So the conclusion of the study brings out, though a meticulous comparative analysis of anatomical fractionation variable using isometry methodology, our research sheds light on the distinct physiological profiles of senior national level male Kabaddi and Kho players in Manipur state. Our finding reveals notable disparities in key role anatomical metrics, namely body mass, adipose mass, muscle mass, bone mass, and residual mass between the two athlete cohorts. Kabaddi players exhibit significantly high body mass, adipose mass, and muscle mass compared to that of Kho counterparts. These suggest that the physical demand of Kabaddi, characterized by intense burst of activity and physical contact, may necessitate the greater muscular development and overall body mass to excel in the sports rigors. Conversely, Kho players demonstrate slightly higher bone mass, indicative of potential adaptation to the unique biomechanical stressors inherited in Kho dynamic movement and strategies. The conclusion for henceforth, furthermore, the observed differences in residual mass elevate subtle variation in metabolic activity and body composition regulation between the two athlete groups, while Kho players displayed marginally lower residual mass compared to that of Kabaddi players. Further investigation into the metabolic and physiological underpins of these variation could offer valuable insights into the optimal training protocols and injury prevention strategies tailored to each sports. These are my reference. So thank you. If you can not measure it, you cannot improve it. That's said by Lord Covey.
Thank you for your presentation. Which institute are you from?
Ma'am, I'm from National Sports University, India.
And what is the program you're enrolled in?
Masters in Physical Education.
Okay. Good. Thank you.
Thank you.
Thank you, Mr. Cyril. Our next presentation is by Pallavi.
Ma'am, can I do it? I'm Pauli PP, ma'am.
Yes, ma'am, please. Mr. Cyril, kindly stop sharing your screen, please. Ms. Pauli, kindly share your screen and start presenting. Ms. Pauli, is there any issue regarding sharing the screen?
Yes, ma'am. Can you see, ma'am? Can you visible?
No, your screen is not visible yet.
Madam, why don't you send your screen by WhatsApp to the organizers? They can share your screen. They can share the screen and your presentation. Share your PPT by WhatsApp to one of the organizers. Okay. If you are not able to share your screen. Okay?
Ms. Pauli, you can share your PPT to Ms. Gopika. She will be sorting out the situation. So we will move to our next presentation.
Next I will do it, ma'am. Next I will do it. Okay.
By Ms. Pallavi Alarthi. on a topic of a report on exercise prescription adherence among community dwelling older adults and observational descriptive study.
Okay.
Ms. Pallavi, we can see your screen but not your PPT.
Yes.
Can you make it
Now,
quick, please? No, not yet.
not yet.
Yes. Once stop sharing your screen and reshare. I request all the participants to be ready with your PPTs on beforehand.
Can you see now?
No, just give me a moment. Try to share it now.
We are not able to see.
Now, can you see?
No, share it again.
Madam, you can stop sharing and then, come out of it, then again start screen share. Then it may work.
Yeah, now it's fine.
It's fine, no?
Yeah, you're on the last slide.
Yes. Yeah. Very good afternoon. And here I am here to present a report on exercise
Ma'am, sorry to
prescription.
interrupt you. Can you make it on a full screen mode, please?
Can you see now, full?
Ma'am, it's on. You can see your slides, but just put it in slideshow mode, no? Just click on slideshow. On the top, top, on the red bar is there, no, on top.
Yes, madam, but what is happening? It is getting stop shared.
Okay.
So because of that, I have put in this.
Okay, then you zoom it. Zoom your slide if possible.
Kindly tap on enable editing and then keep it in the full mode, ma'am, so that it won't be stopped. Now you can keep it in the full screen mode.
Yes.
Yes, thank you.
As such, adherence is the word where we can see behavioral correction and people are recommended to do the healthcare provider's advice. And we physiotherapists being, what happens? We give prescribed exercises. My sound is getting echo. Sound.
It's fine, ma'am. You can continue with your presentation.
Madam, these slides you can skip. Please skip this and go to the next slide, to the materials and methodology. Okay, need for study, let it be there. Let it be there. Just why you chose this study? Only one sentence.
It was because medically it is proven that people are ready to take medication, but regular exercises, they are some other way lazy or they feel very disheartened when they don't see the improvement. So just they leave the exercises. And exercise is also one of the kind of medicine which is going to prove their improved health status. Therefore, the study was chosen. And the percentage of exercise prescription adherence among community developed older adults are selected. That was my population, and then I wanted to know the percentage. Thus, the aim was to find out the percentage of adherence and to know the factors which are influencing the exercise adherence. And this was simple descriptive study with prescription as well as questionnaire method. 100 samples were collected within a span of 2 years. First, registration, OPD registration and 60 years and above candidates were collected, and their regularity was checked. Those who were regular were also contacted, and not regular also, they were also contacted. Therefore, the inclusion was OPD and home-based program in 65 years and above. Institutional-based rehabilitation subjects were not included. So, synopsis approval and rest of the screening procedure was completed. This table number 1 shows 60 to 65 years were 48, 65 to 69, 36, and 70 to 75 years, 16 patients were calculated. When it comes to gender, female were 47 and males 53. Here it is very important to say that 70% patients were regular as well as 30 were irregular patients. So directly our results suggest that 70% were adherent to exercise prescription. And they were referred by their family, orthopedician, physiotherapist, sometimes self and sometimes by surgeons. And few questions were there. Why they want to do exercises like strengthening my bones? They had accepted that, then able to walk properly without losing support. So there was no imbalance. So such questions were given to them, and very encouraging response was there for them. As they said, their fatigue levels are reduced, strong musculature, balance is active, and important measures. So therefore, adherence to therapeutic exercises is the key to successful safe aging. But accurate measurements are not available. Lack of well-developed measures of adherence are not available. Elderly population is less likely to continue home exercise program. Therefore, follow-up is very important aspect. And adherence to supervised exercise program in physiotherapy setup was as high as compared to home exercise program. Several factors like laziness, busy schedule, pain, forgetting to do exercises were big challenge for us to complete their home-based exercise program. So transportation was also one of the hurdle for our center. So therefore, facilitating the adherence in community is the key factor. These are my few references. Hope that this study will help the community governing individuals. Thank you.
Thank you, madam. You can go to the next person.
Okay, ma'am. Our next presentation is by Ms. Holly. I'm sharing her screen.
Good afternoon, ma'am, and every participants. I'm Pauli PP. I am presenting the topics on effectiveness of yoga therapy upon postmenopausal symptoms among women. Women of all ages can bring the changes and build a happier life when health team is ready to support them in every step of their way. And menopause is an important period in female life cycle. It is a time for in your life when menstruation ceases because of hormonal changes. And menopause is a natural biological process that most women usually face around the age of late 40s or early 50s. By 2026, it is estimated that the population in India will be 1.4 billion, in which people over 60 years 173 million, and the menopausal women 103 million. To improve the quality of life and to decrease the menopausal symptoms, a holistic approach in the form of lifestyle and behavior modifications are required. Women will undergo significant changes and may face physiological and psychological health problems. Hence, an attempt is taken to study the menopausal challenges faced among women between the age group of 40 to 60 years in selected rural area in Kerala. So I have taken many literature reviews regarding these topics. especially the cross-section study was conducted. So for the methodology, I have taken the research approach is qualitative research approach, and research design post-experimental research in that non-randomized control group design. The setting for the study, I have taken the Kudumbasri unit, that is the self groups, 14 wards in the Thanimukam Panchayat, Kerala. Accessible population, post-menopausal women in the Kudumbasri unit, the 14 wards of Thanimukam Panchayat. And the target population was post-menopausal women in the age group of 40 to 60 years in the Kudumbasri unit, the 14 wards of the Thanimukam village. The sample, 400 post-menopausal women in the rural community of Thanimukam village. And the sample technique was non-purposive convenience sampling. The method of data collection, after getting the consent from the respective authorities, participants were informed about the purpose and got the consent, and data was collected. The phases of data collection, informed and written consent were taken from the authorities. The study participants' baseline data of demographic variables, questionnaire, taboos associated with menopause, and the level of menopause symptoms were assessed before the intervention in both experimental and control group. And due to the corona, I have taken the video-assisted yoga intervention was administered to experimental group by the researcher after 2 weeks, followed by which the subject performed the yoga technique for a period of 6 weeks. After that, I did the post-test taboos associated with the questionnaire, level of menopause symptoms, level of satisfaction, well-being index were assessed after intervention and carrying the routine activities. So in the data collection, I have divided the groups like control group and experimental group. Both in that, I have taken the cases. And in the initial of plan study was, firstly, the permission I have taken from the Panchayat President and Medical Officer of CHC Thanimukam. And for that, the explanation of research purpose, duration, procedure introduction, everything has given. And the consent I was taken from 400 participants from the studies. The tools and techniques I was used, demographic performance of post-menopausal women, and the open-ended questions to analyze the taboos associated with menopause. And according to the Kupperman index, the menopausal rate is scaled to assess the level of menopausal symptoms, and the Ken-Person scale, that is was the satisfaction scale on regarding yoga therapy in post-menopausal women. And the final, the WHO well-being index was taken to assess the well-being of the post-menopausal woman. So the mean standard deviation and t-value, I have done the showing the effectiveness of pre-test and post-test menopausal symptoms among control group and experimental group of post-menopausal women. So it was significant compared to regarding the somatic, psychological, and urogenital symptoms. So it was significant in areas. So the intervention I have given regarding the yoga therapy, that is hand and leg stretches, Ardhagadi Chakrasana, Udasana, Padasana, Bhujangasana, Shalabhasana, like different types of intervention I have given regarding to stop this yoga, to continue the yoga. So as a result, I got it. The finding of the study was shows that there is a reduction in menopausal symptoms in experimental group of menopausal women, thus proving it to be effective when compared to the control group of menopausal women. And yoga as an alternative therapy, it's a safe, rejuvenating, free from all the side effects, and can be practiced in their own home settings. And women in the menopause shows a positive attitude towards performing the yoga. Then they were ready to practice this meditation, etc. Thank you. Thanks so much.
Thank you, madam. Thank you. It's an interesting presentation by you. Are you a trained yoga instructor?
Yes, ma'am.
Okay, so these classes you have only taken, all the yoga classes?
Yes, ma'am.
Okay, and then how did you assess their well-being after the therapy? And for how many days did you give the therapy?
More than 3 months, still I'm continuing. I used to give not only the video, otherwise also I used to go and attend because it is belong to our near to house.
So you go to the house and teach them, is it?
Pardon, ma'am?
No, you go to their house and teach them, or you tell them to come
Yes,
to a common place.
yeah, there is in every Kudumbasri, they have got one hall is there where we used to go and visit and do that every Saturday, very frequently. Okay, ma'am. And we'll
Once a
assess
week,
them
is it?
how far it is.
Once a week, is it?
Once a week, ma'am.
Okay, why did you specifically select some of the asanas that you have,
So
you have put up?
I'm a community health nurse. I used to visit the families. So I have heard many complaints regarding the postmenopausal symptoms like somatic, psychological, and physiological. So hearing that, I thought of doing this, ma'am.
Yeah, no, what I'm asking is, I know you have selected that for helping out these people. Why you have selected those specific asanas that you have mentioned? Any reason behind it? You have put up some 10 asanas, isn't it?
Mostly these people are not considered as after the menopause. They will be complaining.
No, I'm not asking about the people. In one of
Okay,
your
ma'am.
slides, you have shown, no, this asana, Vajjanga asana, something and all you were telling it, right?
So that only they can do the specifically every asana they cannot do, ma'am, according to their age and physiological problems.
It was selected based on the age, is it? All these asanas?
Ma'am, based on the age between the 45 to 60 years.
No, actually, it should be based on, don't you think it should be based on the reproductive health, the asanas which will help the reproductive health?
Okay, ma'am. Next studies I do, ma'am.
No, why don't you do that? Just check whether these will have some effect on the uterus, the mind, whether it has a relaxing effect.
Ma'am, in the somatic areas, it all comes, ma'am. Every organ comes.
Okay. That you should
Yeah.
mention. See, you are
Yeah,
told
ma'am.
these asanas, no? What is the purpose behind each one of these asanas?
Yes, ma'am.
That you should tell.
Okay, ma'am.
Anyway, thanks. Interesting. Next
Thank you,
person can present. Thank you.
Thank you so much, ma'am, caring.
We can have the next presenter, please.
Hello. Am I audible, ma'am?
Yes, you're audible.
Our next presentation is by Dr. Hita Doshi on a topic of comparative study, rapid impact of sucrosial inhibition versus MRF on hamstring tightness in healthy individuals, which was a oral presentation.
Hello. Hello.
Yes, ma'am, you're pretty audible.
Am I
Kindly
audible?
share your screen.
Okay. Good afternoon, everyone. Myself, Dr. Hita Doshi, and I just share my screen.
Yes, please.
Is it visible?
Yes, ma'am, it's visible.
Okay. Good afternoon, everyone. My study title, that is the comparative study, that is rapid effect of the suppository inhibition versus MFR on the hamstring tightness in healthy individuals. These are the contents of my study. Then after that, first of all, we will go about the introductory part, that is the muscular strength ratio to the quadriceps and hamstring muscle, which is important to the maintain full range of the motion while preventing damage, as well as decrease the muscle ability to deform causes muscle tightness. Sitting for the longer duration is the contributor factor for all the healthy individuals. For remember, that is the hamstring tightness. The tight hamstring tight, which is responsible because of that produce the postural problem, back pain, decrease the stride length, walking, running speed, and it play a significant role for the presence of plantar fasciitis also. There are the very high prevalence ratio, 82% among adult age, that is 18 to 25 year college going students. In this technique, we have the effect that is the immediate effect, suboccipital inhibition, and MFR. Suboccipital inhibition technique aim to relax the tension in the suboccipital muscle by decrease the myofascial restraint in suboccipital region. Suboccipital muscle inhibition technique, which include the steady pressure, that is to soften the tissue to effect the relaxation and normalize the reflex activity. MFR also that is used for the traction, and it is a prolonged stretching of the fascia that is produce the help the breakdown of the adhesion, but and it will be decrease the muscle pain as well as it will help for increase the flexibility. And the next thing that is it is also helpful decrease the range of motion also. These are the main purpose, that is the immediate effect of the compare the rapid effect of suboccipital muscle inhibition and myofascial release technique on hamstring tightness in healthy individuals. These are the main aims and the objectives of the current studies. These are the null hypothesis. There is the null hypothesis. There is the no significant difference, and the alternative hypothesis. There is a significant difference between both the technique. Now the study that is this is the comparative study. In this I include the healthy individuals, people, those who have a bilateral tightness, hamstring tightness, as per my inclusion criteria, and the sampling technique that is selective sampling technique that is who fits inclusion criteria, and it is randomization done by the computer generated software. Total I have including 36 sample, and both that are the group A suboccipital inhibition technique and group B MFR both have the equal participant number 18 and 18. Then study duration that is the 6 month, and I include the college going students. These are the inclusion criteria. Those who have the willingly participate in this study, male and female both have included. Those who have the 18 to 25-year age, and those who have a bilateral hamstring tightness, those who have active knee extension test, more than 15-degree loss of knee extension as measured with the thigh held at the 90 degree of the hip flexion. I exclude in this study those who have a previous lower limb injury or pain from the past 1 year. Those who have the history of fracture or the surgery of the back, previous pelvis and the hip knee joint. Inflammatory condition, that those who have the affection of the joint range of motion, I exclude that. Those who have not willingly participated and those who have not involved throughout the taking the intervention, I exclude. Spinal cord injury and those who have the problem for the cardiac injury and any surgical patient, I exclude that. These are the study flow chart. For the duration of the study, within a 6 month, I assessed a total 57 participants. As per my inclusion criteria, 36 participants, those are the bilateral hamstring tightness, and they meet all the inclusion criteria. 21 excluded as per they are not meet the inclusion criteria. The randomization in the two group by computer generated software, and both are equally allocated in the two group, that is group A received suboccipital inhibition and group B MFR technique. Before they are received the intervention, active knee extension test priorly measured. After that, they immediately receive the intervention as per their group and as per their criteria. After that, immediately active knee extension test will be measured, and after that the analysis will be done. That all the outcome measure, that is the active knee extension test, patient lying in a supine position and they are holding their thigh by grasping their thigh 90 degree, and after that ask our patient that is do the knee extension and just measure the knee extension range of motion. That is the active knee extension test. Group A suboccipital inhibition, in that the patient lie in a supine position and suboccipital inhibition technique that is given on the suboccipital muscle, and it is hold for the 2 minutes for tissue relaxation. And the group B, those are myofascial release technique, patient lie in a prone position and it is applied over the hamstring muscle, that is the back of the thigh, and by therapist give the MFR by ulnar aspect of the hand, and it is hold for the 30 Second, after that, we are giving immediately measure the activity extension test, and we have done the analysis. That is, all the data will be passed with the normality test, p-value more than 0.05. That is, Gaussian distribution and statistical analysis were calculated by using alpha level of 0.05. Normality test was done by the Sapir-Whorf test, and the descriptive analysis was used to analyze the baseline data. PAT-T test, that is used for the significant difference of within a group pre and post data measurement, and independent t-tests are used for comparison of both the groups for all the parameters. I have first of all used for the pair t-test within a group and pair t-test that is used for the between a group. I measure right and left, both the side we are measured, as well as these are the baseline data. After that, we are measured the first of all pre and post data of right and left side of both this group A and group B, and it is highly significant as the p-value is 0.000, and that is highly significant. After that, this is the between group measurement, and in that is the p-value less than 0.05, and it is also sees that it is a highly significant data. In the present study, we are including 36 participants and those who have received suboccipital muscle inhibition and MFR. As per the previous studies, those who have done the MFR, they also shows that the MFR is widely effective clinically. And it is also shows that the MFR is helpful reduce the delay onset muscle soreness, that is decrease the pain also, and it also modulate the autonomic nervous system, which is helpful increase the flexibility as the kinetic chain. And it is also improve the throughout the intramuscular connection tissue pathway. In the present study also we have done the MFR, and it also shows as immediately it is also improve the range of motion, and it is also decrease the muscle activation of the force. So MFR was applied on the hamstring muscle, and it is more effective clinically. After that, we are also shows that the suboccipital muscle inhibition, that is the previous article we are also referring, as well as in this we also shows that in the immediately effectiveness, it is also improve the range of motion also as a superficial backline, and it is connected to the myofascial posterior kinetic chain, and it improve the range of motion also. After that, this study is also shows that it is also so too much effective for the hamstring tightness, and suboccipital muscle inhibition also proves the effectiveness in the study also, many mechanism behind it. Suboccipital muscle inhibition, that is work for the proprioceptors, and myofascial connector, that is also work as a myofascial technique also. That the major conclusion of the recent study I shows that I found that the myofascial release technique as well as suboccipital muscle inhibition technique both are equally effective, but clinically when it is for the long term, it maintain it by the MFR, and so we have rejected the null hypothesis, that is there is a significant difference between the immediate effect of suboccipital muscle inhibition technique and the MFR. These are the limitations and further recommendation of this study. Thank you so much.
Thank you, madam. Can go to the next presentation.
Okay, ma'am. Our next presentation is by Ms. Salma Musajai on a topic of effects of physiotherapy in cervicogenic headache and conquering thermomandibular dysfunction, a case study. Ms. Salma, are you there?
Yes, ma'am. Yes. Start with.
Yes, please.
Is the screen visible?
Yes, please go ahead.
Yes,
So, good afternoon, respected chair sessions and everyone present here. So, my title of the study is Effects of Physiotherapy in Cervicogenic Headache and Conquering Temporomandibular Dysfunction. It is a case study. Introduction. cervicogenic headache is a headache arising from musculoskeletal disorders of the upper cervical spine and is a form of chronic recurrent headache. The major criteria for CGH according to the Cervicogenic Headache International Study Group of the International Headache Society's classification are shown in the table 1. So basically, this is the diagnostic criteria for cervicogenic headache. These are the major criteria. One is symptoms and signs of neck involvement. Second is precipitation of head pain, similar to the usually occurring one, by neck movement and or sustained awkward head positioning, or by external pressure over the upper cervical or occipital region on the symptomatic side. Restriction of the range of motion in the neck, ipsilateral neck, shoulder or arm pain of a vague non-radicular nature or occasionally arm pain of a radicular nature, confirmatory evidence by diagnostic anesthetic blockade, unilaterality of the head pain. And the head pain characteristics are moderate, severe, non-throbbing, non-lacinating, usually starting in the neck, episodes of varying duration or fluctuating continuous pain. Next is temporomandibular disorders. It can be defined as a dysfunctional pain phenomenon characterized by temporomandibular joint pain and cranial fascial cervical, preauricular, postauricular, mandibular, temporal, frontal and or zygomatic arch area pain. Temporomandibular dysfunction may be divided into joint-related problems caused by the internal derangement of the articular disc and or intra-articular pathology, or the second one could be the muscle or myofascial disorders with trigger points in the muscles of the mastication. Patients may present with one type or a combination of both. So basically, this is a case study of a person who was present with cervicogenic headache and concurring temporomandibular dysfunction. So how cervicogenic headache we differentiate from a normal headache is that in cervicogenic headache, frequently there is only a unilateral head involvement, and it starts from the neck and goes up to the head, to the occipital region, and it can also spread up to the jaw areas. The pain is kind of deep, non-throbbing, and it is constant in character. There are intermittent attacks. It is not very usual or predicted attacks. It is intermittent attacks. And sometimes it is provoked by neck movements or sustained abnormal or you can say poor neck postures. The pain is usually non-radicular, but in rare cases it can also radiate till the arm. And reproduction of headache on palpation of tender areas in cervical or occipital area. So this is how we differentiate cervicogenic headache from normal or other headaches like tension headaches or cluster headaches. Then we have temporomandibular joint disorders. So basically, we have two types of temporomandibular joint disorders. One is because of the joint itself, and second is because of the muscles around the temporomandibular joints. Next, the need of the study. The study shows that the prevalence of cervicogenic headache is increasing every year, and there are very few studies done for the conservative treatment and traditional physical therapy approaches for the treatment of this condition. Therefore, our study can determine whether the physiotherapy approaches used in this study will be beneficial in the treatment of cervicogenic headache with temporomandibular dysfunction. The hypothesis of the study. First, we have null hypothesis that there are no significant effects of physiotherapy in cervicogenic headache. There are no significant effects of physiotherapy in concurring temporomandibular dysfunction. There are no significant effects of physiotherapy in cervicogenic headache and concurring temporomandibular dysfunction. And second, we have alternative hypothesis that there are significant effects of physiotherapy in cervicogenic headache. There are significant effects of physiotherapy in concurring temporomandibular dysfunction, and there are significant effects of physiotherapy in cervicogenic headache and concurring temporomandibular dysfunction both. So the main aim of this case report was to describe that whether the physiotherapy interventions in a patient who is diagnosed with cervicogenic headache and temporomandibular dysfunction, whether the treatment, physiotherapy treatment is effective in such patients or not. The objectives of the study to describe the effects of physiotherapy intervention in a patient with cervicogenic headache, in a patient with concurrent temporomandibular dysfunction, and in a patient who has both of the above conditions present, that is cervicogenic headache and concurrent temporomandibular dysfunction. So methodology is that the patient had arrived at the OPD, and he was a 31-year-old male who reported a 6 month history of headaches and cervical pain. He also had jaw pain with recurrent history over the past 2 years. And since past 2 years, the pain was not much, but it worsened since the headaches and cervical pain had started. So we assessed the patient's pain on 3 levels. The first P1 was the cervical pain, P2 was headache, and P3 was the jaw pain. So the cervical pain, which is P1, and the headache, which was P2, was aggravated by prolonged postures, especially at work, like driving or due to stress, and it was eased by heat, lying down, stretching exercises, and the analgesics which were prescribed to him. And for the jaw pain, the patient did not have a specific reason, or he did not mention any specific reason for the aggravation of P3, which is jaw pain, but he did relate it to stress, like where he was mostly stressed. That time his pain in the jaw had elevated, and he complained of having restless nights and grinding his teeth due to pain. So when the objective examination of the patient was done, the patient was presented with a forward head posture, bilateral scapular winging, and rounded shoulder. Even at rest, the patient had neck pain when he was performing shoulder lateral flexion to the right side. And other cervical physiological examination revealed minimal limitation. The range of the cervical was limited to a very small range. It was not much. And when the temporomandibular joint was examined, there was limited opening. There was a deviation to the right side. An audible click sound was heard at the left TMJ, and there was bilateral TMJ pain. A lateral excursion of the TMJ to the left was limited, and the movement did not reproduce headache symptoms from the temporomandibular joint. So this was what we had assessed on objective examination, and other assessment was also that there was at C1 there was painful bilateral transverse movements on the both the sides. So the physical therapy treatment which we provided to the patient was the cervical joint demonstrating dysfunction, which is the O, C1, C2, and C3, and from C6 to T1. And the temporomandibular joint were treated with passive joint mobilization techniques uh, uh, according to the Maitland's approach and the Maitland's grading of manipulation. So these are basically the mobilization and manipulation treatment. So at the O1, we gave posterior anterior mobilization at different grades. At 1+ grades, there were 10 repetitions and 3 sets, which were progressed to 30 repetitions of 3 sets. And then grade 2+, there was 60 repetitions and 2 sets. And 4- grades, we had 60 repetitions and 2 sets, which were progressed to 60 repetitions and 4 sets. Then at C1, we gave transverse mobilization. At C2, we gave central and unilateral mobilization with medial inclination according to the Maitland's grading. At C3, we gave central mobilization. And C4 to T1, we also gave central mobilization at different grades. And at temporomandibular joint, we gave posterior anterior mobilization of the right TMJ and posterior anterior mobilization of the left TMJ. Since there was not in objective findings, we could not find more of the right side. We gave the mobilization of the right side at lower levels compared to the left side. So these were what we gave to the cervical area. And the cervical paraspinal muscles, which are the trapezius, levator scapulae muscles, the muscles of mastication, were treated with trigger point therapy and stretching. Postural correction was also addressed because when the patient had poor posture, he complained that the pain was aggravated and it was radiating to the jaw and neck. He was advised regarding his posture and at work and in the car. To correct the patient's posture at rest, the scapulothoracic muscles, which are the serratus anterior posterior and the trapezius muscles, were strengthened. He was given a home exercise program which included posture correction, strengthening exercises to improve the scapular position, and deep cervical flexor muscle endurance, as well as stretches for the cervical and upper thoracic muscles. The patient was treated we gave 9 physiotherapy sessions, and then after that he was discharged with and home program was taught to him. This is the Maitland's grading, mobilization, and the treatment which we gave to him. So the results was that after 9 sessions, we had assessed mainly the pain, the range of motion, oral function, and patient satisfaction. So in assessment, we also included active cervical range of motion and active temporomandibular range of motion. We also assessed passive accessory intervertebral joint mobilization of the cervical spine according to the Maitland's grading. We assessed headache intensity, periodicity, and the headache's response to analgesia. And temporomandibular joint clicking and discomfort was also assessed, and the subject score was taken on the basis of a numerical pain rating scale. The subject regained full cervical and temporomandibular range of movement. On palpation, had reduced tenderness of the cervical spine, achieved improved scapular setting and deep cervical flexor muscles endurance, and recorded reduced scores on the numerical pain rating scale. The temporomandibular joint opening was full range with no click, audible, or pain felt by the patient. Mild stiffness on the left temporomandibular joint was still occasionally felt. The overall improvement per treatment was viewed as satisfactory, and the overall outcome was deemed as an excellent result. The patient was followed up telephonically 3 months post discharge, and the patient reported that subjectively the improvement gained from the physiotherapy intervention had been maintained. So, the aim of our study was that our study was successful in treating the cervicogenic headache with temporomandibular joint dysfunction, which was recurring. So, this was the result, which were pretty satisfactory. So this is the discussion that the aim of this case study was to describe the use of physiotherapy intervention in a patient diagnosed with cervicogenic headache and temporomandibular dysfunction, which is a common presentation in patients seeking treatment. The subject in this case report presented with a 2-year history of temporomandibular dysfunction and more recent 6 months history of a cervicogenic headache, which was related to increased stress and prolonged postures. A strong correlation exists between the cervical dysfunction and the temporomandibular signs and symptoms. The conclusion of the study is that this case report discusses the benefits of a multimodal physical therapy treatment approach for the treatment of cervicogenic headache and temporomandibular dysfunction, which is a common clinical presentation. The combination of joint mobilization, trigger point therapy, strengthening, and postural correction may be considered appropriate management for this combined condition. And these are my references for the study. And thank you.
Thank you for your presentation. We can go to the next topic.
Thank you.
Thank you. Our next presentation is by Ms. Kushi Shah on a topic of effect of strengthening and proprioceptive training on internal impingement syndrome in baseball pitcher. A case study. Ms. Kushi Shah.
Yes, hello, am I audible?
Yeah, you're audible. Kindly share your screen.
Is my screen visible? Hello.
Oh, Ms. Kushi, is there an issue regarding sharing the screen?
Can you just give me 1 minute?
Yeah,
Is
sure.
my screen visible now?
Yeah, it's visible.
So, good afternoon, everybody present here. I'm going to present a case study on effect of strengthening and proprioceptive exercises and on internal impingement syndrome in baseball pitcher. So internal impingement of a shoulder is a pathologic condition characterized by excessive or repetitive contact of the greater tuberosity of humeral head with the posterior superior aspect of glenoid when an arm is abducted and externally rotated. Baseball pitchers are athletes who specialized in throwing the baseball with precision and speed. Pitchers utilize a variety of pitches, including fastballs, curveballs, sliders, change-ups, and more, each with its own unique movement and strategy. So, talking about the baseball, it is a game in which there is a pitcher. The main is the pitcher, the batter, and the catcher. So, the distance is 18 meter between the pitcher and the batter. So, the speed at which the pitcher has to throw the ball is 70 to 95 miles per hour, which is quite fast, which occurs in less than a second. So, need of the study is lack of comprehensive evidence, while individual physiotherapy intervention such as proprioceptive neuromuscular facilitation techniques, black burn exercises, and shoulder proprioceptive training that has been studied independently. So there is a paucity of research on their combined effectiveness in addressing internal impingement syndrome. So developing an effective treatment protocol can significantly improve quality of life and functional outcome for these individuals. So aim of the study is to assess the effectiveness of a physiotherapy intervention, including PNF techniques, black burn exercises, and shoulder proprioceptive exercises in reducing shoulder pain and improving quality of life and functional activities of a baseball pitcher. So objective of study is to assess the impact of combined strengthening and proprioceptive training program on pain levels, shoulder function, and pitching performance in baseball pitchers who is diagnosed with internal impingement syndrome. So coming to the hypothesis, null hypothesis, there is no significant effect of strengthening or internal impingement syndrome in baseball pitcher. And second, there is no effect of proprioceptive training on internal impingement syndrome in baseball pitcher. The alternative hypothesis states that there is significant effect of strengthening on internal impingement syndrome in baseball pitcher, and there is effect of proprioceptive training on internal impingement syndrome in baseball pitcher. So materials and methodologies. Coming to the patient description, so the patient, who is a 32-year-old male athlete, presented with a 3-month history of right shoulder pain, which was exacerbated during overhead throwing activities. So clinical examination reveals site consistent with internal impingement syndrome, including pain with provocative test, including the Hawkins-Kennedy and Niels manual. So Hawking-Kennedy test, the purpose of the test is mainly to identify the subacromial or internal impingement syndrome. The technique is patient position is either sitting or standing. Clinician positions is standing adjacent to the patient affected side. One hand is placed under the elbow, the other holds just above the wrist, and the elbow is flexed to 90 degrees. The action that will be performed is shoulder is passively taken into internal rotation. So the positive test will be pain reproduced, which increased during the end range of motion. So in Neil's manual, the patient position is same, that is sitting or standing. The clinician position is standing on affected side. One hand stabilizes the scapula, whereas the other hand grabs the arm below the elbow. So action is the arm is passively elevated into full flexion. So a positive test is a provocation of pain at the end of passive elevation movement. So distinguishing between the primary impingement and internal impingement was done following the positive near sign. So in that, the patient position is either sitting or standing. The clinician position is adjacent, slide behind the patient. Right elbow is flexed to 90 degrees, shoulder is abducted to 90 degrees, and externally rotated of 80 degrees. So at the end, the lower arm apply resistance to external rotation and internal rotation. So positive test is weakness of internal rotation, which indicates that patient has internal impingement syndrome. So the comprehensive physiotherapy intervention plan was devised, incorporative proprioceptive neuromuscular facilitation techniques, black burn exercises targeting scapular stability, and shoulder proprioceptive exercise to enhance joint position, sense, and control. So the SPARDI, that is Joint Pain and Disability Index, which is a validated self-reported outcome measure, was used to assess shoulder pain and disability in individuals with the shoulder pathology, and it was assessed pre and post both interventions. So the study adhered to ethical guidelines ensuring participants' confidentiality, informed consent, and ethical treatment throughout the study period. So proprioceptive neuromuscular facilitation, in which there is diagonal movement of upper extremity. So in D1 flexion, there is diagonally upward and away from body's midline. So in D1 flexion, the shoulder is taken into flexion, adduction, and external rotation. Elbow is in flexion, forearm is in supination, and wrist and fingers are flexed. In D1 extension, that is diagonal downwards and away from body's midline. The shoulder is in extension, abduction, and internal rotation. The elbow is in extension, the forearm is in pronation, and wrist and fingers are extended. So in D2 flexion, there is diagonally upward and away from the body's midline position. So in that, the shoulder would be in flexion, abduction, external rotation. Elbow will be in extension, forearm would be in supination, and wrist and fingers would be extended, as you can see in the image. In D2 extension, the diagonally downwards and across the body's midline. So shoulder is extended, adducted, and internally rotated. Elbow is extended, forearm is pronated, and wrist and fingers are flexed. So coming to the proprioceptive exercises, there was balance with both hands on the floor. The patient positioned his upper extremity on the floor and hold an isometric contraction. Second was balance with one hand clockwise on the wall. The patient moved the affected side of hand, that is the right one, on the wall in clockwise direction. So double arm balance in kneeling push-up position on the balance board. So both the hands are moved in the four direction, that is back, forward, left, and right on the vocal board. So the rotation of on the wall was using a ball. So the patient moves his affected side hand on the wall using a ball. Double arm balance in kneeling push-up position on the foam. So the patient with both the hands were on the foam and were moved forward and backward. Scapular stabilization on the floor with one hand. So patients stand on the floor with affected side hand and hold an isometric contraction. So dynamic stabilization of the exercise of the ball with one hand. The patient moves up affected side hand on the floor with the ball. And these exercises were given 3 days a week for 6 weeks. So Blackburn exercises. These exercises are mainly the one which help to treat patient shoulder pain and regain proper mobility and increased painful movements along with strengthening. So first is prone horizontal abduction, in which the in neutral position the patient lie on the table facing down with arms hanging and palms facing down. So the patient was told to raise arm parallel to the floor. It was then hold for 2 seconds and was then further lowered slowly. So position in second number is prone horizontal abduction, that is in full external rotation. So the patient's position is same as that of before, but the thumbs are rotated upwards, that is full external rotation. So raises arms out slightly in front of shoulder, which is parallel to the floor. The patient was told to hold for 2 seconds and lower slowly. Prone horizontal scaption in neutral. So lie on the table facing downwards with arms hanging and palms facing down. And patient was told to raise the arms at the side but slightly forward by 30 degrees. This also hold the position for 2 seconds and lower slowly. Fourth is prone horizontal scaption in full external rotation. So same position, but the patient raises the arms to side slightly forward by about 30 degrees. This position was also hold for 2 seconds and then lower slowly. Fifth is prone horizontal external rotation, that is lie on the table facing downwards with arms abducted horizontal to side and elbow bent to 90 degree pointing down. So the arms were rotated external. The hold for 2 seconds and then lower slowly. Sixth is prone horizontal extension. The patient position remains same, but the palms will be facing forward. So the patient was told to raise the arm to the horizontal parallel to that of thorax. The patient was told to hold the position for 2 seconds and then lower slowly. The treatment protocol started with a simple warm-up and ended with the cool-down exercises. Then we proceeded with Blackburn exercises, which were given 3 days a week with 10 repetitions for 3 weeks and progress into 20 repetitions from the 4th to 6th week. So the result that we got was the baseline assessment using SPARDI questionnaire indicated that there is significant pain and disability with a total score of 54.6% to 36.15% after 6 weeks, indicating a decrease in pain severity experienced by participant and enhanced functional ability. So the objective measures of shoulder range of motion showed improvement with increased flexibility and decreased restriction observed post-intervention. So the participant exhibited gain in shoulder strength and demonstrated enhanced performance following completion of physiotherapy intervention. The participant reported high level of satisfaction with physiotherapy intervention, indicating perceived improvement in shoulder pain, function as well as athletic performance. So coming to the summary and conclusion. So a 32-year-old male athlete which presented with right shoulder pain was indicative of internal impingement syndrome. Clinical examination confirmed the diagnosis, promoting a comprehensive physiotherapy approach incorporating PNF techniques, Blackburn exercises, and shoulder proprioceptive exercises. The case highlighted the effectiveness of the multimodal physiotherapy approach in addressing shoulder pain in athletes. The utilization of a comprehensive approach incorporating PNF techniques, Blackburn exercises, and shoulder proprioceptive exercises gained significant improvement and achieved emphasizing the importance of addressing both the muscular imbalances as well as proprioceptive deficits for optimal rehabilitation outcomes. So these are the references. Thank you.
Thank you, Ms. Kushi. You have done a study on, this is one case, isn't it? Just one case.
Yes, ma'am, it is just one case because in the sports club which I am going right now, the only one case was affected with, had this.
Okay.
Okay.
And this baseball, is it a game which is played commonly in our country?
No, ma'am, but I go to the sports complex, so that there baseball is there. So there are 16 players. So I have
Okay.
11 players there.
Okay. Thank you.
Thank you.
We can go to the next presentation.
Hello, ma'am. I'm sorry for the inconvenience. I'm ready with my presentation.
Your name, please?
Drashti Kotak.
Okay. Which is your slot number?
My slot was 2 priorly, but due to network issues and I was not able to share the screen.
I'm sorry, may I know to which one you belongs to?
Parul University.
Not
No, I
university.
don't think your name is in this list. Your
I
name is not in this list.
guess you're a participant from hall A, not from the main hall.
Okay, sorry.
Okay, kindly join the breakout room. Our next presentation is by Shilpa S on topic of deep learning models for classifying skin cancer images.
Good afternoon, ma'am. Good afternoon, everyone present here.
Good afternoon.
Yeah. Ma'am, is my screen visible?
No. No, it's not visible.
ಶ್ರೀಶಾ ಸುಮ್ಮನೆ ಇರೋ.
One minute, ma'am. Loading.
Ms. Shilpa, any issue regarding sharing the screen?
Yeah, ma'am. It's loading, ma'am.
So Ms. Shilpa in Zoom chat
Yeah.
box. Oh yeah, now it's fine.
No, not this. Actually, PPT I have to share. Yeah. Now it is visible, ma'am?
No,
No?
no. Once opening the PPT, then you have to share the screen.
Now, ma'am?
Yeah, now it's fine. Keep
Yeah.
it in the full
Yeah,
screen
yeah,
mode and
yeah.
continue with the presentation.
Good afternoon. This is related to deep learning model. This is one neural network model to classify skin cancer image. I'm basically from engineering background, and here I'm developing one model to check whether the given picture or image, whatever we are capturing, that is skin cancer image or not. What I'm planning to do here is because skin cancer is most widespread cancer diagnosed in the world. So we thought of developing a model for computer vision so that the normal person also can understand whether that skin affected area is belongs to the skin cancer image or the normal skin disease like that. For that, I selected one neural network model, that is CNN, convolutional neural network, because the CNN is having multiple advantages like multiple layers. It will do the pooling and all classification it will do. By learning the nonlinear interaction, the model can tell whether the new images are corresponding to the normal benign or the cancer image like that. And one more thing I am using here because this is mainly for some healthcare centers that is there in the rural area like that for that we concentrated this model. And one more thing I adopted here, all the model after creating the model, I will deploy this model in the cloud storage so that the normal smartphone can access that, and through the smartphone the person can detect or person can understand this one given image is belongs to the skin cancer image or not. These three objectives I did. First one is preparing the model by using CNN. Second one is like deploying after preparing that model, deploying that model in the cloud storage, AWS storage I planned for that. Because of this RAM intensive advantages. And the third objective is like extracting that model by using any one smartphone. Here I planned like only the Android phones. Android phones are working now and it is detecting. This is the approach and survey I made based on this. Already we have so many approaches under this, but all the approaches will do only that is one system. We will take some test and train data based on that in the system. It will identify. But the same thing I'm adopting in the cloud storage, and also I'm extracting by using Android phone. This is the main approach of my work, and this is the architecture I'm using. User will capture the data through smartphone or any one application provided by the user. After that, through that application, it will go to the cloud again, because cloud is having all the test data, everything. I took the test data from already existing test this one images only. It will go to the cloud and it will verify cloud through the cloud database. It will start comparing with the data present already existing in the cloud, and then CNN model will perform these three operations: convolutions, pooling, and filtering. Convolutions, pooling, and filtering, through that it will identify whether the given image is affected skin cancer image or normal skin infection kind of. After that, the same image, after evaluating that image, it will send back to the server through server, the cloud will take that, and it will through the cloud the user will get that image with the result. For that, we need supervised learning. Supervised learning is nothing but for each and every image, we have to give the labeling. For testing data as well, we have to give the labeling. Through that labeling only, it will identify. If the labeling is wrong, then even it will predict the wrong answers. For that, labeling is very important here. After that, supervised learning will do the process, then it will give the thing. And the progress of the work, these are the works I did. Current work is the concept of transferring the learning of the classification. The way of the model, like a previous model, only to avoid starting from the scratch, identifying manually or by using biopsy or something. What we are doing, we designed a model, and that model we will adopt in one application, related application. Through that, we are identifying the disease. And the pre-trained model is trained as a large benchmark data set. We used around 10,000 images for testing purpose, training purpose, and 1,000 images for testing purpose. And whatever the new image we are getting, that it will compare with already existing one. And model is designed by using pre-trained model like Inspection V3 and ResNet 152. All these are the frameworks we have. Using that framework, we designed this model. And data set I am using, this is publicly available free data set. ISIC website will provide this data set, but the data set we have to resize it according to our model. My model will work according to 224 cross 224 pixel size. This is a pixel size. Now the same pixel size, we have to convert that image according to this pixel size. Then we will do this. This is not 1,000. This is 10,000 for validation and 5,000 for timing we are using. This is ISIC website only. I downloaded all the images. Like that. Not only this images, any kind of images, first we have to convert that image into this pixel size, then we can use in our model. And these are the results I got. Accuracy, loss, we will produce based on that. Number of samples I took, out of number of samples, 50 samples, 96% of accuracy I am getting, and very less loss I'm getting. This is my result. And also I did this prediction label convolution matrix. This is a true false matrix I did. Only 32 samples I took for this calculation. More than that also we can take. Even one more image I have, one more analysis I have, that is around 256 images like that. This is my result. And also like this, it will predict, and finally it will give pigmented benign or melanoma, what kind of 4 different kinds of images we classified here. All that 4 different images, if it is true, it will display with the label. And this size is 224 pixel size. In the same size, even results we will get. And the ROC curve, that is accuracy. For accuracy, we will prepare this ROC curve. Accuracy, I'm getting around 96%. That is high compared to all existing models present in the market. And also, this is a comparison for recall and precision, everything. 96.11 accuracy I'm getting from my model. And even I deployed and I checked the result through this one Android phone and cloud computing, AWS cloud only I used. It is working, and it is detecting based on the whatever the predefined models I gave. Based on that, it is working. And the conclusion for this deep learning or artificial intelligence nowadays, it is like one vast area, and everywhere in all the fields we are using this deep learning approaches to identify or to predict the diseases, everything. Even this one medical example I took to apply this deep learning approach. According to this comparison, demonstrated supervised diagnosis accuracy may be attained by fine-tuning extremely deep learning CNN convolution neural network only I'm using. And all dermoscopy images I'm using for this experimental purpose. And after this, in the smartphone, it will give along with the label. Along with the label, it will give the result through the smartphone only. Smartphone only, Android phone I'm concentrating here. In Android phone, it will display the result. And these are the references I used. Multiple medical related papers I referred, even some technical papers for AI algorithms, artificial intelligence algorithms. Then I verified like CNN is the best solution for classifying the images. So I selected CNN model. Thank you, ma'am.
Thank you. It's a very nice presentation, something very innovative. And I think you should, are you collaborating with any institute?
Yes, ma'am. I'm working in PES University as an assistant professor.
Bangalore? Or
Yes,
Kuppam?
Bangalore.
Okay, good.
And I'm doing PhD under Jain University.
Okay.
Almost my PhD is also under completion. This is
Okay.
one objective in my PhD.
Okay. So that means you're using something like machine learning, isn't it?
Yes, ma'am. Machine learning algorithm for detecting skin cancer.
Skin cancer. Okay. And how much it is able to predict the skin cancer by just looking at the lesion?
Ma'am, only whatever the website images are available. We are concentrating only available images.
Okay.
All
Have you taken images only from the internet or even from
yes,
page?
ma'am. There's so many websites are available related to that. In that, with the label, so many images are available. Even I am checking from so many hospitals we approached for our research purpose, but I'm not getting proper images.
Yeah,
That's why I'm
you
using
see, it
for
may present differently in another population. We are an Asian population, isn't it?
Yes, yes, ma'am.
You must get some images from patients, no? After getting the consent.
Even hospitals will provide.
Yeah.
Few hospitals will
They
provide.
give you an accurate kind of a picture. Okay.
Yes, ma'am. First, we will prepare the model is ready now for existing images. If the model is working for existing images, then we can try for the new images as well. So we started preparing the model and deploying in the cloud everything, because we don't know what kind of systems everywhere they will use. If we will deploy this model in the cloud, then easily using the smartphone, they can access the data. If they have internet connection, easily they can access the data anywhere in any time.
Okay. So just
So
give
I
collection
thought of
kind of collection
yes,
of lesion photos, is it?
yes, ma'am. That is like photo only. Photo we will take and we will arrange based on my model will work only for 24 class 224 pixel size. In that way, I have to resize that, and even size is limited for that. And after that, we have to labeling that what kind of image we are getting.
Okay. Thank you, madam. We can go to the next presentation.
Thank you, ma'am. Thank you so much.
Thank you, Ms. Shilpa. Our next presentation is by Ms. Santoshi S. on a topic of evaluation of the antimicrobial and antibiofilm activity of Ruta graveolens against Klebsiella pneumoniae. Ms. Santoshi.
Good afternoon, everyone. Am I audible?
You're audible, but you can be a bit loud.
Is it better now?
Yes, please.
Let's start. So, hello, everyone. I am Santoshi S., a research scholar at Jesus Academy of Higher Education Research, Mysore, Karnataka. So, I bring into the conference my background research, which is of evaluation of the antimicrobial and antibiofilm activity of Ruta graveolens against Klebsiella pneumoniae. So here, my objective of the study was biofilm formation. Biofilm formation on medical devices in one of the sources of nosocomial infection that are difficult to treat due to acquired bacterial resistance. The goal of this work is in vitro evaluation of antimicrobial and antioxidant potential of crude ethanolic extraction of Rutagranulins leaves against Klebsiella pneumonia.
What is the common name, madam, of Rutagravulin?
So the common name of Rutagravulin is RUE. Common RUE. RUE.
RUE. Okay, fine,
Yeah,
fine.
I have mentioned here. So it is common RUE. So it is a perennial herb native to the Mediterranean region, but it's also cultivated in various parts of the world for its medicinal and culinary uses. And when it comes to Klebsiella pneumonia, it is one of the gram-negative bacteria. They are commonly found in environment, but also they are as well in ourselves as human. It is in the gastrointestinal tract of humans as well as animals. It can be normal part of healthcare setting, where it can, such as it can affect our pathogenicity, as well as it has a virulence factor. And the major concern where I'm connected here to explain is the antimicrobial resistance. So the antimicrobial resistance of Klebsiella pneumonia is one of the major concern with infection in the increasing prevalence of antibiotic resistance. The bacterium is known for its ability to develop resistance against multiple antibiotics, including carbapenems. They are the last resorts for treating multi-drug resistance infection. So here, Klebsiella is one of the escape pathogen that is listed among WHO's top 3 priority pathogens due to its resistance, as I said. Carbapenems antibiotics over the last few decades. So Klebsiella pneumonia possesses complex, undeciphered signaling mechanism which present in the large biofilms. The entire mass of the bacteria present in the biofilms act as one entity and has a comprehensive and virulent mechanism to infect the host and shield itself from various antibacterial agents, which is a predominant phenomenon seen in drug resistance condition. So the type 3 fimbriae are encoded here. So I've taken genes which are like MRK major fimbriae unit and MRK B, MRK C, and MRK T, and MRK F. So which plays a important role in infectious association with biofilm formation. So as a derivative, at last, rutin being a quantitative derivative exhibits multiple pharmacological activities, including antibacterial, anti-tumor, anti-inflammatory, antidiarrheal, anti-ulcer, anti-mutagenic, myocardial, protecting, vasodilatory, immunomodulator, and hepatoprotective activities. So rutin was my phytocompound chemical, which was concluded as best that can act as an antibiotic resistance. The extraction was in the methodology when it comes. So we took the ethanol extract from the leaves, where it was subjected to, high resolution liquid chromatography and mass spectroscopy to allow the separation of the complex with greater precision and accuracy, and as well identify the abundance of the molecule present in the sample. So, and my major domain where I concentrated was in the computational screening, where my data was taken from NCBI. Excuse me, sorry. No, it was taken from NCBI, where then protein profiling was done. The homology of the protein molecules were inhibited, and the protein profiling was done, and the plot was formed, where we call it as a Raman plot, where it had where it is more than 90% was considered, objective of 90% was considered. Then the data aggregation was taken from PubChem. So where we used protein, where we docked the protein by using Pyrex software, And the complexes were formed and it was subjected to Discovery Studio. So that is one software where we get the hydrogen bonds of the protein profiling done. So where we see the ligand formation of the hydrogen bonds. So higher the ligand formation are the better proteins or the phytocompounds to treat the antibiotic resistance. So where we found five phytocompounds where routine was one of the best phytocompound where it aggregated with we considered its toxicity profile. All these five phytocompounds were subjected to ADME, sorry. So where absorption, distribution, metabolism, and excretion was tested. So evaluation of the properties of major phytocompounds is excuse me, crucial for understanding their toxicity profile and presiding their behavior in vivo culture. So where we found how to culture that all the five phytocompounds got. So we tested their toxicity and we found out best to profile it. So where here, if you see, routine showed the better antimicrobial activity. So at 100 microgram per ml, so concentration against Klebsiella pneumonia. So these are the SEM images where first one is untreated cells, and you can see that against 100 microgram per ml of routine, where the formation of cells are enclosed, they are restricted, where the infection is restricted there. So these are the graphical representation of same, where we have found the results. And when we come again for the biofilm formation in pneumonia is regulated by relative availability of intracellular C-type GMP coordinated by C-type GMP turnover enzymes. So, and sensed by receptor proteins. So when PDE is inactivated or DGC regulated, leading to increased C-type GMP availability, biofilm formation is stimulated. Conversely, when DCG is inactive or PDE upregulated, result is decreased in the C-type GMP concentration. So biofilm formation is inhibited. So the conclusion of my study is routine is a best phytocompound in extraction from rutagravelins, which can act as an antibiotic resistance for Klebsiella pneumonia, indicating a strong antibacterial activity, which can develop into a novel alternative antibiotics also. So I would like to express my thanks to my department, Biotech and Bioinformatics department as JSSCHR for their constant support, and I would like to extend my gratitude for this conference team, AlliedCon, for giving me an opportunity to put up my research here. Thank you.
Thank you. Thank you for your presentation. I think it's back to the organizers now. Who will be presenting next, or have we come to the end of our?
No, ma'am, we are having two more sessions. Our next presenter is Ms. Simran on a topic of exploring the characteristics of potent probiotic bacteria along with prebiotic as a supplement. Ms. Simran?
Yes. My screen is visible?
Not yet. Yeah, now it's fine.
Okay.
I'm sorry to interrupt you.
Now?
I just wanted to know that
Now it is visible?
Yes, it's visible.
Okay, thank you. So, I'm Simran. So, basically, my topic is about exploring the characteristics of potent probiotic bacteria along with the prebiotic as a supplement. So, what we are actually doing is like we are blending the probiotic and prebiotic together to generate one of the food supplement. So, let's go in detail. So here is what are the probiotics one. So probiotic have the capacity to provide the health benefits as compared to when we are look towards the prebiotic. So prebiotics are the one of the having the property or we can say the substrate to grow on a probiotic. So both we get a blend together and get a better result. So as we know nowadays there are the infants facing the severe problem of gastrointestinal problems, like due to the less healthy microbes which is available in the mother milk, or we can say the breast milk. So baby can't get enough amount of good quality food via the breast milk. So after that, what we are doing is we overcome this problem. An effect will be made to formulate a probiotic baby food which have the capacity of a prebiotic together. So let's see the objective in details. So what we are actually doing, we are reviving the bacteria, which is we are collected from the healthy breast milk sample from the healthy mother volunteer in the Anand region. Then reviving it, we diluted it. We can say the serial dilution was made to grow the bacterial cells on mRNA agar plate. Then we check the property like a gram staining, catalase, taste, and all to check for the bacteria that shows the probiotic properties or not. One of the potent probiotic property, which is the screening of probiotic bacteria for a resistance to lower pH. It should be sustained from pH. So for that purpose, we grow our cell culture like we have the two different strain, and one of the shows the resistance to lower pH, like grow the cell culture overnight, then centrifuge it, pell it down, and this sample which we have blend together with the PBS buffer with the pH 2, that is highly acidic condition we provide to our organism to check for its survivability. Then after that, we go for the incubation, and after that, we check for the optical density of it to check whether the bacteria are survive or not. And then after, we go for the tolerance, which is another major property of probiotic bacteria to sustain again bile salt. So for that purpose, we again grow the bacterial culture on 3% bile, or we can set the higher alkaline condition that the organism should survive. Then we go for the centrifugation of it and then incubate that organisms with the higher bile salt. And then in the end broth, we go for the incubation. And after 24 hours, we check after interval of 1 to 4 hours. So we check for the 6 to 20 nanometer to whether it is survive or not. Then after what we actually get, so from we have the two strain, which is the HBM human breast milk 4 and 7 number sample. In that HBM4, which showing the higher acidic tolerance as well as all the 3 hours, it shows the positive result. It will survive in the higher acidic condition as well as HBM7, which is having the bile tolerance capacity, and all of the two strain that having the capacity to survive at 4 hours. And then we check for the gastric and intestinal simulation. It is kind of the artificial condition we created, like in vitro study. So here's the first, we check the gastric simulation, but here we are bind together with the prebiotics like honey. And then what we are doing is in a gastric simulation, we check for the McFarland standard of overnight grown culture. And then after we incubated the gastric juice, that's the gastric juice having the higher acidic condition, we can say, with the same acidic condition with the same as our gut flora have. And then it will serve if we are check if there is survive on the gastric simulation or not by the optical density, or we can say the CFU counts. And then intestinal simulation, which is also done the same as gastric simulation. We are go for the in a intestinal simulation, and that we use the intestinal juice which having the higher alkaline condition same as our intestine. So we go for the check whether it is survive in the both condition or not. So here's the result. We used to have the control, and the control with another only organism we put it in our samples. Then after we having the bind of pre and probiotic. In that result, in case of gastric and intestinal, we found a better result in the HBM7 with the prebiotic, which is honey. So that is a great amount of result that we get. Then here is the result of gastric simulation with probiotics and without probiotics. In case here is showing the HBM4 and HBM7 with the different simulation. And here is the intestinal simulation with and without probiotic one, or one which is having HBM4 without prebiotic, and here is HBM4 with prebiotic. So here is a prebiotic with prebiotic and probiotic that gets a better result as compared to only probiotic. Then we are go for the another property of probiotic and prebiotic, that is the autoaggregation. For that purpose, we grown our culture overnight, then check the aliquot and add the artificial gastric juice to sustain in the artificial condition of gut, and incubate at 37 degrees Celsius for 2 hours. Then centrifuge it, pellet down, and make it two different vial. One just directly go for the optical density because we are having certain formulas to put it at certain values, and the another tube which is again go for the uh, centrifugation process, and then check for the optical density, like before what the cells are having autoaggregation and after what are the cell aggregations having. So in that we are using the different prebiotic samples among all, with the prebiotic sample HBM4 and honey, those pro and prebiotic having the highest amount of autoaggregation as compared to the another one. Then the another, which is the biofilm formation. And for the purpose of biofilm formation, we grown the overnight culture or isolate, and then we are incorporating into the gastric juice and check for the various prebiotics are inoculated like a banana, honey, and all, and in a tryptophan soya broth supplemented with the 0.25% of glucose. Then in a parallel, we set a control also, and then all the tubes are stained after 37 degrees Celsius, 24 hours incubation of this pre and probiotics. All tubes are stained 0.1% saffron in for 20 minutes and observe for the biofilm formation whether it is formed or not. Then after we rinse it with the saline, and then after we resolubilize in glacial acetic acid and measure the optical density. Then after what we get is P1, which is the HBM4, and the prebiotic, which is the honey, that shows the better result as compared to the another one, and P4, which is also showing highest results. So here is the result of biofilm control HBM7 and HBM4. So we took HBM4 as a most potent one. Then we go for the hydrophobicity study. In that also, even get the highest result and highest shows the hydrophobicity study. Then after we also did an antimicrobial study of our cell-free, neutralized supernatant sample, whether it is producing a bacteriocin or not. Bacteriocin is a kind of a peptide. So for that purpose, we having promising effect of HBM4 with the S. aureus, and also for the Pseudomonas aeruginosa, and less effective against the subtilis. So we formulated liquid baby food. We are having 2 into 10 to 7 colony forming units of HBM4, which is most potent one, and 4 gram per liter of mixture of a probiotic, which is honey. Then the blend of these two things with the distilled water that get a liquid baby food that having the higher capacity and most potent for the baby that doesn't having enough good quality amount of food. Then we check for the toxicity study of our liquid baby food against Artemia salina, which is the brine shrimp lethality test. In that, we check for the whether the shrimp which is survive in the condition or not. So in that, we check for the mortality rate. In a negative control, we found a 20% mortality as compared to the test sample, we get a 10% mortality rate. So by that way, we can say it is the best approach to take a HBM4 for the purpose of formation of baby food. And enhance of this property was observed with the supplementation of various prebiotic at a concentration of 5% in the case of gastric transit simulation. In terms of gut adherence property, most efficient result were found with the supplementation of honey that enhances the effect. And bacterial isolates also can produce a bacteriocin, that is small peptide, and having the property may add additional features to this isolates in term of food. We can say the preservation procedure. However, further intense screening concerning molecular identification and in vivo animal studies and is required to explore the isolated organisms. And here is my references. And thank you.
Thank you for your presentation.
Yes,
We
ma'am.
can present. Thank you.
Yes.
Next presentation is by Mr. Ruben, which is a video presentation, ma'am.
Is this the last presentation?
Yes, ma'am, it's the last presentation of the day.
Okay, thank you.
Is that audible, ma'am?
You are audible.
Oh, video is not audible then.
No, I can see the PowerPoint and I can hear you. Who's presenting?
Just give me a moment, ma'am. Is it audible, ma'am?
Yes, yes. Can we start?
Is the video sound is not audible, ma'am?
No, see, actually, you are talking. I can hear you well. But then there's a PPT in the background. So who's operating it?
Actually, it's a video presentation, ma'am. Just give me a moment. I will be.
Oh, okay, I cannot hear it. See, when you're sharing the screen, no, you have to select that share sound also. Just see whether you've done that. Otherwise, you
Yes,
stop.
yes. I
Yeah.
have done that, ma'am. Just give me.
The title of the presentation is Speech by Deaf Individuals while availing speech and hearing services. The authors are myself, Dr. Ruben Burgess, Mr. Karthik,
Oh, I can hear.
Mr. Rajkumar R, Mrs. P, and Mrs. Kesha. And we are from Quality Institute of Speech and Hearing, Mysore. Moving on to the introduction. In India, around 63 million individuals are facing significant auditory impairment, and there are around 8 million people who are deaf. Studies have reported that deaf individuals face 7 times lower health literacy, leading to underdiagnosis and undertreatment. Hence,
See, now I can't hear anything.
View the barriers faced by deaf individuals while availing speech and hearing services includes lack of skilled sign language interpreters, problems further unable to cater the needs of the people. Also, there's a dearth of needed studies focusing on the challenges faced by deaf individuals in speech and hearing clinics or in schools. Aims and objectives. The aim of the study was to assess the barriers faced by deaf individuals while availing speech and hearing services. The objectives of the study is to understand the barriers in terms of communication, psychological, and service related confronted by deaf individuals while availing speech and hearing services. 325 deaf individuals, comprising of 168 males and 158 females, participated in the present study. The age range of the participants were in the age of 18 to 30, with the mean age of 21 years. The survey research design was employed, and the study was conducted in 3 phases. The phase 1 involved questionnaire preparation. The phase 2 involved validation of the questionnaire. And based on the content validation, 25 questions were categorized into 3 parts, that is, communication-related barriers, service-related barriers, and psychological-related barriers. Phase 3 involved field testing the 25-item questionnaire on deaf individuals, and the collected data was subjected to statistical analysis using SPSS 26 version software. The results of the study was very interesting. The data for each question within the 3 domains were analyzed. Each domain had a maximum score of 40 for communication barriers and service-related barriers. For psychological barriers, the maximum score was 20. While comparing the 3 domains, it was found that service-related barriers posed the greatest challenge with a mean score of 35. Communication barriers followed closely with the mean score of 33, and psychological barriers had a lowest score of 70. Moving to the discussion. The study findings are consistent with those of existing literature, which highlight that communication challenges as a central obstacle in the healthcare settings for deaf community. Service access barriers emerged as a significant concern, with participants revealing various obstacles hindering their access to quality speech and hearing services. Nearly 79% of the participants believe that having more materials or videos in sign language at reception or appointment areas would increase awareness of speech and hearing issues. In terms of communication barriers, 78% of the participants reported absence of sign language interpreters, and a substantial portion of the participants expressed the belief that speech and hearing institutes and clinics should take more proactive role in promoting sign language and enhancing service quality. The psychological barriers identified in the study highlight the emotional toll that communication challenges can also can have on deaf individuals' well-being. Approximately 17% of the participants lack confidence in navigating speech and hearing clinics alone, which underscores the vulnerability and discomfort they experience within this environment. Conclusion. The study contributes to the understanding that a multidimensional approach is needed in establishing an inclusive environment for speech and hearing services for deaf individuals. It also highlights the need for speech and hearing professionals to undergo training and sensitizing them to the cultural and communication needs of deaf individuals. In terms of limitations of the study, the participants were from Mysore region of Karnataka. Further samples from different geographical locations across the country must be addressed. Thank you all for your patience.
Thank you, Doctor, for your presentation. I think I hand over the forum back to the organizers.
Thank you, ma'am. Thank you so much for your time and patience for listening all the presentations. I guess there are a few more presentations going in the other hall, ma'am.
Okay.
Yes, ma'am. Once we are done with that, we can conclude our session for today.
Okay, fine.
Thank you.
Thank you.
Thank you.
Ma'am,
Yes.
there's a small correction. In the agenda shared, it is mentioned that my presentation is oral, but I had registered for post presentation. So
Okay.
just update the judges of for the same thing.
Yes, sure. And kindly convey the same to Ms. Gopika so that your certificate will be received as a poster presentation rather than a oral presentation.
Gopika, through WhatsApp, right?
Yes, anything would be working, WhatsApp or email.
Okay. Okay, ma'am. Thank you.
Okay.
So when can we expect the results?
The results will be announced within a week by a mail, and you will be receiving a WhatsApp message too.
Okay. Okay, ma'am. Thank you.
Thank you. I thank all the participants and session chairs of today for giving their valuable time. Thank you all. Everyone can leave the session now.
Thank you, AlightCon, for inviting us as a chairperson to the international conference. Thank you so much, everybody.
Thank you, sir.
Thank you, everyone, for being a part of this program.
Thank you, sir.